It's happening, I think.
Okay.
We got it. But
What's your name? That's nice to meet you.
nice to meet you. Nika. This is Nika, and I'm Konstantina. Yes, I don't know. He asked about us. Okay, amazing.
Please press your page. Then. Let's remember to share our screen.
Yeah.
Maybe we also need to put our live stream in our website.
The live stream.
We accept the basic. So that was they can. I also don't have it actually.
Which one?
Live stream. I really can't find it on.
Yeah, we can. I can do it easily.
Yeah.
Just let's.
I give you my laptop so you can update this.
Yes. I will need my laptop. I can also do it now, actually. And now that everything is fine,
Yeah.
I am sharing this one. Hello.
Hi. Max, is that you?
Yes.
Nice to meet you. Fine. I'm Nika.
Yeah, I believe that we met on Seoul here.
Yeah. It's true.
Yeah.
Very nice to see you.
Slide now.
Okay, yeah.
Because I want to use the same laptop, really. Like
Okay.
this laptop. And the other box. Konstantina, is still live? All good?
Yes. So then we would need to just do the.
Hi.
It's much nicer here. Yeah. So there'll be AC, you know. I'm gonna hang it up here.
Okay, it's not receiving?
No, it's fine because up there in a big hole.
Oh.
It's warm.
Ah.
It's nice, but warm.
So you are in another station? Or?
I'll start downstairs. I got 3 workshops.
Pretty much.
Yeah. So. Yeah.
Did you cover all the workshops in September? So
No, they're really ones.
oh.
We're not covering the AI for compiler.
Okay.
There are more interesting things, like what you do, right? I can see it. Yeah, it's more interesting. More interesting.
Yeah. Not AI for everything, right? Yeah.
That's true.
I'm talking now, like someone like the toilet we need AI to flush.
That's true. Exactly. Maybe there should be a workshop titled AI is not everything.
Yeah.
How much I'm reading this also.
Yeah, exactly.
Exactly.
Now even the papers I get to read, it's like, hey, I like so boring, let the machine do the things and then.
That's true, yeah.
I find so interesting the things that I have to think about it.
Yeah, exactly.
Upload my thinking to a machine.
Is everything okay?
Yes, we are just trying to put the live stream on the website.
Okay, that's good.
Watch live.
Yes, join us live.
John. Let's see if this can work.
Like what? You alone.
I think we got it here. Don't worry.
Thanks, Irene.
Okay, I pushed it, but let's see. I don't know about the aesthetics. I'm gonna maybe need to adjust. It takes some time, right?
Yeah.
That's the Wi-Fi.
Oh, I see the camera also. It's not it's good. Also, I cannot move hands. How you like?
Yeah, I can see me.
You're not moving though.
I can see me in my phone moving.
Oh,
Real time.
I was doing live stream. It takes the picture.
Yeah,
It's fun.
I was, yes, sorry, Nika, you're right.
Hi, hello.
Hi.
Hello.
Hello.
Nika.
Maybe we can do it from my laptop next week, or we can make it.
Yes, I would assume that this didn't work. I mean, I cannot see the Zoom window. Like what is that? It's here, but where is the rest? This one. Okay, I'm confused with this. So what is? Yeah. I'm sorry. Can I get a mic? Yes. Just you have to unmute it, right? I'm testing here. So.
Do you hear me?
Yes.
Where is the clicker? Ah.
The clicker is there. And let me again. I'm gonna do your phone, I think.
Konstantina, can you check the audio again?
I'm hearing.
Still hearing?
Yeah, I have
because
my...
I turned on one of them, I forgot which one.
No, I have my earbuds.
Good.
Stay here.
Mm-hmm. You can go to the GitHub later. Oh, I think it started then.
Yes.
Okay. So we can get started now. Hi. Okay, so we're going to get started today with the third workshop on architecture for health, or Arch4Health. Welcome, everyone. So I'm going to give you a little bit of overview of what are our visions for this workshop and then get started with the first talk. So the point is that the recent biotechnological advances that enable high throughput and low cost and accurate biological data generation. And these are different modes of data, for example, genome sequencing, continuous, wearable sensing, medical imaging, and so on and so forth. And all this wealth of data enables unique opportunities for advancing healthcare in many different ways, such as precision medicine, bedside personalized care, discovering early warning signs of communicable diseases, continuous physiological tracking, and enhanced capabilities for the diagnosis of different diseases, including complex ones. But the challenge that we have, despite all these opportunities, is that efficient analysis of this large-scale biological data poses significant challenges for conventional computing systems in different ways, such as performance and scalability, energy efficiency, privacy and security, and cost effectiveness. And so motivated by all these factors, the goal of Arch4Health initiative is exploring the key computational challenges in health-related applications and the vital role of computer architecture in overcoming them to advance healthcare. So in this initiative, we hold a series of workshops. So this being the third one, we will soon announce the upcoming ones as well. We also try to make the videos of all the talks available online. So you can check the previous ones, as well as I will show the links. And basically, the areas we aim to cover are very broad. Computational biology, which is one of the most data-intensive application domains related to what I mentioned, neuroscience, wearable systems, medical robotics, mental health, medical imaging, agent-based simulation, and many more. Also, I invite you to check the new review paper that Konstantina, Onur, and I recently wrote. It tries to explain different modes of let's say, data that we want to focus on, or like different high-level goals we have and what we have done so far, and also call to action for future. So this is the agenda for today. We will soon get started with Hasindou's talk. And yeah, here are overview of organizers. So I've recently defended my PhD. I am currently a postdoc researcher and lecturer at Safari Research Group at ETH Zurich, which is where I also got my PhD. And I will be in academic job market. So if you know of anything, I'd be happy to talk. Konstantina is a postdoc researcher and lecturer also in the same group. And Onur is a professor at ETH Zurich, leading Safari Research Group, and is our advisor. So you can get more info about today's workshop from this link. And this is a previous workshop which we had in Sydney earlier this year in conjunction with HPCA. And this is the first one which we had several months ago in October 2025 at Micro in Seoul. And as I mentioned, we will have information on the upcoming iterations shortly. So yeah, thank you so much. That was my kind of like brief welcome and overview. And yeah, we can now set up Hasindou.
You.
You send your bio? Did you send your bio? Bio. Yeah. Oh. Okay, good.
Okay, I think we're ready.
You have his bio?
Yeah.
Sure.
Sorry. Use this.
You can let me show you
How to do it.
You just
Good.
maybe you can you start with this?
Maybe here. Can you?
Yeah, we can hear just fine. Good.
Thank you.
Thank you so much. Okay, so we're ready.
Okay, so Hasindou Gamarachi is a senior lecturer at the School of Computer Science and Engineering, UNSW Sydney. He is also a visiting scientist in the Genomic Technologies group at Garvan Institute of Medical Research.
Okay.
Thanks for joining us, Hasindou.
Okay, thank you, everyone. This one, right? Okay.
Yes.
Okay, so just a brief introduction to myself. My background is in computer engineering, not really biology. So I did my bachelor degree in computer engineering in Sri Lanka.
You can use this clicker.
Good. That's useful. And after that, I moved to Australia to do my PhD in bioinformatics, in particular this area called genomics, where I apply computational engineering techniques to make things faster. Currently, I'm a senior lecturer. So for those who aren't familiar with the system, there it's in between assistant lecturer and associate lecturer, you can think of. We have so many levels there. Also a scientist at Garvan Institute of Medical Research, where I try to apply my work in practice. Yeah, so my talk outline is going to be like that, a quick introduction to a DNA sequence analysis, and then I'm going to present you some example works that we have done in this area. So first, let's move on to the background. And the interesting thing is, in the previous talks, like others gave a lot of background. So always my slides used to be very useless because the background. And today I removed them before I thought, okay, everyone would have covered it. And suddenly my talk was moved to the beginning from middle. So I don't have many introduction slides, but I'll try to speak them verbally. If you don't understand anything, let me know, okay? Okay, so DNA sequencing, what it is. So DNA is like the blueprint of life. You may have heard, like every cell has got DNA, which encodes how a living being is, made from the scratch, how it functions, everything is said to be there. Scientists are trying to still reverse engineer how it's working. Successful to a certain degree, not perfect. So it's made up of these 4 types of bases called ACGT. So you can think of it as a large string with these letters. In the human genome, it's like 3 billion bases long, this whole sequence, quite long. And the process of reading this DNA onto the computer, we call it sequencing, okay? And obviously, there are so many applications in DNA sequencing from any field that involves biosciences, from medicine to basic bioscience research. And also, importantly, nowadays you would have heard of this concept called precision medicine, where you look at the DNA sequence and try to predict which drugs are suitable. So that also is quite a useful thing that comes from genome sequencing. Okay, so this is an overview of how DNA sequencing analysis works. So we have blood or some tissue sample, and of course, then there's a lot of wet lab processes that extract the DNA, various chemical steps that actually makes it ready to be sequenced on the sequencer, like shown here. And you can think of the sequencer as a large sensor array that sequence strands of DNA in parallel. Even though I told you the DNA sequence is very long, the chemical processes typically make it broken into tiny pieces. So what we get out of the DNA sequencer is what we call genomic reads. They are not going to be millions of bases long, but maybe in the range of thousands of bases in long read sequencing and hundreds of bases in short read sequencing. So as you can imagine, now we have to go and try to put together all these tiny pieces to form the overall sequence that was broken, like solving a jigsaw puzzle. So that is a very hard problem. Therefore, that is done on computers using various algorithms and software. And depending on the application, there can be different types of pipelines. That's why, like I have shown just an example generic pipeline. So a pipeline is a series of software that runs one after the other. And this software is made up of a number of algorithms and importantly, a number of heuristics to do the job. And this changes depending on the actual use case. But the idea is typically these workflows are run on high performance computers most of the time because of the amount of compute needed. And of course, once you do that, you get a result which could be medically relevant or some bioscience related result, depending on the use case again. So that's the idea behind. So sequencing is followed by sequence analysis. And of course, as computational researchers, that's where we can help things make faster and better. So these are some examples of algorithms used, but I should tell that this is just a snapshot of a few. There are so many types of algorithms, but the most common ones could be like for sequence alignment, you may have heard of Smith-Waterman. That is one of the common algorithms, but that's not the only algorithm. There are so many other heuristics algorithms in this field. So it's a vast field. That's what I wanted to highlight. So like computers had generations, even sequencing devices have had a number of generations. In 1970s, these things called Sanger sequencing came. First generation sequencing, as you can imagine, a huge device and a lot of laborious tasks to sequence. So not very automated. But then the second generation sequencing came in 2000s, which is still in use today, especially because it has been well established, all the algorithms and stuff. So these are also like huge devices and very costly, few $1 million to buy one. So Illumina is one of the second generation sequences, and an important feature here are read lengths are very, very short, around 100 bases to like 200, 300 bases. They are very accurate, like 99.99% accurate, but very short, making it very difficult to be used for certain tasks like genome assembly. And nowadays, there's this third generation sequencing. For example, Oxford Nanopore MinION shown here, which is a portable device. They can generate very long reads. When I say very long, not as large as chromosomes, but few thousands of bases. Sometimes can be even 1 million, 2 million base, the record like largest reads. So there are two types of third generation sequencings at the moment, two major players. One called PacBio, Pacific Biosciences. This Revio device is one of them. And on the other hand, ONT, Oxford Nanopore Technologies, for example, the MinION. So Oxford Nanopore Technologies do this nanopore sequencing technology, and good thing is it's portable. As you can see, it's a USB device. Whereas this one, well, it's probably portable for elephants, 500 kilos. Okay, well, not for human, but this is, of course, you can just take in your pocket. Yeah, but didn't bring one, you know, I was scared of the airport security retaining me, okay? So anyway, this is how nanopore long read sequencing works. So you have the sequencer and you put a thing called a flow cell onto it, okay? So that's a consumable. For every sample, you have to put a new one typically because it just finishes after sequencing for few days. Then on it, you have a like a array of these tiny little pores called nanopores, thousands of them. And what happens is the DNA strands somehow go through the pore like that. I'm not going to detail how it is done, but by biochemical process. So while it is going through that like this, what they measure is the ionic current disruption across the pore, depending on the size of the bases and actually the KM, as you can see, few bases are at a time. The size and other properties, it blocks the ionic current, and you get a picoampere range signal like this. As you can imagine, very, very noisy signal, both in the amplitude domain and warped in the time domain, because you can't perfectly control the speed at which this thing goes through. They have a method to control it using a motor protein, but that's not a perfect way to control it. It still varies. So this noisy signal then can be decoded onto ACGD bases where the biologist would analyze. That's the idea of how nanopore sequencing works. And Oxford Nanopore Technologies is the major player in general, like creating nanopore sequences today. Of course, competitors are coming. So like a Chinese company called BGI recently unveiled this cyclone device. But of course, nanopore has sued them. So yes, obviously. But so anyway, today major player is nanopore sequencing. So the MinION is size of a little Mars bar. On the other hand, you have this large benchtop device called the Promethean, which is size of like a desktop printer. This one is very good for small genomes and those things, whereas this one is suitable for doing like large number of human genomes in parallel. This one, which we have at Garvan Institute, can do 48 human samples in parallel. And then you may have heard of this term called adaptive sampling. If not, I'll tell you what it is. Nanopore sequencing has this interesting feature where you can, get the data while it is going through the pore in real time. And what you can do is on the computer, you can analyze it in real time. And if you find out it to be a read of interest, let's say a particular part of the genome which you are interested, you can let it sequence. Otherwise, you can reject it by sending a command to the sequencer through the API, and it will internally invert the pore voltage that intends to go and spit the read out of the pore. That way you can, release the pore to sequence another DNA strand which is of interest. So this is called adaptive sampling, selective sequencing, all sort of different terms. But as you can imagine, it should be very, very responsive, the algorithms for this. Okay, so now I'm going to talk about a few types of optimizations we can do to, make nanopore sequencing more usable. One of the ways is making it scalable. I'll tell you why. So I told you this Promethean, right? It can do 48 human genomes in parallel. So one genome produces like 1 to 2 terabytes of compressed signal data during a sequence run of 3 days. And as you can imagine, when it is doing in parallel 48 samples at a time, it's going to generate 100 terabytes of data within few days. So that's a large volume of data to process. So imagine in a lab, you have you want to do 1,000 genomes in parallel, 20 benchtop devices like a lab. But then to process, you need the whole country's supercomputer to process that data. So not very scalable at the moment. Things have got better, of course, but still there's a need to make these algorithms more faster to process that volumes of data to, make us possible to use these sequences for large scale cohorts. So that's the need for scalable sequencing. We have worked on a few, areas in parallel. One of the early works we did was for I/O optimizations, where we introduced a new file format. The reason was the Oxford Nanopore Technologies was using this file format called FAST5 based on the hierarchical data format, the HDF file. So we found that most of the processors are bottlenecked because of this format being not able to, access data in parallel. So what we did was we introduced this file format called slow file. So if you are curious why the name, so it was initially started as a joke to push Nanopore to adopt a better format. So we demonstrated using a simple text-based format, you can do much better than FAST5 can do. And it was so fast, but OLT wasn't doing anything. So we thought, okay, let's make a binary format and then released it as slow 5 because FAST5 is taken for a very slow format. So what can we call for a faster one? We just, for a joke, made it slow 5. Anyway, that's how it came. Anyway, with this format, we could demonstrate that analysis that took days on a high performance computer can be done within hours. And also, because of the way the data is stored, the size reduced significantly. And then on top of this, we built a number of software tools and other bioinformatic tools. You can have a look there. And also a number of works that accelerates a lot of different types of methods and so on. And of course, after doing that, Oxford Nanopore said, okay, we are going to change the file format. And then they released this file format called POD5 format based on the modern Apache Arrow format. Well, recently we did a benchmarking just to see how good it is compared to Slow 5. The file size has become now similar, but seems like Slow 5 still have got advantages in performance and the RAM usage. And also it is very simple, means less dependencies. So these are some benchmarks you could see, especially on high performance computing systems. Still Slow 5 reading is like around 8 times faster, but of course not like as dramatic as with Fast 5, which was hundreds of times faster, but still a substantial gain, meaning that like formats that's designed for particular types of data that makes use of the disk structure and the knowledge can perform much better. And also interestingly, the RAM usage remains to be much lower to get that performance. Yeah. And another work that we have been working on recently is called Slorado. The nanopore technologies provides the base caller, which converts the current signals to ACGT bases. So this is done using neural networks. Used to be hidden Markov models, which was giving like 70% accuracy, but they moved on to these neural networks, and now it can give like 99% accuracy. But the software provided by ONT called Dorado only support a particular series of NVIDIA GPUs, those very, very expensive ones, where you like can't buy even nowadays. We have to wait in a queue. So we thought, okay, how can we make it more, affordable? For example, in Australia, there's another supercomputer called Fauci, which has like a lot of AMD GPUs, and we can't use these resources for base calling. So we started a project with AMD, and we developed this in Slorado, and mostly done by, of course, my PhD student, Bonson. And we could make it possible to, provide this fully open source base caller that works not just on NVIDIA GPUs, but also on AMD GPUs. This we could enable base calling on like other types of GPUs than what's already supported by ONT. So let me now walk into portable sequencing. So the Minayan device, as you can see, you can take to anywhere on the earth, and they even have taken outside of the earth and done sequencing. But in those cases, for analysis, they had to send the data across internet to supercomputing centers because the software wasn't there to do the in situ performance. So our goal is to, make it possible to process these amounts of data in a reasonable speed on mobile devices. So we started with one of these software called Minimap, which performs sequence alignment, which actually takes these reads and anchor it to a reference genome. So it has three major steps: seeding, chaining, and followed by the base level alignment. Because it's a human genome, it's like 3 gigabases, you can't search through the whole genome at once. You need to use techniques such as indexes, like hash tables, to find the seeds approximately where they go to. And then we have the process called chaining. We try to find the potential best hits. And finally, we have this base level algorithm, base level alignment algorithm, which does the base level alignment. That's when you get the mapping output. There are so many hardware accelerated out there that covers some of the base level alignment, like Smith-Waterman algorithm. But when we actually benchmark the state-of-the-art long-read sequencing software, we found that also some other researchers also have done and shown the same. The chaining step seemed to take most of the time for long reads, not for short reads, okay? So we thought, okay, let us accelerate this chaining process, the dynamic programming algorithm, using FPGA. So we initially tried what we call HLS, hardware, whatever. So, but the thing was like, yeah, we could get some performance after lot of work, like 30 to 90%. Most of the time was spent on trying to make HLS to do what we really wanted. Yeah, we initially thought because these algorithms are changing very rapidly, it would be good to use HLS because the idea is you give the code and does it. But in theory, it didn't do what we wanted. We had to do a lot of turn knobs here and there are many pragmas. And after all, we could get some performance. Yeah, so. And then, of course, like getting the chaining algorithm itself is not adequate because the data doesn't come from the sky, right? So you need to integrate it to a piece of software if you want someone else to use it. So we went and tried to integrate it. It turned out to be a lot of like work because you can't just like send the data one by one. You have to batch it and do all sort of things to get useful performance. And after integrating, we got like 25% performance improvement for that software. So we attempted to accelerate using hardware. The key challenge, I think that's probably common to everyone, is when you really integrate it to a software pipeline, the speed up you get overall is not as dramatic as the hardware accelerator alone. While a hardware accelerator alone is of course good to show the proof of concept, to actually go and put it in the field, you need to combine it. That's a lot of work. And at the end, if you don't get hundreds of times of performance, it's very hard to convince a user or a biologist to change the way they are working. So some open question that I have in the field, lot of hardware acceleration opportunity, but how do we make it, end to end? And all the corner cases also comes because accelerator, you can assume, okay, this is the data, homogeneous, but in real life, so all those, weird reads are there that will make it crash. So how to get something end to end is an open challenge in the field. And also we explored the use of this RISC-V architecture because you can do extended instructions. That one gave like 2x performance, but of course we didn't fabricate the processor because that's obviously too expensive. So it's a proof of work. So on the other hand, this was something that was a bit closer to like the actual pipeline. We tried this Jetson Kriya board, which is a nice platform, to be honest, because it's not crazily expensive like other FPGA boards out there. We tried to accelerate DTW, which is used for adaptive sampling, and we could get very good performance out there. And we did somewhat integration, but after that we didn't because the software stack for FPGA is so fragile that all those corner cases will eventually come and crash it. So very good as a proof of concept again, but how to make the software stable to work in an actual clinical pipeline is an open challenge. And GPUs is one of the ones we have had good success in integration because you can get some performance and also the tool chains are mature and stable that you can actually plug into a state-of-the-art GPU and you can get performance, the overall performance. So this software, it gave like 3 to 5x performance compared to a multi-core CPU and something that could be run on Jetson platforms. In 2020s, it was very cheap, like 1,000 Australian dollars. But now it's more expensive than a laptop thanks to AI, like $8,000 now. But at that time, it was like a very good thing because no one was talking about GPUs back then. Yeah. And this is something that went into actual, workflows. Like one example is I recently visited those based on invite. The Basel Hospital, they use it during tumor analysis to tumor surgeries to analyze the methylation patterns. So this software we developed. So something we have had like success in end-to-end integration into actual clinical pipeline GPUs. And also, as a proof of concept, we have done some mobile phone related stuff on for like COVID sequencing. Okay, so half an hour, right?
Yeah.
Okay. So now let me go into real-time sequencing. So we discussed about scalable sequencing for increasing the number of samples, and we discussed how to make things portable. And my next topic is how to make stuff real-time. Because unlike second generation sequences, these modern nanopore sequences can stream data while it is sequencing. So you don't have to wait until the end of the run to start processing. But unfortunately, that is how it is done today. You sequence for 3 days and you start the analysis pipeline for another 3 days. So you have like end-to-end time like a week. On the other hand, if we start processing while the sequencer is outputting data, then you could, do the things while it's sequencing and get the end result soon after the sequencing is finished. So that way you can reduce the overall latency by just like pipeline, hiding the fact that it has to anyway do. So it has potential for like diagnostics with rapid results, which is unnecessarily delayed by post-hoc processing. So there's a need for algorithms that do real-time processing. So one of the early works we worked on was called RealFreq, where we tried to do methylation analysis in real time. So while a batch comes, you do it instantly rather than waiting till the end. You get the first batch, you start the analysis, and you somehow store the intermediate results in memory, and then the next batch comes, you update it. So likewise, the person who's doing sequencing will have insights throughout the sequencing run. So that means if the experiment has reached a certain level of accuracy, they can even stop the experiment. So, and as obviously, you don't have to wait toward the end and then process another like few hours or days. You can just get the end result ready here, meaning that the actual real-time nature of the sequencing algorithm is really used here. And of course, we could show that on a desktop computer, we could keep up with the P2 solar flow cell, like all the processing steps. The lines are parallel, which means you can keep up with the data rate of the sequencer. Then I'm going to talk about another approach for genome assembly, which we call Cornato. So apparently this name was given by one of the people at Garvan Institute of Medical Research, Ira Dewson. Apparently there was an advertisement in Australia when he was a kid called "No Boring Bits in Cornato." So the idea of this algorithm is also like, you don't sequence the boring bits which are already, resolved. So he just called, okay, let's call it Cornato. Okay, so we named our tool Cornato. So information is here, of course. I can give you some brief details on how it works. So this is about genome assembly. In genome assembly, you don't have a reference genome to, map the reads one by one. In humans, no problem, right? Because you have the human genome reference, you can align reads to it. But other animals, you may not have a reference. So out of all those reads, you have to somehow stitch the reads together and generate the reference sequence. So that's genome assembly. Unfortunately, I removed that slide thinking someone else would have explained. Same as for mapping. Okay. And also for humans, this is not always redundant. Until you may have heard of the Human Genome Project, right? That started in 1990s and finished in 2003 or something, 2001. That was mainly done using first generation and second generation sequencing for billions of dollars, lot of manual work, and they got the reference genome, but that was only 92% complete. Because there are these very dark regions in the genome, they call repetitive regions, which are not possible to be solved using those tiny second generation short read pieces. So it was only after third generation sequencing matured, they could fill those 8% gaps. And in like 2021, you may have again heard, right? They fully completed the human genome. That's when they resolved that remaining 8%, and you get 100% of the human genome. So now, even for humans, you can, if you want, without relying on the reference, you can just sequence and get the assembly done. There are uses even though there is a reference. For example, some populations can be quite different to the reference genome that is representative out there, right? Especially we realized when we were sequencing indigenous Australians, they have so many structural variants in their genomes compared to the reference genome out there. So reference guided methods do not work that well if you rely on it. So assembling genome from the scratch is still important for humans as well. And also in cancers and certain cases, they can mess up and all things can happen. So assembly can again be useful. So this Cornato approach is to reduce the cost of genome assembly by using this real-time adaptive sampling technique. So here what we do is, instead of sequencing forever using like a number of flow cells, we start a draft assembly using a single flow cell. And then you find out those boring bits, those easy regions which are already resolved, most of them. So, and in the next round, you do the adaptive sampling with that draft assembly and get rid of any reads that are boring. So you only get the interesting dark regions. Not that they are like boring or anything, but just from the algorithm's perspective already resolved, so we call it boring. And then we take that interesting region reads with the draft assembly reads and do the reassembly. So likewise, we iterate and iteratively improve the assembly. And using another additional flow cell, we could get a very nice telomere to telomere assembly. Just to give you some comparison, so this gray color one is the reference genome. That's probably the best you can get. There are 2 lines per each genome. Why? Because the genome is diploid. Every chromosome has got 2 copies. So those 2 are represented there. So in this plot, going towards that side means you get a good assembly. If you are here, that means not good. So if you use a single flow cell, you could see it is quite behind what you would need. But with the Cornato approach, by supplying just one additional flow cell with that real-time method we mentioned, we could move that to this, red and yellow line. And in comparison, if you don't use real-time method and use, all the sequencing, traditional sequencing, you will need like 16 flow cells to get a genome of that quality. So 2 flow cells versus 8 flow cells, of course, 8 times reduced cost. So that's the benefit of this method, and also the time is saved. So that's all the examples I had. And now I should tell all these 3 things: portability, scalability, real time. Even though they look like separate, they have some relationships as well. If you optimize software for scalability, of course, that may improve the portability and real timeness as well. But of course, each problem has its own challenge. Like portable devices have less memory, so we have to optimize for that. Whereas these scalable ones can run on HPCs, which have more memory, but have different types of disk system. So while there are differences, some parts can be reused across the board as well. if you want, of course, more information, it's available in the workshop paper. The link is here. And finally, I should thank all the students, mentors, and collaborators from my institute and Garvan Institute of Medical Research for the help. Didn't put the list because I need like 5 slides. So many of them who like help to make it like implemented. So we have some time for questions.
Are there questions? I have already a lot, but I will.
Okay.
So, questions here. So maybe I can ask you some.
Yeah.
I will give you after. So first, I start with one of them, and then I can go around. Is there any way for Illumina sequencers to change to enable also outputting the reads in real time?
I think the way the technology works, it's probably not possible. The reason is it like do that lightweight sensing on the whole panel at once. So unless the whole sequencing cycle finishes, you can't get the data.
Right.
So that takes a lot of time to mostly the like end of the run.
Okay, so each of those
cycles, they like kind of accumulate the result and then calculate it.
I see.
Yeah. So I think you can't really get real-time information from it.
Understood.
But PacBio, probably you can. But the device, I think, does the analysis internally, so doesn't let us go and mess around with it. So it's provide. That's a good thing is Oxford Nanopore Technologies. They give the APIs and everything bit openly compared to other sequencing technologies. PacBio has fully locked it down. They don't even let us touch the equipment like software or anything. Everything is like a black box to us. Yeah.
Okay. Yeah, interesting. Okay, now to you. Introduce yourself.
Okay. No, I was curious to understand why Oxford Nanopore decided to not select slow 5 and go for pod 5 because so you explained that you presented the slow 5 with very clear advantages with respect to pod 5, and pod 5 came later. So I was curious to know why they are using pod 5.
Mostly there are like few things, right? I mean, technically, yes, slow 5 is quite like good and it works and used by sequencing folks, like by open science community, and it's used very much at the moment. But I think from the company's perspective, they wanted to develop something of their own. That's my hypothesis, because they always do that. They don't really go and adopt like, open ones. They try to build on their own, probably to get things under control to them. Could be a reason. But was open source, but I think they want to tell that they develop. I don't know, I'm hypothesizing, because we few times reached out and we were completely telling, if you pick that arrow file format, you may have those problems. And it turned out we were correct there. It's using so much RAM and the performance was still not that good. But the good thing is the base calling takes some time. So you today don't see that difference in pod 5. Because the base calling anyway takes a bit of time, therefore the IO part doesn't look that. But if you are to improve the base calling to a very fast stage, then IO will again come. So slow. Yeah, exactly. So those things are there. But like, yeah, I think we don't also exactly know how the companies take the decisions, but we believe that's one of the reasons they, and also they said that, yes, like other thing we have is we don't know, like given it's a product from a lab, whether they can rely on it for long term, which is not kind of untrue, right? Because looking at some softwares, depends on the lab funding and things can disappear. So we have managed to maintain it for like now, 5-6 years. So hope to do so. But from the company's perspective, that's one of the reasons they said why they wanted to develop one on their own. So, you can't really rely on the existence of a package in the long term. So looking at how academic funding works, not that untrue as well, right? Yeah, they don't really provide funding to maintain software, at least in Australia.
With AI agents, that would be kind of easier, so that some agents after a while maintains. I'm not sure how advanced that would be.
Yeah, I mean, I have tried with AI. The thing is, it makes things even more difficult to me because I don't know what the hell it did went and do. At least when I do things, I understand exactly what it's doing and I remember it. But if I ask AI to do it, even someone else, right? I don't exactly remember or know what they did. So if I want to later change something, understanding someone else's code is harder than, recalling what I already did. So for me, at least, I find AI gives the illusion of productivity. But in the long run, you have to spend time to learn and remember, because our brains, you can't just transfer things, right? You have to actively do things to learn and remember. So if you spend that, it wouldn't really help in the long run. And the trust is another problem. If you plug something in, how would you know that all the corner cases that is already resolved are not broken? So many things to, be careful when actually putting something that's reliable into production. So yeah, I think it's a long way before AI agents can really, be useful in that context. But that's my opinion.
Okay. Well, first, I like your anger. So yeah, one comment first about the real-time Illumina sequencing. So I'm not sure if it is still possible with NovaSeq, but in the old HiSeq 2000 days, there was a hack around it. So there is a tool called HiLive, and then it was just looking at watching the BCL files and then doing partial base calling. So it was able to do prefix-based real-time analysis. I don't know about NovaSeq 6000. Maybe they blocked it now. So for the slow 5, so back to you, back to your presentation. For the slow 5, is it possible that you can generate the slow 5 files directly from the instrument, or do you rely on pod 5 to slow 5 conversion, which is...
Moment, what we do is we do a real-time conversion to slow 5 on the instrument that's acquiring the software. The reason is we don't have the open access to the instrument software, unfortunately. Minino, which does the instrument control, is a closed source product.
I know, unfortunately, which incurs additional overhead for the conversion.
Yeah, that's
Thanks.
true.
Can you pass it?
Okay. So my question is about what can you do in terms of data? So are there any large public databases which have human, where you can do comparative analysis, and what kind of analysis has been done, and what are some of the challenges there?
Yeah, okay. So there are few repositories such as the SRS, in United States, and then the European ENA, European Nucleotide. So those two are the two biggest repositories for genomics data, and they contain loads of data already archived. But most of them are actually sequence level data, base call data, not really raw data. There are some raw data as well, but given the size, it's not possible for everyone to upload it, and it's very difficult to manage that levels of data anyway. But sequence level data, there's a lot of openly and publicly available data on those repositories. Yeah, some people do those kind of analysis, but one challenge is downloading them takes for a long time. Especially if you are not closer to the servers and if your internet connections are not that great, it's very difficult. And you have to have a lot of buffer space on your thing. You can't, there's no way that you can do remote access, partial access. to those files, the way they are actually stored. But yeah, that's another good thing with Slow 5. It provides region-based access. If you want a particular region, you can grab that through curl rather than having to download the whole file. So that's a useful feature. But the thing is, like, still they are like, you know, whatever file they tarball and then put it. So that capabilities get lost due to the way the infrastructure works. So there are a lot of infrastructure challenges out there as well to make it really easily usable. It's there, but a lot of effort is required to go and access it. And things have got worse in United States recently. So not even getting help tickets answered.
Anybody else?
Maybe I can actually also ask a question, although it's a technical one. Regarding your implementation on the FPGA, I'm wondering, because I also have some students that are working on it. What was the most challenging part in fitting the entire pipeline inside the FPGA? Like, which part was the most demanding in terms of data generated? And how did you decide on an appropriate batching technique to make sure it fits?
So I think it depends on a case-by-case basis. If it's a base call, it would be different. In mapping, at least, what happened was the FPGA we use could, like, you know, in parallel has resources to do multiple reads at once, rather than one at a time. So to enable that happen, we had to, you know, send a batch to it and get a batch. So the existing software, unfortunately, works in a bit different way. So we had to restructure it to match that batching approach.
Okay.
Yeah, that was a challenging part. Yeah.
Okay.
Yeah.
Okay, good to know. Thank you.
But after all, corner cases are the challenges.
Yeah,
because of the stack and the AMD software stack. Lot of documentation issues, as you may know, right? There's documentation and wrong. It's worse than no documentation, right? And sometimes there are like 5 different places that tell 5 different things, and the way to really do it is not any of those. So a lot of time we have to spend to, you know, debug these cases. And when the next name, what is not Vivado, is it changed now? Some other name, right? Yeah, if something else comes, then it doesn't work anymore. And algorithms also have changed. So combining all those things together to actually working thing, probably you need like a whole company working on it. And we obviously don't have that many engineers working on it in an academic setting. Yeah, so I think it's a chicken egg problem. If you want it to be adapted in a clinical setting, it should be reliable and it should be very polished. And the person who's not aware of the details should be able to just put it and use it. The amount of inertia otherwise is too much to move the field in the application setting, because their priority is not the hardware efficiency. Those who are working in a hospital, their like priority is to solve the biological problem. So they would be happy to put some money into it and get more CPUs if that thing is not that reliable and it's not going to get like thousands of times performance. But to get to such a product, even though the potential is there, you need a lot of engineering hours, which is not always supported by academic funding. So it's a chicken egg problem. Yes.
Thank you.
Thank you so much, Asendo, for maybe you can use the mics.
Yes.
For covering very interesting topics. Thanks a lot. So now we have Max. Max Douglas received his PhD degree in computer architecture from the Barcelona Supercomputing Center, BSc, and the Universitat Politècnica de Catalunya, UPC, in 2026. His doctoral research focused on accelerating genomic applications through software, hardware co-design solutions. He has also been a visiting researcher at the University of California, San Diego, and Cornell University.
His broader research interests include computer architecture, domain-specific accelerators, and high-performance computing. Okay, thanks, Max.
Thank you for this warm introduction. And thank you to Jacinto, because now my background will be much lighter. And for sure, his explanation is way better than mine. Here I will talk a little bit of the work we have been doing, especially in my PhD that I recently got. And mainly my topic on the PhD is accelerating sequence alignment from the hardware-software co-design perspective. First of all, I will start with this brief introduction. As you may know, and Jacinto better explained than me, a lot of different scientific fields has been evolved really fastly recently. Especially we have now precision medicine. During the COVID-19 pandemic, we were able to trace in real life the different mutations of the virus, and just recently we get this new data structure called pangenomes that at the end, as Jacinto said, is a way to better represent the diversity among us in a better reference representation. All these new research topics are based on same thing, that is this genome sequencing, that is the process of decoding the DNA sequences from a given organism, can be human, virus, etc. And all these new research topics are nowadays available, since the genome sequence genome technologies has been evolved pretty rapidly. The trend is that we have an exponential increase on this genomic data production. Even this production has overcome the Moore's law, and this genomic production is getting cheaper and cheaper. Now we can sequence an entire human genome for less than $1,000, even less than that. And apart from that, one thing that Jacinto explained is that nowadays with newer technologies, we are able to get longer and longer DNA fragments or reads that at the end will raise some algorithmic problems in the analysis of all this data. And also one of the main bottlenecks on these genomic analysis pipelines is this sequence alignment problem that we will tackle during this presentation. But what is the sequence alignment at the end? Sequence alignment, it is a process of comparing two sequences in order to identify its similarities or differences. Mainly, or traditionally, has been computed using dynamic programming, in which we have a two-dimensional dynamic programming table, in which each of the dimensions represents the two sequences that we want to compare, and each of the elements in the dynamic programming table, it represents the alignment score between two of the subsequences. At the end, each of these dynamic programming elements are computed using the three adjacent elements on the left top and left top, and we have a computational pattern that starts to the left top corner and ends to the bottom right corner, meaning that for computing this sequence alignment problem, we have a quadratic complexity on the sequence length. Apart from that, we have that it is a very regular and parallel computation, since we can compute all the elements in the same anti-diagonal in parallel and totally independent. And here we have a very simple example of one of the scoring schemes, that is the edit distance, that is the simpler one. If we only compute the dynamic programming table, at the bottom right corner, we will have the alignment score between the two sequences. But if we want to know which are the modifications we have to apply into one sequence in order to obtain the other, we have to compute the following step, that is computing this traceback of the optimal path. In this case, this is a linear algorithm. It will be much lighter. However, it's pretty irregular, because each of the steps depends on the last one. And at the end, we will get the CIGAR or alignment that it will show how to convert one sequence into another. The main limitation of this dynamic-based solution of sequence alignment is that we have these scalability limitations in terms of the sequence length. Since we have this quadratic dependency on the sequence length, we'll have, if we want to align two of these very long new sequences that can be up to 1 megabase long, we will have to compute up to 1 tera dynamic programming elements, and we will need to store a dynamic programming table that is up to 4 terabytes of data. Apart from this obvious limitation, we have that each of these dynamic programming elements can be really expensive to compute. We have seen the most simple computation that is edit distance, but even edit distance, we need several arithmetic operations, minimum and maximum operations, and most important, we will need to fetch several elements in order to compute each of the dynamic programming elements. Meaning that this sequence alignment is not only expensive in terms of complexity, but also each element is expensive to compute. There are several state-of-the-art solutions in order to improve the performance of this problem, or there are several algorithms to improve at least the complexity. Some of them are the ASTAR pairwise alignment algorithm and waveform algorithm that at the end they achieve to reduce this quadratic complexity by computing the elements of the dynamic programming table in increasing a score ordering, meaning that when they finish the optimal one, they avoid to compute the elements that are over this optimal score. However, they introduce some irregularity in the computation that increase the complexity on designing new hardware acceleration prototypes for that kind of algorithms. And then we have all the sequence alignment hardware accelerators that mainly what they try is to improve or to improve the performance of computing each of these dynamic programming elements by introducing new custom functional units in order to compute several elements in parallel, or to introduce these functional units that compute one element at a time or in one cycle. However, they tend to lack some flexibility because they are targeting very specific algorithms and use cases, and then they have relatively high area and resources consumption, since they tend to allocate a lot of SRAM memory in order to allocate partial parts of these dynamic programming tables. Then what we are proposing here is a hardware-software co-design called, in this case, QuickETX, that at the end it proposes a new software algorithm called QuickET that tries to reduce this quadratic complexity while maintaining the regularity of the computation of the dynamic programming table, meaning that we will be able to pretty easily use hardware accelerators. And then in the hardware side, we propose a new ISA extension called GMX that accelerates this dynamic programming computation, but rather than accelerating the entire QuickET algorithm, it introduced key building blocks, in this case called dptile, that computes regions of this dynamic programming table, meaning that it can be reused to compute any of these algorithmic variations of this sequence alignment algorithm. Then let's just start on the software side. The main idea of this QuickET algorithm is this bound and align strategy that we split the alignment in two different phases. First of all, we have a bounding step that we will use very aggressive heuristic algorithms in order to fastly identify upper bound of this optimal alignment score. In this case, all these algorithms that we are using are linear in time, meaning that it will not be the bottleneck of this entire computation. And these heuristic algorithms will always give us an upper bound of the optimal score. And then taking this upper bound of the optimal score, here called S hat, we will use it to restrict which part of the dynamic programming table we want to compute to ensure that we find the optimal solution. In this case, both steps are pretty regular in the computation, meaning that we will we can extend this algorithm on hardware accelerators. In this case, Quicket is an edit distance implementation of this bound and align strategy. However, it can be extended to any kind of scoring model, for example, linear gap or gap-affine models. Then the actual implementation of Quicket, we have in the first step Quicket bound that uses the banded and windowed algorithm, both of them linear in the sequence length. It has a linear memory footprint because we don't want to compute the traceback path, and we only care about the last score in the last cell. And these banded and windowed algorithm will always give upper bound of the optimal score, meaning that it can be used on the second step. And then Quicket align is an exact algorithm that at the end, it uses this upper bound to restrict which part of the dynamic programming table we want to compute. Apart for reducing this, the number of cells explored, it retains this very regular computation of the dynamic programming table, meaning that we can use several algorithmic and hardware proposals in order to accelerate the computation. In this case, we are using bit parallel Myers algorithm in order to compute several elements in one word. And apart from that, we are using a SIMD version of this bit parallel Myers in order to improve more the performance. And apart from that, we, thanks to this regularity, we also can use other algorithmic approaches like the Hitchberg algorithm to reduce the memory footprint or the memory complexity of the algorithm, meaning that we will be able to scale to very long read alignments. Okay, then when we have already know how we can reduce this quadratic complexity, we can analyze the performance analysis of Quicket. And at the end, what we saw is that most of the part of the time expended on computing this Quicket algorithm is devoted to compute this dynamic programming table, meaning that it makes sense to produce a specialized hardware to just accelerate the computation of the dynamic programming table. However, as we can see, we have several algorithmic variations of the sequence alignment problem in order to compute this Quicket algorithm, meaning that we cannot rely on a monolithic hardware accelerator to accelerate all of that. Then we need a flexible hardware solution for accelerating this dynamic-based computation. And here we introduce this GMX ISA extension for this scalable, fast, and efficient sequence alignment acceleration. The classical way of computing this dynamic programming alignment is computing each element one at a time. We need to store all these elements in memory in order to reconstruct the traceback. And apart from that, each element is usually encoded in a 32-bit word. And what we propose here is a new ISA extension that compute tiles of this dynamic programming table at a time. In this case, using one simple instruction of, depending on the frequency we want to achieve, of 1 or 2 cycles, we can compute up to 32 by 32 elements at a time, meaning that in one simple instruction we can compute up to 1024 elements. Apart from that, taking advantage of this tiling computation, we are able to only store the edges of these dynamic programming tiles, meaning that we will, on top of improving the performance, we will reduce the memory footprint of the computation. And apart from that, we are using differential encoding to even reduce more the memory footprint. At the end, the microarchitecture of this ISA extension is pretty simple. It will be encapsulated in a functional unit inside the processor pipeline. And inside this functional unit, we have a systolic array of compute cells. On each of these compute cells is implemented with a variation of the bit parallel Myers algorithm tailored to hardware that helps us to even reduce the cost of each element computation. Then now we know how we can accelerate this computation. Let's see how we can introduce it in this Quicket algorithm. As I explained, we compute the original Quicket implementation was based on the bit parallel Myers algorithm that at the end it computes columns of dynamic programming elements at a time. But since it has a very regular computation, we can replace the bit parallel Myers kernel with our GMX ISA instructions. And then with that, we can accelerate all the different algorithms inside Quicket, banded, windowed, and the Hitchberg one. And on top of that, as you can see, since we are only storing the borders of these tiles, we can, on top of the performance improvement, we can reduce the memory footprint of these algorithms. Then let's see which are the results of this software co-design methodology. First of all, let's see the performance results of the Quicket software implementation. Here, if we can compare Quicket to the state-of-the-art sequence alignment algorithms, in this case, EDLib and Wavefront, we can see that for these very long sequences from Oxford Nanopore, we are around 3-4x speedup, computing this edit distance model. And if we take a look on the memory footprint usage of these algorithms, we can see that either EDLib, the big Wavefront and Quicket has a very low memory usage, very reasonable for any kind of computer system, even aligning these very long sequences that can be up to 1 megabase long. And then since we can take advantage of this ISA extension, we have compared the execution of EDLib, the baseline Quicket, and QuicketX, the accelerated version, aligning different data sets from very short data sets to very long data sets. And we have seen that for shorter data sets, Illumina or PacBio, Quicket is around 4 to 6x faster than the baseline software implementation. But if we move to longer data sets or longer alignments, the performance speedups can be up to 21 or 30x. Basically, here we have that in longer alignments, there is more regularity on the computation, and the time devoted to the dynamic programming table computation is larger, and in this case, we can achieve higher speedups. And then if we have done a small analysis on the instruction reduction that we gain with this new ISA extension, and we have seen that more or less it maps with the performance improvements that we have. Although all the other experiments were done in Gen 5 simulations and RTL emulations, in this case, we had the opportunity on fabricating this new ISA extension on an Intel prototype, in this case, using our in-order CPU called Sargantana. And this tape-out was done in Intel 3 technology node. And after all the place and route, we have seen that the ISA extension of GMX is only, it occupies around the 2% of the entire CPU pipeline, meaning that this 2% is a very low area usage, meaning that this ISA extension can be implemented to any kind of CPU, meaning since Sargantana is a very simple in-order core. Then with this chip, we have achieved a maximum frequency of 1.25 GHz. That is not really high, but more than enough to show or to validate the results. And the overall power consumption of this small in-order core is around half a watt. Then we have compared the performance results that we get on this Sargantana core with and without the ISA extensions. And we have seen that this GMX ISA extension on this in-order core improved the Quicket performance around 3x on short reads and up to 20 something x in longer reach. And if you compare this this GMX IC extension on Sargantana compared with a high performance core, in this case AMD Zen 5 core with a very big SIMD pipeline, in this case AVX, we can get half the performance of this high performance core, however, with a much lower power consumption, meaning that the energy efficiency of this small design on longer reach is up to 10 times bigger. Then, concluding a little bit all this work done, the goal was providing a software hardware co-design solution to first reduce the quadratic complexity of this sequence alignment problem, but also maintaining the regularity to provide new hardware solutions to accelerate, to further accelerate the implementation. The key contributions are first on the software side, Quicket, and on the hardware side, the GMX ISA extension, and one contribution is the actual implementation of this ISA extension from this 3 nanometer tape out. And the key result is that we can accelerate up to 2 orders of magnitude the sequence alignment performance compared to the state-of-the-art EdLib CPU implementation. And this small in-order processor with this ASA extension can be up to 10 times more efficient than a high-performance core, in this case, AMD core. And thank you very much for your attention, and I will be pretty happy to respond to any of your questions.
Okay, thank you so much, Max. Do we have questions?
Okay, so I have a question. Can you go to slide 17?
Oh, yeah.
So here my question is, like, perhaps it's not really related to your work, but like here on the performance results, like, do you know why like WFA like consumes a disproportionate amount of memory compared to everything else?
Yeah, the thing is that EdLib, Wavefront, and Quicket, these 3 implementations include the Hitchberg optimization, meaning that it can more or less fix the maximum amount of memory consumption usage. However, Wavefront doesn't include that. Then it needs to store all the intermediate computation in order to reconstruct the traceback. And this is why for these very long sequences, they need as much memory.
Okay, thank you.
More questions? Okay, maybe I can ask you something. It is very interesting to see what you did with alignment. And I wonder, what do you think we can do next? Meaning, considering the entire route mapping pipeline, can we also do these types of flexible approaches of acceleration for other, let's say, steps? Let's say seeding, chaining, etc. And how do you think that would be?
Yes, the main thing is that for seeding, I can imagine that since it's a memory-intensive operation, maybe a CPU is not the best candidate to be used as accelerated seeding. Probably, in this case, near memory accelerator could be a better solution. But for chaining, probably there is a way to introduce some of these techniques there. Maybe a simple ISA extension to accelerate chaining could be useful. Since it's also a quadratic algorithm, maybe there are some of the techniques we have used in Quicket that maybe can be used. And one thing that is more or less useful of this work is that since we are using a ISA extension, the data that is fetched to the CPU, for example, if you are doing the chaining step on the CPU, you already have all the data there. You don't have to move it around to accelerators to compute it. Then maybe if is possible to have several ISA or ISA extension with several instructions for each of these kind of computation. Maybe having all integrated in the CPU reduces the memory transactions.
Yeah, makes sense. Actually, you are also touching a little bit into the second question I had, which was, did you kind of like see any challenges or any insights into integrating this whole alignment into the entire pipeline?
Yes. The thing is that we, apart from this work, we have some detached accelerator or loosely coupled accelerators. And one thing that we face that Jacinto commented is that you have to modify the entire pipelines to do batches, because if not, the memory communication kills the performance. And having the sequence alignment accelerator integrated to the CPU reduces that, because at the end you can just accelerate the actual computational flow without losing this, or without the penalty of this communication. And maybe for accelerating the actual implementation of these genomic pipelines, this is a good approach. Most probably, if you rewrite the entire pipelines, it will be more efficient. But as most of them are implemented or are designed to be executed in CPU, maybe having a ISA extension is one of the best solutions without modifying the entire thing.
Yeah, I understand the trade-off. Yes. Cool. Yeah, thank you so much. More questions? Okay, then I guess we're done with this session. Thank you so much again, Max. Okay, so the next session will start at 11. I think there will be some coffee downstairs, right? Just
Okay, it's true. Yeah.
Yeah.
It was shorter.
Yeah, see you after the coffee break then.
I mean, yeah, in my case, my
Oh my God. It's a very fast genome. Yeah, but that's one of the best ones. Yes. Yeah.
I mean, it was not only this code.
No, it wasn't for this, it was for another thing, and then we had some space in the area.
Yeah, since this process is really, this CPU is really small, and the ISA extension is really small, you could do that.
Oh, yeah, so it's like part of a big tape out. Yes. So the good thing of this tape out was that we have a memory controller, a PCI Express controller, and then the performance is good. If you go with a simple tape out without this very facilities, and you connect an FPGA to access memory, then performance. Yeah, exactly. Why it was competitive with the Xeon core, that was a big surprise.
And even with that, having a very simple core that we didn't thought about the proper
So we're using these microphones to capture the sound for Zoom.
Mhm.
You can just press it here and then attach it somewhere close.
Okay. All right.
Can you speak a bit?
Hey, how
Yeah,
is everybody? All right.
Great.
Yeah.
And then you can also use this change your seat.
You can make it like 2 minutes.
Yeah.
There's also maybe join from the break.
Whenever you're ready, this is for you.
Okay, great.
Okay.
Okay, I guess let everybody come in.
And maybe you can give it a minute. It's okay.
I think Jacqueline sent the Google slides link.
Oh. So there are Google slides. I'm gonna practice how to do it in the lunch. I'm gonna start in a minute. So, you wanna read it? Oh, it's fine. Don't worry about it. A bit heavy. Did follow on its own?
No, no,
Ah,
no.
okay.
I think
Then,
it's, I probably should move.
Oh, yeah. No, come on, don't worry. We will
Okay.
catch it again. Okay, so should we wait a bit more or start?
We can get started.
Okay, great. So I think we can go on. Welcome back, everyone. So we're very happy to introduce Professor Kristina Kant. He's a professor in the Computer and Information Systems Department at Temple University, Philadelphia. Previously, he has worked at the National Science Foundation, Intel, Belcor, Penn State University, and Northwestern University. His current research areas include the integration of logic reasoning with computer vision, intra-body networking, enterprise configuration management, and storage systems. He has previously worked on a very wide range of research topics, including energy management, cyber-physical systems, telecommunication systems, computer architecture, and wireless networks. So I think the floor is yours. Thank you.
Thank you. Thank you for the introduction. Okay, so this is a topic which is quite different from what we heard in the morning. Thank you for your great talk on genomics, but I'm not at all into genomics. But we have been looking at this problem of chronic disease management for last many years, actually. And we've done a bunch of work in it. And I'll introduce that to you. Yeah. This one. This one, right?
Yeah. Can you try again?
Nope. Where is this? Okay, now it worked. Okay, so what is the problem? The problem really is that as people get older, and generally the population in many parts of the world is getting much older. For example, there's almost a crisis in nations like Italy and Japan and Korea and so on. So a lot of old people, including me, I guess. So, and as you get older, you develop these chronic diseases. And there are other reasons for chronic diseases, also increasing pollution and so on. And so here's the statistics that of 65 years or older people have 3 or more chronic illnesses. And there's a question of effective management of these chronic illnesses. And this can be done fairly easily if you can collect the right amount of data and then possibly have a mediation technique. So in many cases, you need sensors which are implanted, not necessarily, but some might be on body, but the other sensors which are better put inside the body, implanted, and then maybe some actuators. And the actuators could be for giving electric shock or giving some drugs like insulin and so on. So one example of this, for example, is in older people, they have a problem with their bladders. And for effective bladder control, you need about sensing from about 4 different places and put it all together and then have a mechanism for bladder control so that the older people can live a more normal life. Okay. So here is a picture. You have a number of implanted nodes, and obviously you want to be able to network these nodes through wireless means so that they can collect all the data and make the decision. So, and also another issue is that when you're going to have these implanted nodes, the biggest problem with these nodes is the energy supply. You can put batteries, but batteries typically are the biggest element in the implant. And also there is a problem of batteries that eventually at some point, maybe 5 years, maybe 10 years later, you have to change the batteries. And you don't want to do that. So we look at not only communications, we also look at wireless energy transfer. So the kind of networks that we have looked at is that you have a node on the wrist, something like a smartwatch. And that acts like a central control center and also an energy provider. And it provides energy to all the nodes inside, it collects the data and so on. Okay, so we need to look at what are the mechanisms by which you can do this efficiently. So that's one. And obviously, one of the things that's extremely important here is that even with the low loss, the amount of energy that you can actually provide to the implanted node will be fairly small. So your whole network has to be extremely energy efficient. In fact, the number one issue here is how to make things as efficient as possible. And secondly, the network has to be adaptable so that it operates for the rest of the patient's life. So we had some NSF grants to explore this reconfigurable interbody networks. And I'll show you some of the results and some of the ongoing work. Okay. Now it turns out that your regular RF communications don't work inside the body very well because of aqueous media. So there are technologies for this. And one of the technologies that we have explored quite extensively is the magnetic resonance communications. Another technology that's quite popular in this space, typically used for very short distances, is ultrasound. Now, MRC, basically the principle is that you have a coil along with the capacitor, which has a resonance frequency. And you have a transmitter, you have a receiver. And if they are both tuned to the same frequency, then there is a very effective energy transfer between them. Now, in the air, of course, there are these, I will not go through the details of the equations, but there are these fairly straightforward equations which tell you how the energy transfer will happen between these two coils. The reason to show you this equation actually is not to really talk about the equation, but that this equation doesn't work at all inside the body. Okay. So everything that this equation predicts is false inside the body. So for example, because of this cosine, the two coils have to be facing each other perfectly for the best energy transfer. If one coil is like this, the other one is like this, then the energy transfer will be almost zero according to this equation, because the cosine will be zero. Not true at all inside the body. So that's why it's important to study this, especially in the body medium. Okay. So here is one simple diagram which shows you that, at a resonance frequency, there is a peak in the induced current on the receive side. And depending upon what is known as a quality factor, this peak could be very sharp or it could be flat. You want it to be very sharp, but if you make it too sharp, then there's a question of what happens if there's a drift. How do you keep it near the peak? Okay. So there's a question of auto-tuning that we have looked at, because we would like the peak to be as sharp as possible, but at the same time be able to keep it tuned. Oh, one other thing here. Typically, these things operate at 13.6 megahertz, which is the RFID frequency. But what we found is we did a frequency sweep. Typically, the transfer is best around 25 to 30 megahertz. Okay. So what's so special about the MRC communications or the electromagnetic communication through the body? Well, the body is extremely heterogeneous medium. And if you look at electric permittivity or the conductivity, it varies quite widely through different organs. Okay? So for example, fat has a relative permittivity of only 25, whereas skin has 285, blood has 210. Okay? And the same deal with the conductivity. That varies quite widely through different organs. And also, it depends on the frequency. So you can imagine that if you were to study the electromagnetic communication through the body, through Maxwell's equations, for example, it's not easy to solve those equations because you have extremely complex surfaces, and there will be, of course, whenever you have two different surfaces with different permittivities, there will be diffraction of the signal. So it's an extremely complex medium. And that's basically the reason the varying permittivity and significant conductivity, that all those results that we have in the air medium don't really work. Okay, so I'll show you some experiments. So these are the three different coils that we designed with different diameters and so on. And we just did a frequency sweep. And this is the kind of diagram that we get here. Okay? So you can see that the channel gain is highest around this 10, 20 megahertz here, typically. But it depends upon a lot of parameters. We, of course, did a whole bunch of human experiments. So that's the hand of my student, actually. So we have a coil there, and we wrapped it real good with the magnetic shielding because we don't want to cheat that we don't want propagation through the air, obviously. We also did some experiments with chickens. Okay, so we bought a whole bunch of chickens from the store and put them together to have a continuous media, essentially, to be able to study. And here are some of the results that you see here. So on the x-axis, you have the distance, and on the y-axis, you have the packet delivery ratio. That is what fraction of the packets do get delivered. So you can see that in the air, it falls off very, very quickly. Because if you look at that equation predicts that the power goes down as a distance to the power 6. Okay. But in the body, you can see that you can go much longer distances, and then it decays much more slowly. I will not talk about this galvanic. Okay. And this is packet delivery ratio as a function of RSSI. Let's just skip that. Okay. So if we can only do the on-body experiments, you might wonder what happens inside the body, but we cannot do any experiments right now. So we looked at the simulations, and there are a number of packages that are available. There's one package called Sim4Life. What they have done is that they have produced a number of very detailed models. For example, I show you. These are all the models that they have. These are models from the cadavers. They did the slice by slice reconstruction of all the organs within the body. And we use this guy, Duke, for most of our experiments. And with this, we can do experiments. We can put transmitters and receivers wherever. And here's the results that we get. So again, on the x-axis is the frequency, and on the y-axis is path loss. And you can see that this is red is simulation, and the blue is measurement. This is on-body, essentially. And the simulation is also for on-body, just to have a one-to-one comparison. And you can see that the simulation works pretty well, essentially. Okay. Also, you see that the path loss actually goes down with the frequency. But it doesn't keep going down all the way. So there are some very weird dependencies, essentially. What you see is that the path loss depends upon how many turns in the coil you have. It obviously depends upon the frequencies. It's minimum path loss at around 20 to 25 megahertz, and so on. And the size of the coil also matters. So everything matters because of all of this very complicated coupling. And here are some other very detailed results that we did with a bunch of volunteers, including myself. And what we wanted to see here is, I don't want to go into the details, what we wanted to see was that how robust is this? Because what should not happen is that if I have something going on, let's say I have some energy transfer from here to somewhere here, and I bend down, and all of a sudden the whole thing changes. And that will, or if I move, and the whole thing changes. So we don't want that. And so we did a lot of these experiments in different positions, movements, and so on. It seems to be fairly robust. Okay. So your Lenz's law, which describes the propagation in in the air doesn't work. Now, because the body is extremely complex media, we cannot really come up with an actual equation based upon the first principles. So instead, we have an empirical characterization. And it goes something like this. And another interesting thing here is that if I have a transmitter here, and if I have only one receiver here, most of the magnetic flux will go there and the reception will be stronger. But instead, if I have two receivers, one here and one there, then the magnetic field, magnetic flux kind of splits. And therefore, these two receivers will receive less. So what happens is that as you increase the number of receivers, the path loss to each individual receiver goes up. Okay. And this is very important for energy transfer. So imagine we have a network of, let's say, 5, 6 nodes. There wouldn't be too many, obviously, in most cases. So if, let's say, I have 7 nodes, which is a case that we looked at, and I transmit energy from this wristwatch, and well, everybody can grab at that energy. Everybody can harvest that energy. That's not a good idea, it turns out. It's a very bad idea. So you really need to select which nodes you want to give the energy to. And then that means, obviously, being computer scientists, that means we have to come up with proper algorithms for doing these kinds of things. Okay. So in terms of the major issues, energy distribution is a major issue. How to do it well. Another issue, as I already talked about, is the drift. Okay. Now, drift can happen, obviously, because of aging and long-term illnesses and so on, because the electromagnetic properties change. There are also short-term changes. So, for example, if you run for a while and your blood pressure goes up, that means the communication through the body actually becomes more efficient. So, so things like that. So physiologic properties actually change the transmission of signal through the body. So things can drift. Now, obviously, you're not going to chase every little change because it's futile. But if, let's say, you are exercising for more than, let's say, 10 minutes or something, your blood pressure is up or something like that, we want this, network to adapt so that it remains near its peak. So that's another one. The third one is energy efficient communication. So the total loss that we find if you have a transmitter here and receivers in different parts of the body, typically will have a 20 dB loss. Okay. So 20 dB loss means that if you send 1 milliwatt of signal, then you get only 10 microwatts. So which means that the amount of energy that's available to these implanted nodes is very, very low, essentially. Even with this such a fantastic medium as body, where you don't have R to the power 6 decay, you have a much slower decay. So this entire network is then ruled by the energy deficiency. Okay? So you have to make things as efficient as possible. Okay? Now, in terms of the data that you're transmitting, typically this is a very small amount of data, a couple of bytes, and you send it every, whatever, 30 seconds. Some cases much more frequently. But as you probably already know, that communication, especially wireless communication, typically is far more expensive than computation. So you want to have a trade-off between communication and computation. And what you want to do is that instead of sending every sample or you can obviously do some filtering. If the things don't change much, then you don't send it. You can do all those simple things. But beyond that, if you can smartly reconstruct the signal and significantly minimize the communication, then that's a huge win. Okay? So that's another thing that we have done some work. We're still working on it. And then the other thing that can happen here is that there might be situations where some of the nodes really run out of energy. Okay? Why would they run out of energy? Well, so if you're sending energy from a smartwatch, that the smartwatch needs to be charged. So there will be periods where your smartwatch is not on, it is being charged. Okay? And if somebody got in the bad habit that at night, they put their smartwatch for charging, in the morning forgot all about it for a long time, then things could run out of energy, essentially. And if things do run out of energy, then you should be able to handle that situation gracefully. That is, at first go into a degraded mode. And if there is not enough energies, you shut down the least important applications first. Eventually, you shut down maybe part of the network. And then when you come back, then there is again a problem of graceful recovery. And this is what happens in large computer systems also, that you don't just shut things down. You shut them down gracefully. And similarly, when you do a disaster recovery, which is what this is essentially, you recover gracefully. Okay. All right. So let's look at some details of the energy distribution problem. We'll not go into too much of detail. So let's start thinking about some very simple mechanisms. Okay. So by the way, here is a network. So OBN is on-body node, which is your smartwatch. Okay. That's the central controller. And then what you will have is, for each chronic disease, typically you will have a separate cluster. Okay. So there's one cluster here, which has two sensors. And then there is a hub node, which collects the data and makes some decision. Maybe it has an actuator also in there. Similarly, for another chronic disease, you have three sensors, a hub, and possibly an actuator. So that's the kind of a structure that this network has. Okay, so in terms of the energy distribution, what's happening is that the OBN is the one which is providing the energy. Everything else is receiving the energy. Okay. So you have to charge your hub and all your sensors, and obviously we are assuming that each one of them has a supercapacitor to hold certain amount of energy. Okay, so one simple thing that you could do is that have a kind of a totally centralized mechanism where each time the OBN decides, okay, which nodes are going to harvest the energy, you tell them, okay, you are invited to harvest the energy, so you connect your circuitry, others disconnect their circuitry, they harvest the energy. So that's one simple mechanism. Now you could have a totally opposite mechanism where each node acts independently. So its probability of harvesting the energy depends upon how low it is on the energy. Okay. So those that are high on energy don't harvest. Those are very low, they have a higher probability, and therefore they get a better chance to harvest the energy. It turns out neither of them, they look very simple. Neither of them works very well. I will not go through the details. You can give you a reference to the papers. Now, then we came up with a simple hybrid mechanism. And if there is adequate energy, then you use centralized, because centralized works well in that regime. And when you're low on energy, then you use the distributed mechanism. So more recently, we came up with a more complicated mechanism, which uses this notion of contextual bandits. And we consider many things in the context as a part of the context. The remaining energy of the node, the recent rate, projected deficit, and so on. So, and I'll show you some results here. Not going into the result. Okay, so what we are doing is that we are every hour we are providing energy for 5 minutes. And then at night, there is a 10-hour blackout when you take off the OBN and you charge it, and therefore there is no energy being supplied. The parameters are number of nodes that are charged together. And there are these thresholds that above 80%, you don't harvest energy, and below 30%, it's an emergency. And we can see that the contextual bandit, which is the purple, it works the best in all the cases. So here you show the mean energy, average number of nodes, which are low on energy. So average number of nodes that are low on energy is the lowest for the bandit, and the mean energy is the highest. So that's one. Okay, let me briefly talk about auto-tuning. So what you want to do is that you want to make sure that your resonance stays near the peak. Okay. Now, you might think that if, let's say, you do the control only on one side, because remember, both the transmitter and receiver could drift, okay? And we don't know how they are drifting. So you could say that, okay, I'm going to do control only on the transmit side and match it to the receiver, or do the control on the receive side and match it to the transmit side. Okay? So one-sided controls. It turns out that the one-sided control doesn't work at all. So we decided to do a two-sided control. Okay, what is a two-sided control? That both the transmitter and the receiver are taking turn to adjust things. And obviously you don't want to have much communication between them, only necessary communication. So both are what they're trying to do is they're trying to adjust the capacitance and trying to see what impact it makes on the current, and accordingly trying to come to a optimal point, essentially. Okay, I will not go through the details. There are some other things you can do. If you're getting closer to your goal, you reduce your step. That's the adaptive. Okay, show you some results. And in the capacitance, it's the same thing. You can have other kinds of drifts. So here what we are showing is that, okay, the transmitter had a big change in the capacitance that it stayed for a while, then it came back to normal. And then at some point, the receiver had a big change in the capacitance and then it came down. So that's one scenario. There is the step scenario. There's a ramp scenario where things are drifting slowly. Obviously, the step scenario is a lot more challenging than the ramp scenario. And it turns out that in all the cases, you're able to recover 99% of the original disturbance. So this works pretty well. Certainly it works better for a ramp scenario. Even if there is a 10% there, it comes back and works pretty nice. Okay. So I want to talk about some other fun stuff here. So in terms of, so we talked about two issues, the energy distribution and the auto tuning. Okay. There are other things that obviously you can do. Let's see, did I miss something here? Yeah, I guess. Okay, so before I talk about this, so in terms of trying to reduce the amount of communication, what you want to do is you want to do a closed loop control, essentially. So what we started out with a Kalman filter on the receiver side. It tries to reconstruct the signal, and every time it gets an actual sample, it calculates the error, sends the error on the other side, which decides, what to do with it. That works pretty well, and I can claim that it works extremely well, except it doesn't. The problem is this, that if you're just talking about the mean error, RMS error, or something like that, anything works pretty well. But that's not important. What is important is that if the signal was like this and then this happened, can you reconstruct that? Okay. Now, if there's a single blip, maybe it doesn't matter. But if it persists for a while, that's very important. So you should be able to reconstruct that very well. So that's what we're working on, and we have some good results, but they're not done yet. So that's another problem. Yet another issue that we talked about was this graceful degradation, graceful recovery. We've done that work, and that's under submission. Okay, some of the other things that you could look at is that providing the energy both externally and internally. You can harvest the energy through the lungs and the hearts, and then how do you integrate the two together, and when do you use what? Okay, so that's another issue. Security is another issue here. Okay. So if you're going to go through the, so remember that this network is supposed to be reconfigurable. So if some attacker comes in, makes a body contact, all of a sudden you can attack this network. So how do you avoid that? Okay, so that's another important problem that we have some solutions for. Another thing that we are looking at now is this mobile ad hoc network. Okay, how do you get a mobile ad hoc network? Right. So here's the deal. We have been working on this, what is known as ICAP, or ingestible capsule. And ingestible capsules have been around for a very long time. But what we want to do is that in addition to all the sensors, we want to put this MRC communication in there. Okay. And then the new thing that we'll look at, so typically, or in fact, everything that we are aware of, you swallow one capsule, goes through your system, monitors the data, and then it gets out. What if we tell the patient to swallow 3 or 4 capsules, let's say every 2 hours or something like that? And these capsules are moving, and they can establish a mobile network between them. And then, of course, they also talk to a stomach patch, some OBN, which could be on the wrist, but preferably on the stomach. So you have that kind of a network. And here, what might happen is because the amount of transmit power that these capsules can provide will be very, very low, right? So you don't expect a very high power signal from there because space limitations and so on, battery limitations. So this truly will be a mobile ad hoc network. That is, if the capsules are close together, then they will be able to communicate. If they are further apart, they may not be able to communicate, essentially. Now, why do we need this? Well, if you can do something like this, then you can monitor the entire GI tract comprehensively, and you know what's happening in different parts of the GI tract, because GI tract, of course, the biochemical processes there are extremely complex, and it would be good to do something like that to be able to understand them well. Okay. So that's what we are working on right now. Another issue that look at is that to what extent can you do AI? Of course, I have to talk about AI, right? So when you talk about these nodes implanted in the body, you can't do much of an AI there, obviously, for obvious reasons. But you could perhaps do some level of AI in the OBN. The question is, what can you do? And then there is a question of, essentially, what can you do offline, collect the data, and do some sort of a meta-learning kind of a thing, that you collect the data and you partially train the model, and you put it in, and then you continue training. So those are some of the things that we're working on. Okay, so I finished before time. Questions?
Thank you so much for the very interesting talk. Questions? Maybe I can get this started first. So you mentioned the various different aspects regarding security and robustness. I wonder whether there would be also some considerations or like new designs needed for, let's say, some electric or magnetic interference with these sensors. Meaning, you mentioned various factors about how aging or illnesses might change the interpretations of these sensors. Would, let's say, the physical location of the body, let's say if it is close to, I don't know, antennas or I don't know, different receivers or I don't know, routers, digital devices in places also impact the readings of these sensors?
Okay, so great question. Okay, so a couple of things here. One is that if it's a static magnetic field, that does not affect it. I mean, in fact, we tested it.
Oh,
We
great.
bought a big magnet from Home Depot and we tested it out. Essentially, it doesn't have any impact. In terms of the electromagnetic interference, so there is the shielding, essentially. So you will have the node itself will be in a magnetically shielded environment, essentially. Okay. So arbitrarily being able to disturb from outside, that ability should be fairly limited.
I see.
Now, obviously, it has to contact the tissue or wherever it's implanted, right? So there is some level of exposure there to external magnetic fields, but from outside. So if you think of it like this, and it's like this, then on the top it will be shielded. And also another thing to consider is r to the power -6. So unless I am, let's say if I'm an attacker, unless I'm actually touching the body, the transmission is going through the air. And therefore, you have this r to the power -6. So it decreases very rapidly. And therefore, again, the chances of interference are much lower. Now, still, obviously, you could have a really, really strong interference. And there might be some issue, but I guess at some point you have to stop worrying about it.
Yeah, or like maybe in that scenario, the person would have other problems
Right.
at that point.
Tell him to go away.
Right. And I was wondering also, depending on changes that might happen due to other factors, let's say exercise that you mentioned, or illnesses, are there ways to, let's say, do some compensation of signals or like adjust them so that kind of like they would read to accurate readings even in those scenarios?
Okay. So here, in terms of communication, we are worried about the resonance, right?
Yeah.
So are you Is that what you're talking about, or are you talking about something else?
For example, if the sensor is reading changes based on, let's say, a physiological condition, can we kind of like compensate or adjust or offset the
The
signal?
sensor itself has an error. It doesn't.
Or like, yeah, some changes in the weights, reading it based on other factors.
Yeah, so you could.
Like some sort of error correction in a sense.
Right. I mean, you could use some of the known methods. You could also use, in some cases, what is known as proxy sensing, which means that if you have one sensor sensing something very specific and there are three other sensors, something related, and there is a relationship. So you can use that in two ways. One way you can use it is for sanity checking, make sure the sensor is working. Okay. The second thing you can do is that if you're low on energy, then you can shut down some sensor and you could deduce some of the results. The third thing that you can do is that you can maybe use it. We have not looked at that. Maybe you can use it for correction.
Great. Yeah, thank you so much. Other questions?
So, go ahead.
Oh, sorry.
But just quickly related to what Mr. just asked. So, would there be some sort of setup where you can reconfigure how each of the implants work? So right now you have distinct roles like the receiver, the transmitter. Could you do something that it can be adjusted if there are changes?
Yes, so we do have this configuration capability through the OBM. So the communication is two ways. So from sensor or the hubs to OBM is the data. The other side is the configuration. Okay, so you can configure it. Now, obviously, you cannot change its location or anything like that, but you can, for example, you can shut down certain sensor, you can test it, you can do those things.
Pushing happens. For example, you could switch one off and make the other one act as
right. You could do that. Sure, sure.
So thanks for the talk. It was very interesting. I wanted to ask something about this last thing that you showed us about the different sensors that are in different parts, let's say, of the gastrointestinal. So these sensors are like homogeneous. They're kind of the same, like they're the same, and they're just put in different places of the tract, let's say.
Yeah, so you are limited by what you can stuff in the iCap. Okay. So iCap, so we have been able to stuff actually up to 6 sensors in addition to the communications and the battery, which is the biggest piece essentially, unfortunately. So it can monitor, for example, the pH inside.
Yeah, basically
it can monitor the microbiota. It can monitor the temperature and a couple of parameters like that. Okay. So the idea would be that if you can simultaneously tell what the pH value is, for example, in the esophagus, in the duodenum, and so on, all at the same time, and look at its temporal evolution, then you can really understand what's going on.
Yeah.
Much better.
Yeah, I was just thinking about, as you said, for example, the pH, because the environment, especially in those like organs, is completely different. So basically, I was questioning whether like you have some fine tuning, let's say, in the way that the sensors are in the stomach or like in the intestine, in order so like for the pH, for example.
Yeah, that's a great idea actually. So we thought about that you could have different types of iCap. So first I swallow iCap number 1, which has certain types of sensor. Then I swallow iCap number 2, which has different types of sensor. That's also possible.
Okay, thank you very much.
I have one question. Thank you, Krishna, for this very nice talk. How much processing do you envision in these sensors currently?
So all that those sensors are currently doing is trying to figure out as a part of this closed loop control, whether to send the sample or not, essentially, more or less. So they're not doing any, I mean, they're doing simple things like checking against a threshold and things like that. So they're not, well, there's another thing that the sensors often need to do. If the sensor is sampling quite fast, you might want to collect a vector, essentially, and then transmit it. So there is that. There's a little bit of storage and then vector transmission. But beyond that, it's really not doing much. Now the hubs, since they are getting the signals from multiple sensors, you can think of it doing something a lot more sophisticated. So, for example, in terms of reducing the amount of communication, one simple-minded way is that for each sensor, you determine when it should transmit or not. The second thing you can do is that you can fuse all these signals and determine that what is best in terms of what or how often each sensor transmit in terms of the ultimate result that you're trying to, ultimate decision that you're trying to make. So we haven't gotten to that, but if you get to that, then it could be fairly complicated.
Thank you.
Okay, thank you again for the very interesting talk. Onur, you are up next.
Nice to meet you. Good to see you. Yeah, a long time.
I think we can just do the introduction meanwhile to Carlos, maybe. So while we're setting up the slides, maybe I can introduce Onur. So Onur is our advisor and also is a professor of at ETH Zurich, so he's leading Safari Research Group. He previously held the William D. and Nancy W. Stecker Early Career Professorship at Carnegie Mellon University. His research interests are in computer architecture, computing systems, hardware security, memory and storage systems, bioinformatics, with a major focus on designing fundamentally energy efficient, high performance, and robust computing systems. He also has many awards, which we then copy in the bio.
Maybe you can remove that. Anyway, it's good to have a full room over here. I think this is one of the more popular workshops. And the smallest room. It's cozy and we can ask questions. No, it's also nice that we have AC here, which is a problem in Europe these days. If you've been around, especially in Zurich. I'm coming from North Carolina, South Carolina. I was there for 2 weeks for DSN and NSK conferences and our group retreat. So it was nice over there. Okay, so I'm between you and lunch, and I have like 300 slides. So you can make a choice between me and lunch. I won't be offended in any way. Don't worry. You can go. You've seen this talk like 100 times. Yeah. Okay, should I start or?
No, not yet.
No, okay.
I'm sorry something happened.
But I mean, the gist of it is I'll talk about genomic analysis. You've seen some talks earlier. I think it's a big area. But I think the workshop, maybe before I start, I'll talk about this architecture for health a little bit, because I think it's important for us to. That's good. Okay. It's important for us to broaden the community in this area, and I really appreciate Krishna for giving a different talk than genomics, because this area is, I think, huge, and there's a lot more to happen. Everybody's like a bit too much focused on AI, in my view, these days. AI is certainly a part of a lot of things, but we really need to be focusing on things that matter for human lives, etc. I think AI will help these things, but we should not be doing only matrix multiplication in our systems. Maybe I'll start with that statement. And there's a lot more than matrix multiplication in the world, right? Hopefully. It's a bit hard to believe that everything can be reduced to matrix multiplication, but okay. Okay, so with that, now that everything is set up, I'll start. So I think system design for bioinformatics is a critical problem. It has large implications, scientific, medical, societal, personal. And I'm going to focus on a very small aspect of a key step in bioinformatics, genome sequence analysis. And even then, I'm going to focus on read mapping and base calling, which are fundamental steps when you start genome analysis. And there are many bottlenecks that exist in manipulating and accessing huge amounts of genomic data during analysis. And there are many recent ideas to exploit read mapping. I'm going to look at some of these. I'm going to give you also my personal journey, but we don't have a lot of time, so we're not going to go into a whole lot of details. This is an area that's quite interesting to me for various reasons. We started working on when I was at Microsoft Research. I have a collaborator who used to be at the University of Washington, Genome Sciences. He decided a long time ago, maybe US is not a great place to live in, and he moved to Turkey. So, and
I am right.
Yeah, it's all about the long term, as you can see, as long as you don't die. Yeah, basically, we brainstormed a lot with them on Mount Rainier, for example, and we worked on some things to accelerate things. But basically, the question is, can we have an embedded device that can perform genome analysis very fast, low latency, low energy? You can embed it in places potentially, and you can answer many questions with that, right? I'm not going to talk about the questions because we don't have time. And this may have a lot of implications because faster, scalable, efficient genome analysis that's done in the field can enable us to understand things, can enable us to predict, can enable us to prevent, and can enable us to do many things that could be helpful for human lives, right? And I think the good news is today we have some sequencers that can help us get there. These are nanopore sequencing devices that Hasunda talked about earlier, I think. And if we can accelerate the genome analysis by these devices, that'd be good. I should also mention that this is not an area, this is an area that's not easy to get into because bioinformatics folks, when they write papers so that they're not understood. Whereas in architecture, I think we try to, at least I try to teach my students to write papers such that it's broadly understood. And I think bridging the gap between two different fields is always difficult, even within computing and electrical engineering. I think here we're talking about biology, systems, etc. So we write some papers that help others hopefully get into the field, and I will mention them over here. You can find more. Okay, so what's the problem basically? The problem we're trying to solve is we have, I think this is a fundamental problem that we have in many places, many systems, but we have very special purpose machines for generating data. And we have, unfortunately, general purpose computers to analyze data. And there's a huge mismatch here because this machine is specialized to generate data with specific properties. This machine is specialized for not much in the end. You need to move the data out of this machine. That's caused a problem. That's slow and energy inefficient, as Krishna mentioned. Computation is much cheaper than communication. And this affects us significantly. And this, the mechanisms to analyze data are not specialized to the data types and models that are generated with the special purpose machine. So these two problems make things much worse. If we can solve these two problems, meaning don't move data, do the processing in situ, and specialize the machine for the data that you generate, then I think you can solve a lot of the data analytics problems. That's what we are going to try to do. And this is genomics. This is one type of sequencer. But this is true. What I said is true, I think, for many data analysis, data generators, like video. Video is a great example of this, I think. I think similar mechanisms that people have proposed, but I'm not going to talk about those. Okay, so I think if you can do this, maybe you can enable the application scope and accuracy to be much better for genomics. And in the end, if you want to do this, you need to think across the stack because it's very difficult to solve all of these two major problems that I talked about by just looking at devices or just looking at algorithms. You really need to think across the stack. Okay, this is the 3-hour version of the talk. I don't know which part I'm going to pick, but I'm going to pick some parts over here. Essentially, I'm not going to talk about
just the last one.
Future opportunity. Okay, yeah. When we're getting too close to lunch, you can tell me that. But I'm going to talk about a few things over here. I'm going to skip these. This is DNA. I think Hasunda gave a nice background over here. The goal in DNA sequencing is to find the complete sequence of ACGT. The challenge, there's no machine that gives you the full output sequence of DNA. Right? All sequencing machines chop DNA into pieces and you need to identify them later. And there are many of these sequencing machines that we have. These are relatively old pictures. Some of these are huge, high throughput. Some of these are small. And they, like we have a paper on nanopore sequencing that analyzes the computational tools to analyze nanopore sequence data, for example. And in the end, these machines produce data with different properties. So even if you look at sequencing, the data that you produce has different properties. So you need to adapt the techniques, algorithms, and architectures that you design to the machine type or type of thing that you do. For example, nanopore sequencers are short. Some sequencers are short, like Illumina, but very accurate. Some other sequencers produce long reads, long fragments of chopped DNA, but they're not as accurate. Okay. So genome sequencing, basically, you chop things into pieces, and you need to identify those fragments so that you can put them together to construct the entire genome. So this is the read mapping problem, essentially. DNA logical looks like this. We turn it into text. We chop it, the sequencer chops it into pieces, and we have a reference genome, and we try to map these pieces to the reference genome so that we can identify what we are dealing with in the end. And all of the analyses after that follow this, right? Okay, the problem is this is expensive, and it becomes more expensive if you're dealing with some more things that you don't know. For example, this is you sample something over here, try to figure out what's lurking here. You may not want to know, depending on where you are. I think the streets in Belfast are a good example. Like if you sample something, who knows what you will get over there, right? So you sample the genetic material, and then you try to, you basically get a bunch of reads that you don't necessarily know what they belong to, and you try to map it to a database of reference genomes, which may be hundreds and hundreds of terabytes, right? Or bigger, actually. And this mapping problem is more expensive, but you're trying to basically identify what species you're dealing with potentially, so that you can ask other questions later on. Okay, this is metagenomics, and I think Nika is going to talk about that at some point. This is an area, for example, in metagenomics, you have so much data that it doesn't even make sense at some point to think about memory even. Today's memories are terabytes, but you need to really think about storing data in storage, for example, or sensing data and doing this extremely quickly. So we need different methods or different ways of thinking when we need this feature. Nika's thesis was on in-storage computation, for example. Okay, so let me go into this relatively fast. Essentially, we're dealing with text, and bioinformatics people love their FASTQ files. I don't care about the names of these things, but essentially what you're dealing with is in this text of reference genome, and you're trying to map the sequence reads somehow. You're trying to identify where these sequences are basically. And you can see that as a human, there's some similarity over here, right? And the similarity is essentially text matching, approximate string matching. And if you do it, without optimization, it's very expensive. So what are we trying to compute? We're trying to compute edit distance, which is the minimum number of edits that are needed to make the read to exactly match the reference. That's the idea. For example, if your read is Netherlands, you're matching Switzerland, you can see that there are some mismatches over here, there's a deletion over here, and there's an insertion over here, right? You can pick your favorite countries in the World Cup these days. Yeah. I don't know if these two are still running, but I have no idea. Okay, yeah, I don't, yeah. No, okay. Okay. So what are the challenges? Basically, computing this edit distance is actually expensive. And doing this for huge genomes is even more expensive, right? So you need to find many mappings of each read because you have a short read. Even long reads are relatively short compared to large genomes. A short read may map to many locations, especially with high-throughput sequence technologies. How do you find all mappings efficiently? You need to tolerate some small variances, meaning each individual is different, and also sometimes you get errors in the machines, sequencer errors. How do you efficiently map each read with up to some number of errors present? And sometimes you want to actually have this E large so that you can actually look at larger variations, right? And that becomes more expensive as it increases. And you need to map each read very fast, because if you want to do these analyses real time quickly, how do you design a much higher performance mapper in the end? So I will not go through this in detail. Traditionally, this has been done. There's been a lot of algorithmic work, dynamic programming algorithms, etc., but these are very expensive. So ideally, you would like to reduce computation as much as possible, and there's some limits to it. And there's a lot of good work that has been done in this area. So naive way is very bad, basically. You go through, for every read, the entire genome and do edit distance computation for every single piece, and that's expensive. And we don't want to do that in the end. The practical way, what people usually do is they essentially, from an index of the genome, so that you can quickly and accurately reduce the search space. And you basically take a fraction, a piece of the read called seed, and you do some sort of sketching, seeding, and access a hash table that gives you potential locations in the reference genome that you should focus on. That's the idea. That enables reduction in computation. That way you can actually reduce the number of expensive computations that you do, which is edit distance computations, as much as possible. So this is all basically mechanisms to get rid of computation, expensive computations, by building an index and seeding and chaining mechanisms. That's the idea over here. So this is what a modern read mapper looks like. And the key is basically you can improve all of the steps basically. Get rid of computation as much as possible, make this faster. WFA, for example, that Mikhail and others have developed, makes this faster, right? So there's actually a lot of things that you can do even in this space, right? Which is very interesting, right? We're not even talking about acceleration yet. How do you actually design algorithms to make these faster? Okay, so we actually looked at read mapping for a while. This is a paper that we wrote some number of years ago. There's been a lot of algorithms developed for essentially all of these. And people are still developing them, which is good. If you're interested, this is a paper that introduces a lot of these and tries to summarize them. It's 111 pages, it's a bit long. The published version is shorter, but the archive version is long. So if you're interested, there's fascinating things that's being done. Okay, I'm going to talk about acceleration a little bit. So because you can accelerate all parts of that read mapping pipeline. I'm going to talk about some specific parts soon. And if you're interested, we have papers that summarize more. So why do you want to accelerate it? Because this may be the bottleneck. This may not be the bottleneck everywhere, but it may be the bottleneck in some sequencers. And if you accelerate this bottleneck, something else will become a bottleneck, and then you accelerate that. That's how things operate, right? That's Amdahl's law in the end. And you can read some papers that describe how this is a bottleneck. I'm not going into that. In the end, it gets you back to peak. I'm going to skip some of these because we don't have time. Okay, let me talk about some ideas of acceleration. I'm going to focus especially on hardware acceleration, specialized architectures. But before that, this is the paper that we did with Can Alkan. Nobody used that mapper, I guess. Why not? You tell me. Basically, that's one of the issues also maybe we should talk about. You can do some good things, and I think this is a good mapper that he developed as a postdoc. Unfortunately, it's very hard to get things adopted unless they're in the mainstream. And this is one of the issues with, in general, multiple communities, right? I'm right, folks. Adoption is something that we need to discuss maybe in these workshops. Okay, but basically, how do you actually make a read mapper faster? And I think that's a general idea. Don't do expensive computations. One expensive computation is alignment, so avoid that as much as possible. Filter fast before you align. That's the idea, basically, we're going to focus on a little bit. So you have a bunch of sequence reads. If you can very quickly decide which ones not to focus on with very fast mechanisms, you can actually expand the expensive computation effort to things that are similar to the reference. Ideally, if you're perfect in this decision making, then you design a better mechanism for edit distance computation, right? It may be different. So, but we're going to be more approximate, meaning some things we're going to make it their own decision. Some things we will think that are dissimilar to the reference. Hopefully, they're not going to be similar to the reference. You don't want to have false negatives over here. But some things that you think are similar to the reference may not be actually similar to the reference. So you may misclassify the reads. But ideally, you would like to focus processing power on things that are really similar to the reference so that you don't waste computation. So we have designed some software mechanisms to do that. We're not going to talk about that. I'm going to talk about this because I like this idea. I think this is a simple idea, bitwise computation, that is applicable to actually going forward some more processing in memory type of systems also. We published this a number of years ago. The idea is if you want to compare two strings, one way of doing that is by using Hamming distance, right? You can see that these two strings are the same. So if you do Hamming distance, you compare each character and you have 8 matches, 0 mismatches. Now the difficulty becomes when a read, let's say the bottom is the read, has an edit. In this case, there's a deletion. Clearly, this Istanbul doesn't match Istanbul. And if you do Hamming distance, assuming you pad something over here with a 0 or whatever, what you get is 3 matches, 5 mismatches. But we can see that this is very similar, right? So how do we actually overcome the effect of this deletion that you really have over here? You do multiple Hamming distance computations. This is one Hamming distance computation. And then you shift the read to the right once, and you do another Hamming distance computation. Okay, this is the first one, 3. This is the second shifted version. You get 5, and then you do an OR over here, sorry, an AND over there. Okay, that's the idea basically. You create shifted versions of the read. Okay, this should go quickly, hopefully. And then Hamming distance computations are simply XORs. These are the XOR results. And then you AND these masks that you generate. And once you AND them, you'll see that there's only one place that doesn't match actually, and everything else is zeros. And if you compare this or count the number of ones over here, if they are less than some threshold, they're similar. If they're greater than some threshold, they're different from each other, significantly different. That's the idea. Does that make sense? So basically, it's shifting, and this you can generalize. This way, you can see that there's 7 matches and 1 mismatch, and you can do more complex computation because you think this is close enough, and you can generalize this. So this is what we essentially do if you actually look at the reference and the query, or read and the reference. These are normally what you look at is the diagonal over here, and you can actually have deletion masks are actually here, and insertion masks are over here. So by shifting right, you cancel the effect of deletions. By shifting left, you cancel the effect of insertions. And you can keep shifting basically to cover for n deletions. If you shift right by n, you're actually really simulating the effect of n deletions. If you're shifting n to the left, you're simulating the effect of n insertions. And you can do Hamming mask on all of those and then do an AND. Okay, that's the idea. And it turns out this is very nicely compatible with SIMD, because SIMD is essentially bitwise. But it's also nicely compatible with FPGA. So we had an implementation of this on FPGA Gatekeeper, which essentially showed very good results with very little hardware cost. I'm not going to go through this again. You can read the paper for more detail, but you get significant speedups. Okay. So this is an example, I think, where you can think about the algorithms differently so that you can improve performance and energy. I will talk about a couple of other things that are related to this. I know you don't have a lot of time. Are we ready yet? Okay. Y'all kind of savvy. Yeah. So we did actually develop some other algorithms. So it turns out doing this at a large scale, like if you have 100 base pair read or 250 base pair read, and if you want to target large error, you start getting not very good results because you're doing approximate computation in the end, right? You're doing shifting and you're doing ANDing. And how do you decide what is similar and what is dissimilar? It becomes polluted because you have too many ones potentially in the end, right? So the idea over here is if you want to do this more accurately, look at smaller windows. Basically. That's the idea. As opposed to looking at a 250 wide read, do it within windows of 4. And that gives you a better approximation in the end. That's the key idea over here that I will not go into more. And you can design it nicely on FPGA. I'll go into this a little bit more because I think this is also a very interesting algorithmic approach that maps nicely to the hardware. And essentially, we're comparing a reference to a read. And the people have observed in the past that correct alignment of the reference or read to the reference is a sequence of non-overlapping long matches. There are some pieces that match, some pieces that don't match. You really want to stay on the sequence of non-overlapping long matches over there. And here we observed that you can reduce this approximate string matching problem to the single net routing problem in the LSI chip layout. And the idea of the single net routing problem is you have some entry points. These are the inputs to the chip. And you have some exit points that you need to route this. And you need to route the signal from the entry to the exit. And there's some blockage points over there. And you want to essentially change the routing paths once you actually reach a blockage point. I like this because it's actually a nice approximation. I'm not going to talk about the key result, but I'll give you how we map this. This is essentially the matrix that I showed you put in a different way. This is the main diagonal. Think about this as the Hamming mask without shifting. These are the first, second, third upper diagonal and lower diagonals. So you can actually construct this based on the Hamming distance computations that I mentioned. And we're looking at a maximum error of 3 over here. That's why we have 3 diagonals upper, 3 diagonals lower. So I constructed this Hamming mask for you. Now, how does this translate into a single chip routing problem? Essentially, you can see the ones as blockages, because 1 means it doesn't match, 0 means it matches. And your goal is to get the longest matches possible, so that you go from entry to the exit. Now this becomes a routing problem. That's the entrance. You start with basically the point that gives you the longest match. You go there, and then there's a blockage, meaning there's a 1, there's a mismatch over here, or there's some deletion or insertion, whatever. Then you switch to the second longest match, but you decrement the blockages that you've seen. So we have maximum 3 blockages allowed. We've seen one over here after we went through this matrix. And then we switch to decremented. We go to the next one. This is the next longest sequence match. It's all zeros. And then we decrement it once we reach the next one. And then the next one is, it could be either one of these actually, but we pick one of those. And then that takes us to the 8th. So now we have a very efficient bitwise method of going from entrance to exit, which is a problem that people have solved in the LSI layout. Again, I'm going through these relatively quickly, but I think hopefully this picture explains the algorithm nicely. The good thing is over here, you don't need to build the entire matrix from the get-go. You can do this on the fly. And the paper discusses how you do that. So this becomes very efficient, and you can put it on an FPGA, and it becomes a very nice filter to filter things out. Again, this is a filter, right? We're not doing perfect computation over here. We're basically trying to figure out whether this read is similar enough to the reference so that we can go and do edit distance computation. If you can do this computation extremely quickly, you can filter out things that don't match. And the things that don't match are actually the things that reach 0 before coming to the exit. That's the idea. Okay, so and we showed very good results with this filter also, and you can incorporate this filter in your favorite read mapper. Just don't use Mr. Fast. I'm just kidding. You can try to use Jalacon's Mr. Fast. Okay, so this is very interesting, I think, and we're very interested in different kind of algorithms to accelerate this mapping. And this is a work where we looked at other text matching algorithms that people have developed in the 1990s. And one of the algorithms that they developed is BiTab. I'm not going to go through this again in detail, but this is a framework that looks at acceleration of multiple genome sequence analysis that it is based on this BiTab algorithm that was published in the 1990s that uses fast and simple bitwise operations. We adapted this to essentially to be very highly parallel, and we also introduced the traceback mechanism, which I will not talk about in detail. And we designed an accelerator for it, a specialized accelerator. It looks like this. The distance computation part generates bit vectors and performs distance calculation, and then there's a traceback part, and they communicate through some SRAM buffers. If you look at the distance calculation part, this is actually a linear systolic array, which is also interesting. It's good to, in my opinion, look at systolic arrays that are currently used for machine learning for the purpose of genomics. I think there's a good area over there. Today's systolic arrays are optimized for clearly matrix multiplication for ML computation, but our systolic array is simpler because we don't do matrix multiplication over here. But thinking about it in the other way, maybe you can use the power of existing TPUs or systolic array-based accelerators for machine learning for genomics. And I think there's an opportunity in that area. But again, this is a systolic array. Nice. The traceback engine is a little bit more complicated, but you can again build it. Still, in the end, you get very simple logic. We work with Intel to look at like how Intel was very interested in doing this sort of genomics acceleration at that point. I don't know what they're interested in these days, but they're survival.
They're interested in survival.
Yeah, survival. Yes, that's right. They're interested in survival. Sure, but yeah. Hopefully they'll be interested in something else other than survival in the future. But basically, you get significant, relatively low area cost for this sort of accelerator. Then we showed that you can accelerate both filtering, like we talked about, and also read alignment in this paper, and also edit distance calculation. And you can see the results in the paper. So we get significant results. compared to both software mechanisms as well as best hardware accelerators at the time. I will also talk about, I'm going to skip this. We have better versions of this that I'm not going to talk about right now, but you can find it online. I'm going to talk about some other problems. This is like what should we look into the future? This area is very interesting. There are clearly classical ways of doing things using linear sequences, but I think the area is also evolving. We're representing genomes not as linear sequences anymore. They're more graphs, right? So these graph representations are necessary because you really need to have a better representative power of the genome in the end. A linear reference genome, for example, doesn't represent the entire human, let's say, population. So we have these graph-based references that are not linear anymore, but you have sequences of bases that are connected to each other in a directed graph. Now, the problem becomes if you have a linear read that you need to map it to a graph right now, and this makes it more complicated. If you have a read that's more like graph representation, it's even more complicated, but we're not going to talk about that. We're going to talk about mapping a linear read to a graph-based reference. And there's been no prior hardware design for this, even though there were software that was certainly designed. So we designed the CGRAM accelerator as an algorithm hardware co-designed that could do sequence to graph mapping, sequence to sequence mapping for both short and long reads. Again, I will not go into a lot of detail because we don't have time, but you can see that this is the graph sequence to graph mapping pipeline. There's some pre-processing that you need to do so that you can construct the graph genome and you can index the graph genome, and then you need to do seeding, filtering, and sequence to graph alignment. CGRAM is a little more complicated than prior accelerators because it needs to take into account essentially a lot of these steps over here, not the offline part. We're not accelerating the offline part, we are accelerating the online steps. So you have an accelerator that's is specialized for minimizer-based seeding mechanisms. So again, I'm not going through the data flow, etc. And then you have an accelerator that's very similar to CGRAM. Very similar to what we have seen earlier with Ghiasi, which generates the bit vectors and performs the traceback. And if you combine them, you can get a significant speed up compared to existing software mechanisms. There's no existing hardware mechanism for sequence to graph alignment, at least at that time, hardware accelerator. So this was the first. But this could also do sequence to sequence alignment because sequence to sequence alignment, linear alignment, is really a subset of sequence to graph alignment. But you need to be careful because if you're optimizing for graphs, you may not be optimizing for linear sequences. So it's good to think about that. This is my PhD student, Anna's last work. She was the last student at CMU who graduated from my group, so you can watch her talk. Okay, thinking back a little bit, I think it's always good to ask the question, can we do better, right? And maybe it's always good to ask the question, are we doing even the right thing? And here I will challenge things a little bit, because all of these accelerators and a lot of the work that is out there in genomic read mapping assumes text, right? You convert some electrical signals into text. But that actually comes at a cost, depending on how you generate that text. So the nanopore sequencing devices, for example, you have some raw reads that are electrical signals. These actually represent ACTGs, and we do base calling. It's essentially a translation mechanism that converts electrical signals to text. And this is very expensive, actually. People use neural networks for this. A lot of them are proprietary to nanopore. Some of them are open source. And then you do read mapping after that. So we talked about this part. But what about this part? That's the idea. Are we doing the right thing in that part? Okay, so let's talk about nanopore. I think Jacinto gave a nice overview of it, so I'll go through this relatively quickly. Essentially, what we're doing is, in a nanopore device, you're passing DNA molecules through a nanoscale hole, and while you're doing that, you're measuring the change in current. And because the bases have different electrochemical structures, the changes that they produce in the current correlates with the type of the base, and you're really trying to figure out how does that electrical current map to the type of base in terms of text. And people train neural network models to do that. This is where AI has been quite successful in genomics, I think, at the let's say device level scale. And you can read more about that in papers. And we've actually designed mechanisms to accelerate these things. This is Gagan's work with AMD. So you can actually use FPGAs to accelerate base calling, all of the AI, interesting stuff. I'm not going to talk about that. But can you do something different? And that's the idea is here, why do we need to convert things to text? Basically, can we do, and it's not our idea, there have been people who tried to work on this, of course, in the past in their papers. Can we get away by just processing electrical signals or performing operations on electrical signals? So I'm going to give you a sequence of works over here, but talk about the basic idea very quickly. So what we have is we have noisy raw signals, we have costly AI models. Can we instead just directly operate noisy raw signals without doing text? So how do you actually map the raw signals to a reference genome that's represented as text? Well, the idea is maybe represent the reference genome in terms of electrical signals. That's the idea. If you get electrical signals here, compare those electrical signals to a reference genome that's represented as electrical signals. But of course, it's easier said than done because there's noise in these signals. How do you represent noise over here, and how do you eliminate noise in this comparison? Right? So what we're trying to do is basically have raw signals. We're going to compare them to raw signals. And the observations that identical molecules generate similar, but not perfectly same raw signals. So people have done similarity estimation based on vector similarity search, nearest neighbor search, and there are a lot of approximate nearest neighbor search accelerators. If you look at architecture conferences, you will find them. Those are good. People should work on them. But maybe this is too costly for large genomes in the end. Can we do something that, okay, we have shown that this is too costly for real genomes, for example, human genome. You cannot do real-time analysis while you're generating the data if you actually do this nearest neighbor search. So what is real-time analysis is also interesting to, I think, look at. So while you're doing sequencing, you do the real-time analysis at the same time while you're generating the sequence data, as opposed to having a sequential analysis that comes after sequencing. Okay, I'm going to skip this. So the basic idea is how do you quickly do the similarity estimation? And the key idea in this raw hash work is represent a set of raw signals as a hash value and compare that hash value to the genome, the same set of signals in the genome represented as hash value. Then the question becomes, how do you actually represent that hash? Because if it's an inflexible hash, you may actually think that these may mismatch, right? Because there's noise. How do you generate the same hash value for similar enough signals with various noise types? Again, I will not go through this in the paper, but the key idea is really adaptive quantized hash-based matching. You need to quantize the signals so that you eliminate some of the noise and pack them and do this adaptively. And this will give you a fast and accurate match. There's still more work that needs to be done in this area to improve this, but that's the key idea over here. Adaptive quantized hash-based matching. And if you do that, you get significant improvements in terms of computation speed that you get compared to the data generation speed of the sequencer. And you can see that with humans, we get even more improvement. And accuracy, it's also accurate, relatively accurate in the cases that we tested. But there's more to do. So we invite the community to build on it. This is John Fortuna's work. He's right now at the University of Maryland as an assistant professor. So he's building on this topic if people are interested. Okay. I don't want to make Jalkan suffer from not eating. But maybe this is a good place to almost stop. But I will mention that there is a whole world where we can do computation. So I've just given you some ideas over here that in the past, but I think genomic data is sensed. So there needs to be processing in the sensors. Genomic data is stored in storage. There needs to be processing over there. Genomic data gets into DRAM at some point. There needs to be processing there. Genomic data goes here. There's processing there. There's SRAM over here. There should be processing over here, right? So today what we do is we're very processor-centric. So we're doing processing only in a fraction of this chip. I think we need to be thinking bigger and doing processing everywhere, right? So if your genomic data is huge, terabytes and terabytes, in my view, it doesn't make sense to move it over here. Why not just let the genomic data sit and do the processing over here as much as possible, unless you really, really have some computational power that's absolutely necessary to be in a compute chip. Sensors also, right? When you're sensing, do as much filtering as possible. Right? So I think we need to think the bigger system such that we optimize computation versus communication. And today we're communicating too much in our systems because the data is moving all around. And I think Nika will talk about this more. But we're examining essentially how to do processing in memory to accelerate genomics. Again, not just genomics as a general idea. You can actually do this processing in memory for any type of processing. And there are clearly a lot of work in this area that shows that you can get significant benefits on many, many applications that are memory intensive. And I think genomics is one application that's extremely memory intensive. I will not keep you further. Maybe this is the last slide I will have, given the requirements. But essentially, we need to design algorithms that fit processing in memory, or we need to design algorithms and architectures together so that we can improve how we design data in the end. This is, in my opinion, one of the biggest challenges of computing today. Not necessarily genomics, but I think genomics suffers a lot from this challenge also. I can talk more about some of the works in this area, but I think we're all hungry. I'm happy to talk further. Juan over there has done a lot of work on processing in memory, so you can talk to him. Okay. Cool.
So any questions?
Who?
Yeah, so when you were talking about comparing those strings, what flashed in my mind is some of the technologies that are used in storage space. For example, for deduplication, ribbon fingerprinting and things like that. And I suppose, I don't know if that has been, this is not my area, so I don't know if it has been explored or not, but that could be useful. I mean, obviously you split it in such a way that it's tolerant of some small changes like you were showing. And that's the ribbon fingerprinting, and then you do the hashing so you can compare things a lot more efficiently. So I'm wondering if you can comment on that, has those technologies been used in this space?
Yeah, that's a great question. Not directly, but people are looking at storage systems in general and making as little as possible modifications, because some of the substrate already exists, right, in storage. You do comparison. Even, for example, built-in self-test that is there in DRAM, it's really checking and comparing to rows, for example. I think a lot of those technologies can be used, but they need to be adapted, as you said, to the type of computation that we're proposing. But I think this lowers the cost, in my view, because a part of this computation is already done in systems for other reasons. We just need to piggyback on that computation and add a little bit more.
Yeah, I assume because there is a lot of redundancy there. So I think some of those techniques will help.
Yeah, absolutely. I think, but I don't know any work that looks at what fraction of it is already done and what minimal thing that we could add. That could be interesting, actually.
Okay.
Thanks.
Thank you. Any other questions?
Let's not make Jalkan late for it.
Okay, I think you scared them.
The first time he finished on time.
See you after the lunch.
Okay, sure.
Are they muted or not? They're all on. Everything is on. So I can use this right now quickly. This is also on. I just introduce you. Maybe we can wait 1 minute or maybe we can start. Should we wait a minute? Or
What is it?
we can also wait 1 minute, 1 more minute.
Yeah, let's wait 1 more minute.
Yeah, sure.
Good idea.
What teams are playing today? What teams are playing today?
Spain, Portugal.
Oh wow. Okay. You should
Very close by, right?
What?
Very close by countries.
Yeah, very strong. Yeah, nice. And we have fun, so it will be
Close
interesting.
by.
They're in the same area, right? Geographically? No?
Still a few kilometers away, Spain and Portugal from here. I mean, the two. Okay, the two thought neighboring countries, yeah.
Okay, Konstantina, okay. Good. Okay.
Still close from here. Considering
On
that
the global scale, yes.
Okay. So I get started. Bertil Schmidt is a full professor at the Institute of Computer Science at Johannes Gutenberg University, JGU, Mainz. Prior to that, he was faculty member at Nanyang Technological University, NTU, in Singapore, and the University of New South Wales. Yes.
Told you about
it. His research group has designed and implemented a variety of massively parallel algorithms and tools focusing on the analysis of larger scale datasets on numerous platforms, including GPUs, FPGAs, supercomputers, and systolic arrays. Thank you so much, and we can get started.
Well, thanks. Thanks for inviting me to give a presentation here today. In my talk, I want to talk a bit about key challenges in accelerating omics pipelines, mainly on GPUs. And I will focus on 2 case studies that we developed in my group. One is accelerated protein structure prediction. The other one is untargeted food metagenomics. Okay. Is it on? You switch this on and this off.
just need maybe some.
Yeah. Okay. Okay. So using these accelerators in this area is motivated by continuous breakthroughs in these high throughput technologies. You saw that in several of the other talks. Genomic sequencing, for example, many different sequencing machines are around producing tons of data and becoming cheaper and cheaper, so getting more frequently used in practice. And this also means that when you just use conventional CPUs, for example, often not able to keep up with massive amounts of sequencing data. Of course, there are also other data areas, but here I mainly focus today, not only, but mainly on sequencing data. So we already talked about this, like Onur also in this talk. So the workloads that we have in this area are quite different from the traditional HPC workloads for, okay, matrix multiplication was mentioned. Yeah, matrix multiplication was mentioned. So we have often different workloads and different algorithms from streaming, sampling, indexing, bit parallelism, for example, you saw that in the previous slide. Also AI, of course, and typical workloads, alignment, assembly, and so on, K-mer counting. So quite different workloads than just traditional flops. Yeah, often you just count flops. And this is often less used in these workloads in omics sciences. So one accelerator that is now frequently used are GPUs, and we are entering an age of computation where GPU performance or GPUs are getting more and more powerful. Here I have 3 recent generations from NVIDIA GPUs, Ampere, Hopper, Blackwell, transistor counts. You see it's rapidly increasing, motivated also by the growth of AI models. Yeah, you see a massive growth of AI models, which again leads to evolution of GPUs rapidly increasing. And so this is really an unprecedented amount of growth. For example, here also NVIDIA capitalization growth from 2022 to 25. Yeah? See here? Don't even know what number is here, $4 trillion. And this is actually fastest growth of a company in human history, fastest growth in human history. And looking at the architecture, GPU architecture itself, also rapidly developing, and typical accelerator contain massive amount of parallelism, in addition to also specialized units, as we will see in the next few slides. Specialized units for different tasks. And we have NVIDIA GPUs, AMD GPUs, but also other type of architectures that are evolving. Onur mentioned some of those in the previous talk. Looking in the detail of GPU architecture, we have different types of computational units. Looking at, this is NVIDIA, AMD GPUs look similar. GPU itself contains a large amount of so-called SMs here, streaming multiprocessors, which can be viewed as like independent, more or less independent computational units that share some larger memory. And within each streaming multiprocessor, we have different units. For example, CUDA cores. These are more or less like general purpose cores. Then we have more specialized cores like tensor cores, which are optimized for the already mentioned matrix multiplication. Then there are also other cores, so-called ray tracing cores. Ray tracing cores are also quite interesting. With ray tracing cores, you can do accelerated intersection computation between a scene of triangles and rays. Okay, you can shoot huge number of rays in parallel, and they compute an intersection, the nearest intersection in the scene. Most of the talk I will use the general CUDA cores. We also use a little bit, not much, of the tensor cores for matrix multiplication. And what I'm not talking about, but what we also have in our group is also important for bioinformatics things. So these ray tracing cores look first like you cannot really use them a lot, but you can also redesign problems in databases, which are also important for bioinformatics, to use those ray tracing cores. I will mainly focus here on CUDA cores. When we use these GPUs for omics pipelines, what are typical challenges? First of all, when we design our algorithms, we need both combination of fine-grained and coarse-grained parallelism, like single instruction, multiple data parallelism, and also more coarse-grained MIMD parallelism. And we need to deal with data growth. This means that we need to use techniques to handle this data growth, to fit often in limited amount of memories. Can be like data reduction techniques or also partitioning techniques. Very important when you use GPUs efficiently are memory hierarchy optimizations, because compute power is very high and you need to get data in at least at the same speed as you do computation, unless otherwise you have a data transfer bottleneck or data access bottleneck, and there are many possible optimizations there, ranging from shared memory, coalesce accesses, kernel fusion, and so on. Large amount of those. What are people always also concerned is energy consumption using these big GPU accelerated machines. So we would like to optimize performance per watt. And overall, what I also will talk about, when you just have an algorithm or pipeline itself, just doing a straightforward parallelization of the existing pipeline is often not sufficient. You often need to redesign how the whole thing works. Yeah. So you need a tight integration between algorithm design and hardware capabilities, which in turn requires to often redesign the problem originally maybe for CPUs to GPUs. So looking at the hardware bit specification, many of you might know that CPUs versus GPUs, we have many more computational units on GPUs. Memory bandwidth is much higher, but memory size, main memory size, much smaller. So we now look at two case studies. The first is accelerated protein structure prediction. And this is using a tool we have recently developed called MM62 GPU. And the second case study is accelerated untargeted food metagenomics with a tool called AFS Metacache. Yeah. So let's start with the protein structure prediction. So in this protein structure prediction problem, it's around many decades old. People have looked at this from different algorithmic perspectives. We input a sequence, a protein sequence. And from this protein sequence, we want to output the atomic three-dimensional structure of the input sequence. So that has been studied for many years, and there always was or has been or still is a competition where these different algorithms are evaluated in a competition. And this competition is called CASP, Critical Assessment of Structure Prediction. And there's a so-called global distance test that measures how good your predictions are between 0 and 100. Yeah? Between 0 and 100. And people said 60 is quite okay. 90, if you get a score of 90 on average, is as good as experimental structure. So that has been done over many years. And over many years, the top algorithms hovered around 30 to 40. Yeah, so very difficult to solve this problem with conventional approaches. And then AlphaFold came. First version already reached 60, and then AlphaFold 2 close to 90. That was already a few years ago. So now, of course, still better. But anyway, with this tool, you can now basically predict the structure of an input sequence. So that is used heavily in practice in all of the labs around the world. And this is a very important problem in this life science area. And that's why it got awarded the Nobel Prize in Chemistry in 24. So when you look at the AlphaFold algorithm itself, it consists of two steps. We have a neural network. Let's say this is already trained. So we only look at now using AlphaFold 2 for inference. We have a neural network here. Before we have the neural network, we have another step, which is called the MSA step. It's for multiple sequence alignment. In this multiple sequence alignment step, following happens. You have the input protein sequence you want to predict, and you have a database, here SequenceDB, a big database of other protein sequences. And then you do something called homology search, and this step now finds the most similar sequences to the input sequence. This is displayed here. So you have one sequence, and you search the database, and it outputs very similar sequences, and you put them in a so-called multiple sequence alignment. And this multiple sequence alignment is represented as a matrix. So at every position here, you get a frequency vector. Yeah, it's called a position-specific scoring matrix. And then you use the scoring matrix to search the database again and refine. It's kind of an expectation maximization algorithm. Refine your matrix. Yeah. Often around 3 steps are used for the database search. And then this multiple sequence alignment, SPSSM, is input to the neural network, and then the neural network will come out with a 3-dimensional structure. So we were looking at accelerating this MSA step. Why is this? When you look at the runtime of this pipeline here, of the 2 steps on a CPU, the MSA step takes much longer than the inference step on the neural network. So we were looking at this acceleration of MSA step, and the problem is, as I mentioned before, you have an input sequence, for example, this input sequence, and then you have a huge database, let's say billions of sequences. And you compare the input sequence to all these other sequences, find a number, so hundreds or thousands of most similar sequences in this database called homology search. So these databases of proteins also rapidly growing. And this means, as you already saw on the previous slide, traditional CPU processing power takes much too long to do this step. So let's have a look a bit more again in the sequence comparison business. Already mentioned a few times in this workshop, Smith-Waterman alignment computes the optimal local alignment of 2 sequences with a scoring scheme. Here, a simplified scoring scheme with a gap penalty G and a substitution matrix for comparing 2 letters. Here it's +2 of letters are equal -1 if they're different. In practice now, this will be a much more complicated scheme because you don't have the substitution matrix here anymore. You have this position-specific scoring matrix, which has a different value for every character in your input matrix. So anyway, so this computation sets up 2D matrix and initializes it with 0. And then you fill out the matrix here with this recurrence relation. You see the recurrence relation here. Upper neighbor, left neighbor, and upper left neighbor. This is the dependency relationship. Yeah? And then with this dependency relationship, you can, for example, sequentially fill out the matrix. The maximum is then detected, and the traceback procedure is done. And this returns optimal local alignment of 2 DNA sequences. In our application, we have protein sequences, which has a much bigger alphabet than DNA. One of the most important algorithmic techniques for sequence alignment based on dynamic programming, compute and sensor for large-scale data sets. Now, when we soon will look at parallelism, we have this data dependency relationship, left, upper left. So one of the approaches would be to look at here diagonals, or wavefronts. And those within a wavefront here can be computed in parallel from the previous diagonals. It's called wavefront parallelism. And the MM6-2 algorithm had a CPU version before. So this is very much in line with what Onur said before the lunch break. We have a big database, and we have filters of different computational complexity. Normally, when we use the biggest database, we have a filter which is a very low computational complexity. And that filters out a certain amount of sequences. And then the surviving sequences, then for those, we use more complex algorithms to further filter them down to the very similar sequences at the end where we just have hundreds or 1,000 from a database of hundreds of millions originally. So in the CPU version, you have 3 filters. The first filter just checks if there are 2 k-mer matches on the same diagonal. So very simple filter. So all sequences that have 2 k-mer matches on the same diagonal go to the next filter, which is called an ungapped alignment. I will talk about that on the next few slides. And those that are larger than a threshold in this ungapped alignment will go to this Smith-Waterman step with the algorithm on the previous slide. CPU version has these steps vectorized with AVX instructions. The first step basically hardly does any computation, right? That has a massive lookup table of k-mers. Yeah? K-mers, and it just looks up this table of k-mers with a lot of other k-mers to detect a double match. And that uses, with a big database, will use massive amounts of RAM. I think for big databases can be more than a few terabytes of RAM. I now talk about these. Okay, here we have these 3 filters. And now let's look at these 2 ones. So this is this Smith-Waterman that we talked about before. Here we have this other filter, ungapped alignment, or we also call it, we not always call it ungapped alignment. Some people call it gap-less alignment, which is the same. Just to use a different word sometimes. Okay, so this was the Smith-Waterman dynamic programming matrix. Similar dynamic programming matrix also in gapless. Here we have this dependency relationship, right, that I mentioned before with the 3 neighbors. So now the trick is to do the following here. This. Gaps deleted. That's why it's called gap-less alignment. Yeah? Gaps deleted. So you basically only score full diagonals, but in a local alignment way. So not the whole diagonal. You can start at any position, also end at any position. That's why you have this 0 here in the equation. Here, the 0. And then upper left neighbor plus letter xi and yj compared. And here we have this dependency. Now we have this dependency relationship. And this means that parallelization can be done in a different way. So in the Smith-Waterman, what we often do is this diagonal wavefront parallelization. And now in the gapless alignment, through this simplified recurrence, we can have an even more efficient way of parallelization, which is a row-wise parallelization. So a complete row can be computed in parallel with lots of processors. So number of dynamic programming cells is still the same, but this row-wise parallelization has a few advantages also in terms of memory access schemes that makes it much more efficient. So in the GPU version, we therefore looked at this whole pipeline. First, we deleted this first iteration step that just uses a lot of memory and lookups. And so we got rid of this. And instead, we just focused to make this extremely fast. So this is actually on a GPU, super fast. So we don't really need this filter anymore. And then we still have this Smith-Waterman then after that. So gapless parallelization. So this is the fine-grained parallelization on GPUs. We use a warp of threads or a subwarp, smaller number of threads within a warp, also called a coalesced group. And we assign a number of columns of the dynamic programming matrix to each thread. And we then compute. So this would be the dynamic programming matrix that we then compute in row-wise parallel order. So here we have one letter here of the target sequence from the database. And here along the column, we have the query sequence. So actually, we do not really have the query sequence because the query sequence is never really used. What is always used is the position-specific scoring matrix. And this is actually a matrix. So this is of size number of characters, so around 25 for proteins times the length of the sequence. And this is stored in shared memory, CUDA shared memory. And this is very important to optimize the accesses to this position-specific scoring matrix. So we developed many different schemes how to store this matrix in the shared memory. Yeah. So in every computational step, you have here a character, right? You take a character and then compute these values. And you would need to look up in this matrix the score for every position here. And then you just do this like one addition and one maximum operation. So within this very short time frame of 2 or 3 computational cycles, you need to look up the data very fast. So this is highly optimized in our scheme, how we access this matrix in shared memory. This requires in turn like matrix shuffling and so on, and one of the key elements here. So critical scheme to get fast speed on GPUs here. Another scheme, of course, this dynamic programming matrix here has the recurrence relationship from the upper left. So threads need to communicate data to other threads. This is done with warp shuffle operations. Another important aspect is the arithmetic precision. And you can use float or integer. Normally, this data type is far too big. You don't have such high possible number of values in your alignment computation. And for example, you can use half precision arithmetic here. For example, instead of floating point numbers, you convert them into half-2 numbers. And then use arithmetic like hmax to h@2. And you again need, if you do that, you have 2 values in a register. You need to kind of shuffle this query sequence in a certain way. Now we also, on newer GPUs, it's also, this is 16-bit arithmetic. We also have 8-bit arithmetic on newer Blackwell GPUs. Another alternative are these DPX instructions that we can use on Hopper, also on Blackwell architecture that have specific instruction to support these dynamic programming relationship like max3 ReLU ABC can compute maximum of A, B, C, and 0. Yeah. Or y at max ABC maximum A plus B and C. So these are very useful for the gapless, but also for the Smith-Waterman is ideally suited to support those. And so this resulted in this MM62 GPU program, and originally developed collaboration, my group with NVIDIA, and this is, which university is this? Cannot detect, you know that, right? You know that? Nobody knows it, right? Which university is it? Seoul National University. Seoul National University. Martin Steinecker. Martin Steinecker is one of the co-authors of the Nobel Prize winning paper that I mentioned at the moment. And in the paper, I have tons of more performance evaluations. So of course, resulted in very good speed up and was published in Nature last year. So you can also look at what I say is the end-to-end pipeline. So you have this MSA step and the deep learning step on the neural network. And there you optimize the MSA part, but you can also still optimize the neural network part. AlphaFold 2 is a closed source neural network. So that is difficult to optimize. And that's why there are now also open versions of this neural network, OpenFold and so on. Boltz 2 is a new one that are then also optimized with different data types to reach optimal performance. Okay, what I wanted to say now, since last week, we have a new version, also MM62 running on AMD GPUs. And this was developed with this HIP interface where you can write program for AMD GPUs. And we also use this Hippify tool that can more or less automatically convert CUDA code to this HIP code. Not everything, but most of it. And an MI 325X and an H100 that have around the same performance for this task. The second case study I want to talk about is untargeted food metagenomics. So Onur mentioned that before the lunch break, metagenomics, we are sequencing from an environment. He mentioned the streets of Belfast, right? But I think the most prominent case was New York Underground, right? New York Underground, that was sequenced around 10 years ago, and they found organisms that has not been heard of before. Some said aliens, yeah. Some said alien DNA was. I mean, I think that's the name for it, alien DNA. Well, I'm not sure if it was aliens, right? But New York Underground, that's an example. So mostly you look for microbial communities. So like what I said here, soil, air, and gut. You look for microbial communities from a mix of genomes, and you try to find out what genomes are in your sample. So we're looking now at food. With food, you not only have microbial communities, you also have what we call the main components of the food. Yeah? Can be different type of meat, for example. Yeah. So not only microbia, also main genome components. So that started some time ago. Here's sausage, for example. It's called MetaCache, and it has an AFS version or MetaCache in the All-Food-Seq pipeline. We classify food samples using a database of food genomes. Not only food, can also be what I said before, all kingdoms of life, microbia, and so on. Important for food control, supports abundant estimation of organisms at specified taxonomical rank, like genus, species, and so on. Has different type of filters to reduce false positives. And often, when you build reference genome databases, it is a bottleneck because these are really, really big. This was the our original All-Food-Seq pipeline. And this kind of didn't really have any optimized things. It just used standard read alignment tools. Kushaw is given here, but we also use BWA or Bowtie, and then we use BLAST. So this was kind of so it worked for a very small number of genomes, but it worked kind of for our food samples that we had, but didn't really scale to big reference genome data sets. So we looked at it again and redesigned the whole pipeline and did a redesign for GPU acceleration. So first of all, instead of alignment, we use some other index to compare strings. It's called the Jacquard index. In the Jacquard index, you extract so-called k-mers, yeah, substrings of length k from a sequence, and you have a set. If you compare two strings, you have two sets, and very simple, you compute the intersection and the union. And the cardinality of intersection and union is called the Jacquard index. Here you would have a similarity of 0.54. Not much. So in terms of computation, hardly anything needs to be done. You could expect, similar to the previous one, that you probably have some type of index data structure required for this. Using Jacquard index alone is also not sufficient because the amount of data, if you use all k-mers, that will be a very big index data structure. So min hashing is a technique to reduce the data by 1 to 2 orders of magnitude typically. And the idea is that when you use subsampled sets, the probability is similar under different conditions, similar to the Jaccard index. So instead of using all k-mers, we use a so-called sketch. This is another parameter, S, small s here, S smallest k-mers with respect to a hash function. So you apply a hash function to the k-mers, then do a ranking, and you take, let's say you have 100 k-mers, you rank them with a hash function, and you take maybe the 10 smallest k-mers that, if you want to have a data reduction by 1 order of magnitude. And then you use your Jaccard index on this reduced data set. Here, the example of the same data as on the previous slide. So now we got the k-mers here, and we already computed the hash values as integers. And in this case, we use the 4 smallest hash values. Also on the other data set, you use the 4 smallest hash values, and then you compute intersection divided by union on this reduced data set, and kind of approximatively similar to the full Jaccard index, but order of magnitude smaller footprint. So IFS, metacache key concepts, is first windowing. You have these reference genomes. You partition those in windows, normally similar size as your read data set. Then we have this min hashing concept. So you apply a hash function and only keep a certain selection of those values, which are here, these in colors. And those are stored in a hash table. This is called a pre-processing step. And then a read is input, and you want to classify the read in terms of sequence similarity. You also take the read. Fast. You take the read and again compute subsampling and compare it with the database and get the Jaccard index. And you have also GPU acceleration, what I will talk about on the next slide. Oh, okay. All right. Oh, those didn't survive the data transfer. Those didn't survive the data transfer. Anyway, you see the sausage, yeah? You see the sausage, right? We have sausages. These are real sausages. Butcher prepared them for us. Butcher prepared them for us, and this is why they're called calibrator sausages. They have a calibrated mix component. And now you sequence those, and you could check if you get, as a result, the amount of different components that the butcher put in. Yeah? And you normally then measure the quality of your result in terms of false positive and deviation. And we get quite good results on those sausages compared to, for example, Kraken 2, which is a frequently used tool in metagenomics. So how do we accelerate this on GPUs? Here again, this pipeline. And the main step is a hash table here, hash table. And hash tables also already, Onur mentioned that in the talk earlier, used in tons of area. I think you are also using a hash table. Didn't you mention that? No. Who gave a talk here? From Barcelona. Where is he? You didn't use a hash table. Not yet. But you mentioned the hash table. You mentioned the hash table. Okay. Hash table was mentioned. Okay. You mentioned the hash table also. Yeah. See, everybody mentioned hash table. So this is a hash table here. Different types of hash tables are around. So this is a hash table that we can also call a key value store. We have an array and we have 2 entries, the key and the value. And when we want to insert the key and a value, we apply a hash function that points to an index. And if that index is empty, you can store it. This is the insertion step. Yeah? Insert tons of k-mers. And when you get a collision here, it's already occupied. You have a so-called probing scheme, many different probing schemes. Here I just use a linear probing scheme. Go to the right. This is not the best probing scheme. It's just for the presentation. Linear probing is not so good. Normally you have other types of probing schemes, more complex. And then you find a free slot. Query works in a similar way. Again, if there were collisions, that requires a few more steps. So that also has been studied on GPUs a lot. So the key here for single GPU hashing is atomic compare and swaps to check, if many threads work on the same entry, only one of them can succeed. So this is using atomic CAS. And you have fine-grained parallelism by memory accessing using a coalesced group of threads to probe consecutive slots to reduce data transfer to main memory. And we developed a system called Warp Core. It's a library for those hashing algorithms. Also supports multi-GPU. In the multi-GPU case, you distribute data to multiple GPU. Then you do a multi-split where you have one color per GPU so that the values going through the same GPU are consecutively scored. And then you do an all-to-all communication operation between GPUs. Yeah. And this is, of course, a very data-intensive communication operation. So with PCI Express, this is far too slow. But with newer systems with NVLink, that can be done very efficiently. So that is the Warp Core library that was developed in my group. Now it's called the NVIDIA Q Collections library. It's in there. And it's also an AMD collections library, AMD HIP collections. The same name, just replace always the Q with HIP. Yeah. And this was a Warp Core with Daniel Jünger now, works at NVIDIA for that. We have also newer work. There are more data structures that are used in this area. One is called a Bloom filter. So we have a lot of work on Bloom filters, and also cuckoo hashing and cuckoo filters are other type of filters. And that was just accepted last week at the ICPP conference in Singapore. But performance of the Metacast GPU version, massive speed ups. Compared here, the blue one is Kraken 2, which is a well-known tool in this area. So reduces database construction from hours to minutes, and this really enables on-the-fly metagenomics. On the fly, we mean that you build the database on the fly. Like for fish, I use a different database than for sausages. Or paella, you need a different database. Paella we did, very difficult, because there are also all these vegetables inside, and the reference genomes of the vegetables are not very good. Yeah, not very good. So sausages is kind of an ideal case because the reference genomes are also very high quality. Yeah? I'll show you a bit other food that we are working on soon. Well, it's very difficult because reference genomes are not so good. Talking about Oxford nanopore technologies, food sequencing, ideally suited because it's also portable technology. Yeah. So that is very useful for food control. So we compared in our new work, AFS Metacache 2, sequence the sausages with both Illumina and ONT. Yeah? Of course, ONT gives better results because the reads are longer. Yeah? Reads are longer. Let's say I want to find out from which reference genome a read is from. When it's longer, the decision is easier, right? And we have also faster and more scalable data construction schemes here. And new results upcoming on honey. Very difficult, honey. You can imagine, right? And fish, also more difficult, but not as difficult as honey. There was a big honey scandal recently. It was not really a scandal because, I mean, it's clear that the honey you buy in the supermarket is not from bees. It's not. I can tell you already. I hope you're not too disappointed. But it's not from bees, the honey in the supermarket. Although it says original bee honey on it, it's not from bees. Can already tell you that. But the other, that was kind of known. That is not the main result of this. But It's one of the results. It's not from bees. Okay, key points here. Accelerators can offer order of magnitude speed up. We looked at protein structure prediction, warp cooperative in-register dynamic programming computation. That was one of the key concepts here. And the food, metagenomics, data sketching, and these hash tables. Real performance requires algorithmic redesign at several levels, memory hierarchy optimization, multi-level parallelism, and these sketching, partitioning techniques. Accelerators have moved from niche area to core infrastructure. So basically all labs, although they claim they are not having access, but all labs actually have some type of access to accelerators. And outlook as biological data keeps exploding, tighter hardware, software, co-design, and scalable algorithms are main drivers for breakthroughs. Okay, thanks.
Thank you so much for a very interesting talk. Questions? I have all of, oh.
I want to ask something because in the end, what I see is that you mostly focus on accelerating the computation itself by playing all the tricks that you can with warps and shuffle instructions and so on, DPX instructions. So all of that accelerates the computation itself. But two things that I want to know with respect to memory: how much of a headache is for you data movement, so bringing the data to the processors, the GPUs in this case, and storing the data in either memory or disk.
Well, you always have to see how much data you require as input and how much is output, you know. Of course, what doesn't really work is when you compute a dynamic programming matrix that you store the values that you have computed. That doesn't work. The performance, it's like 100 trillion cell updates per second it can compute on a single GPU, right? So that much data you cannot store. I mean, store in memory. So you have to compute it and then use the results and then overwrite those results. No? So you have to look at that you, only store what you really need. Yeah. So all of the data you can I mean, in any disciplines, all the things you compute, you cannot store. You need to kind of store some type of final result or something like that.
Yeah, but I don't mean only storing. I mean also like bringing those, for example, those sequences that are in.
I mean, for alignment itself, when you look at the computation, it's n squared complexity and the data amount is n. Yeah. So that is already quite okay. Yeah, already good. When you have these databases, one other technique that is often used is that you do not only use it for one query, but for hundreds of queries, yeah, to reuse this kind of data loading for many, many queries. So this is also something that we have implemented. But yeah, I mean, it's important and you always need to look at this.
Batches?
Batches you can also do. Yeah, batches. Yeah, we can do batches. Batches of input queries, for example. No? That is important. Or also when your database is bigger than your memory, right? Then you also only have batches. Yeah, good point.
Big data structures are you using for this GPU?
Which data structures? Well, in the metagenomics part, that is this hash table. And for alignment, doesn't really have a data structure. This is only computation. Yeah.
Yeah, because most of the things that you have in the metrics domain numerical, right?
Yeah.
But it was not numerical and unstructured. Things will be different in GPU, right?
Okay, that I do not know which computation you refer to.
No, just say for this particular problem is fine.
Yeah.
Unless you have a graph analytics.
Well, yeah, sure. I said data structures are important. That's what I said. Now, when you look up like the schema and so on, then you have this lookup structure that can be a hash table. There are also other approaches, right? These to reduce kind of accesses to hash table. You can use this Bloom filter, for example, or other type of filters. And this is the idea also that I showed and Onur showed, that you have kind of a cascade of schemes which filter out things to make computation overall faster.
Thank you.
Yeah.
Very fancy.
Provide like YouTube attendance.
YouTube, huh?
Yeah.
New things.
So I also had some more question. So actually, following off one's point, I will also discuss some of our research on, let's say, handling some things inside the storage system. So maybe avoiding some of the data movement overhead. So in the later session. But I had some questions for you. It was very interesting to see how these huge gains can be provided by these GPU acceleration. And based on your experience, I wonder if you have suggestions on how GPUs can be designed better to provide even, let's say, larger gains. So if you were to suggest or like have some feedback in that direction, what do you envision?
Well, now one new feature is now on newer Blackwell GPUs is this 8-bit arithmetic.
Yeah.
I understand. I used that already in 2013, the 8-bit arithmetic. And then it was not there anymore. Not hardware supported, only as software. But when you use it as software, it's like much slower because it's emulated by 32-bit computation. Yeah. But now on the Blackwell RTX Pro, it's also natively supported. Yeah. And this is also a scheme that doubles performance on the same GPU.
Right. I think it would be very interesting to kind of like see if we were to design GPUs from scratch, let's say, how would we design them to fit? Because like they originally were designed to different purposes. Already I see that some
different purposes,
specialization.
right? That's why, I gave a GPU computing lecture last semester. Then the guy asked me, these GPUs makes no sense. Why are they called GPUs? Because nobody ever does graphics on them. And I said, yeah, that's correct. I mean, I don't know why they are called GPUs. I mean, nobody does graphics. NVIDIA's business is below 0.1% of the revenue is from graphics. But they are still called graphics processors, but nobody does graphics on them.
Or like maybe one day
So
we
they're
can
designed for different purposes, I mean, depends what purpose
so
they are
maybe we can
designed
have
for.
ones.
They probably won't be designed for bioinformatics sequence alignment. Yeah, maybe these DPX instructions. Yeah, but the other parts are open for other applications, I would say.
Yeah, maybe one day we have GPUs like genomic processing units, but
Also GPUs. Yeah, GPUs, exactly.
Okay.
Well,
thank you so much for very sweet insights, including bitter facts about honey.
Looking at the prices of
Why not? Why not?
They're always getting expensive.
What next question for you? But I don't, the other people are seeing them anyway. There are lots of GPUs around. No idea why you do that, right? They have to update those, right?
Nah, they are cheaper ones.
Shuts down.
It's different sizing.
Yes,
I use it before, so it costs
Whatever, this is the full use.
The full, okay.
And we are now getting also G300. Also have those are of course more important.
And these are
Yeah, but you have also lower end.
I think I read the first one, right? And this is my. Okay, okay. Okay. Am I audible, Konstantina? Can you speak so we check the audio?
Hello, do you hear me? Thank
Okay,
you.
great.
Kalimera, kalispera.
Okay. So, getting started. Sorry, but hopefully we can continue in the break as well. Yeah. So, John Al-Khan is currently an associate professor at the Department of Computer Engineering at Bill Clinton University. He received his PhD in computer science from Case Western Reserve University, and then joined the Department of Genome Sciences of University of Washington as a postdoctoral fellow. His work includes computational prediction of human genomic structural variation, characterization of segmental duplications, and copy number polymorphisms using high-throughput sequencing data and acceleration of genome analysis through hardware-software co-development. I also had the chance to have Jalkan in my committee, so yeah. Thank you again.
Yeah, remind me to shorten that bio. That's way too. I just copy paste from somewhere else, you know. That was the show. Okay. Okay, let me, yeah, just remind me to shorten even more. All right, so, okay, I'm going to today talk about our partial order alignment acceleration framework that my undergrad student Zeynep developed. So heads up, Onur does not understand bioinformatics. I do not understand hardware architecture. So there will be lots of beautifully edited slides by Zeynep that I'll just show them to you and then let you read them. Again, this is the work of Zeynep Akçil. So she was an undergrad student, just recently graduated a couple months ago, and she's moving to UC Irvine. And this slide is supposed to be an in memoriam slide for our loss. Right, so she will be in UC. So I mean, she's not here because she was applying for the US visa. Her passport was in the US embassy, and so on. All right. You have seen this slide many times already in Onur's talk as well. So we do have some genomic sample. We generate the sequence data. And then we also want to compare them with a reference genome. So for this, we usually do seed and extend. And in between, we can do some pre-alignment filtering. Again, from Onur's talk, we have seen, for example, Shuji, Sneaky Snake, Gatekeeper, and so on. Okay. And then finally, we do the sequence alignment. All right, so again, we know this. So the sequence alignment itself, the verification step is done by dynamic programming over the read and the reference. And then when we are looking, when we are computing this, okay, ooh, this is how it goes. All right. So when we are computing a value, it is dependent on a very few other cells. And if you are doing stuff like Shuji does, then the dependency is even less. But the drawback is the reference bias. Okay. I'm going to talk a little bit more about reference bias in a couple of minutes. But basically, it over-penalizes the real variation, all right? Because the dynamic programming does not really differentiate between a sequencing error and a real variant. Therefore, we also have reduced sensitivity around the complex variation between divergent populations. All right, so there are two sources of errors in alignment or edits in alignment, right? So one of them is the sequencing error, which is pretty low for Illumina and for PacBio Hi-Fi, but much higher for PacBio continuous long reads and ONT platforms. And the other type of edits is the real genomic variants. Again, in the alignment phase cannot directly differentiate this. What we do is we use the error profiles from the sequencing, the quality scores, and also we also use the redundancy to be able to differentiate between the real variant and the sequence errors. Okay. But if we are misaligning before that error correction, then we are not going to have the chance to recover those. And also it is harder, so that's why it is harder to map a genome that has more genomic variation. So this is called the reference bias, right? So that is the reads that contain the reference allele maps with higher quality score, and the real mismatches, or such as single nucleotide variants, are penalized. So therefore, it is difficult to or impossible to find low minor allele frequencies. Right? And also if you are doing cancer genome analysis, the somatic mutations with low variant allele frequencies. So the variant allele frequency is defined as, so when you are, for example, sequencing a tumor genome, and if you are doing bulk sequencing, so some cells may have some variants, some cells may not, right? So these are different from the germline. So if you are looking for a normal tissue, for example, all the cells have to have the same DNA, but different from somatic. So every cell may have different edits in them. So that is the variant allele frequency. If that frequency is low, then you might miss them. Right. And also reads containing relatively larger insertions and deletions are often left unmapped, that you can work through this if you have long read sequencing data with some specialized indel-friendly alignment algorithms, for example. So here is an example. This is from the 1,000 Genomes project over 10 years ago. So these are different populations or different superpopulations. Is that thing? This one. There you go. Okay, so these are from different superpopulations. So there are 661 African, for example. So the Europeans are 503. And then when we look at the different types of variants, so the Europeans on the average have 3.5 million single nucleotide variants because the reference genome is mostly European. Right? So comparing Europeans to Europeans, you don't see a lot of divergence, but when you look at the Africans, it is almost 1 million more. Right? So there is more genetic diversity there. And then if you also look at the non-synonymous variations, so that affects the phenotype, again, you are going to see more in the African genomes compared to the Europeans. Right? But then if you look at the genome-wide association studies, so these are the variants that are associated with some sort of disease or some sort of phenotype, then their numbers are roughly the same. Okay? So this is because that genome-wide association studies are typically designed using the data from the European populations. This is pre-sequencing. Okay, so this is based on microarrays and then so on. So therefore, even though we have in African genomes, on the average, there are about 1 million more variants than the Europeans, because again, the reference itself is European, so there's a bias towards the reference. We are missing these variation. So that's why we want to overcome the reference bias. Although we already heard a little bit about graph mapping in Onur's talk, this is the reason, right? So if we have many different human genomes, right? So let's say these are all different human genomes. We want to find the parts that are different or common. So for example, here this G is specific to this individual, and in that position there are T's in other individuals. So we want to represent them differently. So we can do a couple of things. So this is in general called the pangenomics. So pangenomics just means that there are multiple genomes. Pangenomics does not mean graph genomes. Graph genomes are a way to represent pangenomes. Another way is just have a bunch of different full genomes, which would be very costly, right? And then we compare one by one. Or we can represent them as a graph. Again, graph is just a representation. And then here the backbone can be the common parts, right? And then we can have those different characters and different edges connecting them. So therefore, a graph. And then when you are doing a sequence-to-graph alignment, things change. So the sequence-to-sequence alignment, even with the much simpler needle-Wunsch way, when you are calculating the value of a cell, you depend on 3 other cells. Right? But with the sequence-to-graph alignment, now the predecessor is not just 3. So here we have 3 predecessors, but here we have 3, 4, 5 predecessors, because this T can be preceded by a G or can be preceded by this A. So there is more dependency to think about. So therefore, the sequence-to-graph alignment is necessary for representing, well, the graph is necessary, is a good way to represent the population scale variation. Again, even with the population, there are, like we said, like 4.5 million variants per African genome. That means there are still pretty much 3 billion bases common, right? But we can keep adding more and more variants to this graph structure, therefore reducing the bias. So when we are aligning, if we see an alternative variant that we have observed before, we are not going to penalize it. So the problem is, again, higher complexity and computational cost. There are branches. There are multiple predecessors to take care of. So here. So again, our purpose is to reduce this computational bottleneck for the sequence-to-graph alignment. So there are different ways of doing this. One of the oldest, the earliest ways, and still one of the most common ways, is partial order alignment. So partial order alignment was originally developed for multiple sequence alignment. Okay, so that is multiple sequence alignment of different sequences, and then the traditional way of doing this, like Cluster W does, or TCAFE or Muscle does, is that every multiple sequence alignment is represented by a profile, so that is column by column character frequencies, and then a new sequence is aligned by those frequencies. So that is lossy information, because once you are in frequency space, instead of saying there are 6 A's and 2 T's and 2 G's, you just say 0.6 A, then you already lose some information. So the partial sequence alignment can be used to retain that information, and then you represent a multiple sequence alignment as a graph, and then you align the new sequences to this graph. Right? So it replaces sequence to profile alignment, basically. So what Zeynep has been working on in the last year and a half, I think, is POACHA. So that is the partial order alignment computation hardware acceleration. We worked hard on this name because it is that. Oops, how do you go back? So it is that. So this is POACHA, nice pastry. Okay. So yeah, we spent like 60% of our project time in finding a name. The rest is easy. So it's an FPGA accelerator for bit parallel sequence to graph alignment. So we based our implementation on bit parallel graph alignment developed by Tobias Marshall and Veli Makinen's group, which is used as part of the popular graph aligner package. Okay, that itself is based on Bayes bit vector alignment algorithm. So it's a graph extension of Bayes bit parallel algorithm. So we develop a Verilog-based modular prototype, and our targets are reduced computation cost and also reduced memory usage. All right, until here, I understand. The rest I don't. So, but we have Berkan here, so I'll just use him as a backup. All right, so obviously part of it is done by software, part of it is done by hardware. On the host side, we process the alignment, the input, the fast. Where is that? Oh, there you go. Okay. So we process the read sequences, and then we find seeds. So it's basically a seed and extent, methodology. So we find the seeds, and also we have a graph to align against, right? So these are prepared in the host site, and the graph here is a directed acyclic graph. If it is cycles, then you have NP-hardness problems. Nobody wants that, right? And then we also do this pre-processing. We find the candidate seed anchors, and then we also partition the graph so that we can stream them. It's partitioned into smaller subgraphs. And then we pre-compute equality masks. So this is, again, a part of the Myers bit parallel algorithm. And then the core alignment is done on the hardware side, right? So that is given the graph, and then the rig is segmented into 64 base slices. They are sent to the hardware, and then the hardware does the mapping. So basically, we have a configuration that is the configuration of the alignment parameters, basically the sequence length, the direction of its forward alignment or backward alignment. Obviously, DNA is double-stranded, so we do 2 alignments. And then the read is prepared as 2-bit basis. And then it is processed in 64 base slices. So basically, 64 base slice is the seed size. And then the equality masks are prepared, the seeds are found, and then the graph is partitioned and then sent to the hardware. Okay. All right, so this is how the DP matrix works. So when we are aligning the graph versus the sequence, so these are the graph bases here. And then these are the read positions, and then we have a 64-row slice. Again, this is our seed, right? And then the graph, in the graph, every vertex represents 64 base pairs. Okay. And then we calculate the VP-VN matrices. So VP is 1 at the positions when the score increases, and VN is when the score decreases. Again, coming from Myers bit vector algorithm. So this part is not, we did not develop that part, but we are using that these formulations for our value. And then it returns a score. And then since we are dealing with 64-bit slices, then for the next slide, we need some carryover that is done by the HP and HN bit vectors. All right, so I don't know what's going on here, but basically, we have those scores, and then we have the equality masks, and then we have the ways to update the bit vectors for the next slice, right? And then these are update equations, and pH and MH are again for carrying over to the next slice. And therefore, when that slice is calculated, for the next one, we use the predecessor for the next one. And then we update the scores, and then we merge all the scores in profile level. Right, so at the end, again, I don't know what's going on here, so if you have any questions, don't ask. On the software side, we prepare the data, and then you push into the hardware, and the bitwise operations are done on the hardware side. Just as a top-level view, when we find the seed, right? So this is the seed starting location. This is the seed ending location. Starting from here to the right, we do a forward dynamic programming for the extension. And if this is the end of the seed, we also do a backward exact refinement for the reverse complement alignment as well. All right, I just talked about those. So again, as I said, there is a seeded bidirectional alignment. So for we do have the CSR for the forward alignment, but for the backward graph traversal, we generate a backward reverse CSR. Where is this thing? Okay, there you go. So for the backward, we reverse to the CSR, and we do the same thing for the backward as well. Hello. Next. Well, okay. I mean, it did something. So in conclusion, we do have a Verilog prototype for the bit parallel sequence to graph alignment. It's in GitHub. I'm not sure if it's a public repo or not. It's not. It's not yet. It will be someday. So it is a graph reference alignment that addresses the limitations of linear reference mapping and getting reducing the reference bias. It compresses dynamic programming state with bit vectors. So this VPV and bit vectors instead of having the entire DP matrix. Again, this is an extension of Myers bit parallel alignment. And it also supports the graph-specific dependencies through the predecessor state merging. And there is a bidirectional flow for and also with traceback support. Okay. And this is my group, and that is Zeynep over there. And Zülal and Berkan also advised Zeynep, because I have no idea what an FPGA is. And Toygar is another undergrad student, so he's working on the host side of Porsche. And again, Berkan is here if you have any questions. And Onur really did not show my nice, beautiful biofilm logo, but this is our processing in memory project. Well, thanks.
Thank you, John. Any questions?
I said don't ask.
No.
Big question, but
Maybe Berkan can also do.
Yes. So I was wondering, based on your experiences, again, it's like the same type of question. So if you were to suggest some specialization to FPGAs so that it would be easier to adopt these approaches or like to even get higher speed up, what changes to FPGAs would you suggest? Like, can we, like, let's say that we are AMD or Xilinx and we want to design better FPGAs.
What?
Documentation.
Sure. Another thing is, with my very, very, very limited understanding of FPGA, make the data transfer faster, please.
Make what faster?
Data transfer.
Always.
Yeah.
They have so like we are like trying to like implement this on like we turn an old workstation into an FPGA server. So we're trying to like implement this on like real silicon. And like the thing is, like it's like we're kind of like even if it's like PCI Express Gen 4, like we're kind of bottlenecked by it. So we're also like talking with Bertil Schmidt over here about like bandwidth. So like for example, one good thing about like NVLink compared to PCI Express is like it's very, very high bandwidth. So like data transfer is really fast. But like with PCI Express on FPGAs, that's kind of the bottleneck. Especially we have like some host pre-processing on our work. So we'd have to pre-process and then like push the data to the FPGA through PCI Express. That's kind of a bottleneck.
So there were some FPGA systems with HBM.
Yeah. But you.
Even in that case, also you would have to transfer from host anyway. So if you're doing some streaming work, that would still be your bottleneck, even if the memory is more higher bandwidth.
There are also NP stocks that have shared memory that already makes partial extent.
I mean, I jump 5 without because it's higher bandwidth.
Or to put the FPGA in memory.
Yeah. The thing is that now AMD.
That would be nice.
we can only do up to Gen 4, like with our current hardware.
Yeah, the thing is that now AMD has these adaptive socks that they call it, that has like, it's a big FPGA with a couple of ARM CPUs inside the FPGA, and maybe for your work that you have to do some pre-processing, but it's not that expensive. Maybe working on this type of FPGA that has the CPUs and FPGA in the same silicon, maybe can help you on this.
Okay, thank you for this.
It's actually close to my question. So you run this on a single FPGA. My question is if it's particularly optimized or customized to that one, and how easy do you think it would be to scale it to other ones or use it different FPGA devices?
Well, like the FPGA device we're currently using, like it's like the thing that constraints us on is like on the board, like the onboard memory, it's like DDR4, so like we're constrained by that, and also like the PCI Express, like with communication with the host, and like things we cannot store everything inside the FPGA. So like we send the data, like we have some like sorry, we have some like RAM blocks inside the FPGA that's like kind of a cache, but like the bulk of the data coming from the host is stored on the DRAMs. So like those constraints kind of like those are things constraining us on our current board, but like those like memory interface and like host interface, they could be modified because like we have an IP core for Quadra, and like that you communicates over AXI right now. So like that could be adapted on different boards.
So did you try some on simulation level of synthesis results to maybe do a design space exploration to better scale your scale your design and find different configurations that might not work on your platform but could be done in others?
We haven't done yet, but like we'll do in future. Like we're currently focused on debugging mostly, so
Makes sense.
So that's future work.
But I mean, the points you brought up are also very interesting, and I think it's also a recurring theme that we hear in some of the talks. It is a very good effort that we are specializing computation, but I hope that in the future we specialize main memory and storage systems together, because it's like a very data intensive field anyway. So I hope that we have equivalent of like these or like more compute enabled memory or storage systems. I mean, Jalkan mentioned this biopim direction. We will also present some in the next session, hopefully, about storage system as well. But yeah, if we could kind of like add that reconfigurability there, would be very nice. We haven't done that yet, but maybe it's something for future. Yeah. Great, thank you so much. It was very interesting to learn about this.
Thank you, Batman. You earned your lunch.
So we now have 5 minutes till the end of this session. An option is that we go like 15 minutes into the break, or like to move Irine's talk to after the break. So I can do democratic voting. So should we?
Makes sense because otherwise the cake is gone.
Yeah, we also were thinking of the same. So we will then move, shift.
We're talking a lot about food in this session.
Yes, exactly. Okay, good. Then meet you in basically 3:30. Yes.
What about the bag?
Yes.
When do we get the bag?
That's what we pay for, right?
Yeah, also for the kid. Wow. I need to go to the hotel soon. After
Okay, so now we are unmuted and I'm gonna go to reading slides.
I hope the format is fine.
Okay, okay.
I like the new name. It's like a vitamin thing.
That was my initial suggestion.
Yeah.
But yesterday everyone said it's terrible. And I told them it's okay. Keep saying this. I like.
No, it's very beautiful. It's like motivation. It's like vitamin.
It's the Greek word for motive, but I like it.
It's very good actually. Why did you take time with it?
Because of the topology aware.
Yeah.
Yeah. Double. No more double. Right.
They won't be decision then.
Nika, maybe your phone needs a little bit of a boost. So that we don't fall.
Do you want to introduce Irini? I was looking through the one that
Nice to meet you.
You do this and you do it working.
Where did your advisor go?
He Like he just said he'll drop the stuff off that table.
Ah, I see. Yeah, he's very close to the end.
Yeah, he's just like across the street.
Yes, yes.
Can you hear me? You're going to...
I cannot yet,
Yeah,
but Nika is in Zoom, Siri. Oh, I am not in Zoom. Let me go on. I've got the mouse doing now.
Oh, well, actually, like him and Onur, they're about to have some coffee.
Now, really?
Yeah. I mean, like I just remembered. I was like, "Where is Onur playing?" Like they plan to have.
Sabotaging. That's more fun here. So that's they know. Yes, so we are indeed on music. I can hear everything.
Okay.
Oh. Now the world knows.
It would be. Are you listening? Yeah, of course. But it could be now that Onur is in the top of the listening to all the. Maybe he will bring us coffee. Onur, if you're listening. Good job. So I hope they come back. She gave them. Huh? I'm not asked us to take this and keep them now. Oh, we should email them. Or what? Like I can also once we start, I can ask them. What about? You can email and I will check them. Okay, shall we get started? Yes, let me just run the. Here you are.
Okay, then everyone, welcome back. Let's continue with our next talk, given by Irini. Irini Gerbu is currently a graduate researcher in the Safari Research Group at ETH Zurich, led by Professor Onur Mutlu. She recently received her integrated master's in electrical and computer engineering from the National Technical University of Athens and her bachelor's in biomedical sciences from DIRI, which is the American College of Greece. Her research interests lie in the intersection of computer architecture with bioinformatics and biomedicine, with a focus on designing software hardware co-design systems for accelerating bioanalysis workflows.
Thank you, Konstantina, for the introduction. Hello, everyone. I'm Irini, and today I will be presenting our work, Motivo, a high-performance and low-cost algorithm architecture co-design for graph-based genome analysis. So first of all, thank you. So first of all, let's go over the executive summary. And first of all, let's introduce our problem. So sequence to graph alignment is the computational bottleneck of state-of-the-art genome graph analysis pipelines, as Professor Janal can also describe in high detail in the previous session. These tools that perform this graph analysis perform unnecessary expensive alignment operations, which are many times redundant. So in order to alleviate this bottleneck, we have a key insight that variation genome graphs have small recurrent topology that we will name motifs. And we are going to leverage these topologies to alleviate the alignment bottleneck by reducing computationally expensive alignment operations. So our goal overall is to deliver a high-performance and source-efficient design for sequence-to-graph mapping by reducing redundant operations. So let's see our three key contributions. First of all, we introduce a new indexing mechanism called Motivo Index, which is a compact motif-based index encoding motifs and node properties. We introduce a software hardware co-design extension mechanism using motifs and node properties to choose the best candidate alignment path called Motivo extension. And finally, Motivo filter, which is a standalone pre-alignment filter for sequence-to-graph tools that can be orthogonally incorporated to existing pipelines. And finally, let's see an overview of the key results. So the Motivo, which is an hardware accelerator, provides speedups up to 200 times approximately over state-of-the-art software graph aligner, 6.2 times over the best GPU baseline, which is NVIDIA's VG-Giraffe, and 3.2 over the state-of-the-art ASIC accelerator, which is Segram that we also saw in Professor Mutlu's talk. At the same time, it consumes 3.7 less area and 2.6 times less power than Segram. And also we see that the incorporation of the filtering mechanism increases both Segram's throughput up to 2.6 times with a minimal area overhead. And also we're going to see that it also enhances the performance of some tools such as Graph Aligner. So first of all, let's see this is an outline of my talk. Let's see some things in the background, even though I think Janal can cover most of it. But let's get in mind some of the things. So a typical genome analysis pipeline has 4 key steps. First of all is the sequencing, which is basically getting the DNA sample and extracting some raw signals, as we saw previously. These are translated into the characters that we know, which is the alphabet of the DNA, in the step called base calling. Then we perform seeding, which is the process of mapping the reads that we get from the sequencing machines into a reference genome. And finally, we have the alignment step, which is the most expensive one, that actually finds the number of edits between the reference and the read, and also the optimal alignment, meaning disregard most of the tools. So linear reference genomes, as we saw in the previous session, fail to capture population-level genetic diversity. And why is that? Because we might not have a specific reference allele in the reads, which exists in the reference genome. Therefore, we fail to map 3 out of the 4, let's say in this visual example, reads, and then therefore we have a decrease in accuracy. However, in order to alleviate this problem, we introduce graph-based genome representations, which are introduced as a solution, which, as we can see here, include information for all alleles and all genomes found within a population. So in that way, we alleviate the reference bias and increase the accuracy of our mapping. So now let's see a little bit in more detail how most of the sequence-to-graph mapping pipelines work. So the first step, which is the offline step, is basically to So it's basically to get our reference genomes, which are linear, and produce a graph from. There are certain tools that do these things, let's say like VG. So basically here we see that we get the common, this is a variation graph. There are also other types of graphs like the Bruin graphs. Here we use variation graphs. So basically we built our graph, and then as soon as we have the graph, we have the read sequence, and then the first online step of the process is called seeding. So basically we have some characterizing schemers, let's say, which might be minimizers or whatever other scheme we might choose, and we find them in the nodes of the graph. So after that, we have identified some possible mapping locations, let's say. So we need to extend in order to find the entire subgraph that is going to be used to align the read. And finally, we have the alignment step that uses all these paths produced either through chaining, screening, or clustering, and kinds of filtering that also exist in some pipelines, and then finally get the one alignment that is the correct one. So as we said, genome graph representations alleviate the reference bias. However, as we also previously mentioned, they intensify the alignment bottleneck because we have more operations, we have more paths that are possible, and thus we need to perform alignment operations more. So now let's see how we will go about this in our design. So here is basically a characterization of the 2 software state-of-the-art, VG and Graph Aligner, and Segram, which is the ASIC-based accelerator. And we see that for all 3 of them, basically, computationally costly alignment operations dominate runtime of their runtime. So basically, it's from 60% to approximately 99% for Segram, which is a huge issue, since this is a bottleneck. So here we make this, the second observation of ours is that we see that after the seeding step, there are a lot of subgraphs that are past the alignment. However, they do not produce valid alignments, even though alignment is performed for them. So this means that we have a lot of computation that is actually unnecessary. So it can range from 6.3 to 200 times more than necessary, especially for Segram that does not have a filtering mechanism. So this leads us to think that we need to do something in order to reduce this redundancy in the computation. So our first key insight is that we are looking, first of all, at short reads and see how the size of the subgraphs that are finally aligned to them. So we have, now we're looking at the final alignments taken from Graph Aligner as the golden output, let's say. So we see that we have more than 97% of the aligned subgraph span across fewer than 8 nodes. So this means that we have small subgraphs for short reads, And we're not expecting to have a path, big paths that are found sometimes in longer reads. So we keep this insight for later. So now let's see, we're going into more detail on how these subgraphs look, and we focus on their topology. So let's see. For example, this is a part of the reference genome graph. So we have, let's say, a reference genome graph. And then we say that we have some short subgraphs extracted from this genome graph. For example, here we have subgraphs of size 4, as you can see, or subgraphs of size 3. So we see here that basically because this kind of variation, this is topologically sorted, and it's also directly a cyclic graph. We see, for example, even in this situation, which is very trivial, we see that there are some topologies that recur within these graphs. For example, we have this red one. You see, we have the 3 nodes. Then we have this one. Okay, all the nodes are connected with each other because the graphs come from one reference genome. For example, we have this one for 4 nodes, and goes on. So we name these topologies motifs. Basically, this is an intrinsic property of the genome variation graphs because we have basically small nucleotide variations. So we're expected to have small jumps and not big structural variation graphs, at least for the graphs that we're looking at. So basically, keeping in mind the first insight that we saw about the size, we keep only the motifs that are up to size 7, so from 3 to 7 nodes. And basically here, let me show you the occurrence of these motifs within all of our reference graphs. This is for human genome graphs. So we see that more than 60% of all nodes of the graphs are contained the most common motifs. Here it might look a bit weird, but these motifs are overlapping. So you don't expect to add up to 100. So basically, we see that these topologies are indeed recurrent, and now we're going to leverage them in order to alleviate the computational expressive alignment step and create a motif-aware mechanism which will reduce unnecessary computation and accelerate the end-to-end sequence-to-graph mapping. So our goal in this work is to design a software hardware co-design accelerator leveraging these recurring topologies that we will be calling motifs for efficient sequence-to-graph mapping of short reads by eliminating redundant operations and avoiding unnecessary access to full nucleotide sequences. So now let's go a little bit deeper into our mechanism. So this is the first topology-aware software hardware code design accelerator, whose key idea is to leverage these motifs and also some compact node statistics such as the ACG contact and the length to find alignment paths without actually measuring on the runtime or accessing the entire sequence length and sequence information. So in order to do that, we could design an indexing technique, which we call Motivo index, and a post-seeding extension mechanism, which is called Motivo extension. So as far as the index is concerned, which is created in the offline process, it's not measured in the runtime, it encodes motifs in a compact way and finds basically where these motifs lie within the graph. And secondly, the Motivo extension mechanism will find the final alignments of graph after seeding and will leverage the Motivo index representation and also perform some lightweight computation to finally end up in a single alignment, which is the optimal one. So first of all, let's see how we construct the index. We enumerate all motifs. As we said, we have 62 of them. Here is an example of motif of size 3 and motif of size 4. And then we see that for each node within our big reference graph, we're going to search for all these motifs. For example, here we see M1 is found, for example, starts at node A. Then we have M3 starting again from node A. Then again, M4, the same, and then M6. So basically, we mask, in a way, these motifs to each node and see, starting from that specific node, if this motif exists in that specific region of the graph. We do the same basically for all the motifs of our subgraphs, and we end up with this kind of grouping. And as you can observe here in this simple example, we can see that there are some nodes that share group of motifs. For example, node A and node C have the same motif starting from them. So basically, we can also go in a further step further and group them. So we have more the information in a more compact way. For example, B here doesn't have another node, so it has a group on its own. So now let's see the two steps, as I said, is the offline reference graph indexing and the online grid mapping. So first of all, we have also a minimizer-based index. This we still use it to perform the seeding. And then we have the Motivo index, which includes the motifs that I previously described. So in the online process, we have the read sequence. We extract the minimizers, which is basically the minimum k-mer, as you know from my sliding window. It's very widely used. And then we perform seeding, which basically queries the minimizer that we have extracted to the minimizer-based index, and then returns the seed hit nodes and the offsets within the nodes. And then we have the motif extension mechanism, which comprises of 4 basic steps. The first one is to basically select in which case we are at X6. We're going to go in the extension step. We're going to look at it in more detail, and then calculate the motif ACGT content for all these seed hits, then find the minimum difference between the ACGT content of that motif and that of the read, and finally perform sequence-to-sequence alignment with using the GenASM algorithm, the beta algorithm, essentially. So, and this entire system will give us the mapping positions and the cigar. So now let's get into details of this, the motif extension mechanism. So first of all, we use the seed hit positional offsets in the read and in the node seed hit in order to find basically if we need characters to the left or to the right or the both ways to align the entirety of the read. For example, here we have a read. We have queried, this is the minimizer, the blue part, we have queried it in the graph. We have found the seed hit here, and we see that the entire sequence of the read can fit into one node. So this means that we do not need to extend, and we perform a traditional sequence-to-sequence alignment. The second case is that we have such a thing where we have to get characters from the right of the seed hit. That meaning that we need to extend to the right of the seed hit node, and respectively the same thing happens with the left case, and there are some less cases where we need to do it both ways. For short reads, this is a bit rare. So after having done that, we're going to iterate through all the motifs that we have indexed and calculate their ACGT content. Basically, in order to make this more efficient in the hardware, we have used 2 scratch pads, which are on chip. The first of all includes the groups that we saw previously, so basically which motifs are included in which group, and also the motifs themselves, so basically which nodes in the motif are going to participate in the ACGT content calculation, which is done in this block. So after having done that, then we write these different, all this ACGT content motifs, and we find the differences with the ACGT content of the read that we also calculate in the online process. After having found the difference, we only keep the motif, meaning the topology. You can also call it the path that has ACGT content closer to that of the read. So we find that this is the most probable to provide a good alignment, and thus we pass it as a path to the sequence-to-sequence aligner, which leverages the beta algorithm. So it does it performs bitwise computation both for distance calculation and traceback, and then we get the final mapping positions plus the cigar. I should note here that apart from the motif extension mechanism, this can also, as I said before, act as a standalone filtering mechanism by performing some small alterations in block number 2, where basically we take subgraphs of given size and perform filtering. So basically we calculate the ACGT content difference, and then if it's higher than the threshold, we discard that specific mapping position. So let's now see some of our results. Here is an overview of the evaluation methodology. So in order to measure performance, basically it's an RTL design. We synthesize with Ghiasi and then use OpenRow to get area and power. We use an in-house accurate simulator, which is validated against Verilator for some cases, representative cases, but also in order to be able to run this design in for our large graphs and our large data sets. We also made the FPGA prototype, which is also used to validate our cycle accurate simulator against. And then we use 24 human chromosome graphs from chromosome 1 to 22 plus X and Y from the recent version of the human genome. And then also we use simulated reads, short reads, of 100, 150, and 250 base pairs with 1% error. So here are some of the evaluated systems. So we categorize in 3 categories. First of all, we have the CPU-based ones, with the 2 state-of-the-art tools, which is GraphAligner and VG. Then we have a software implementation of our algorithm in order to see how algorithmic contributions actually how much speed up they provide to the accelerator itself. And then finally, we have also software implementation of our filtering mechanism incorporated into the 2 state-of-the-art software, GraphAligner and VG. So this is the CPU-based. We have the GPU baselines. HGA is basically kind of an older algorithm which naively goes and aligns the entirety of the graph with the read. So it's very computationally expensive. It cannot run for very large data sets. And that is why we incorporate our seeding mechanism to it to enable it to run for the big data sets, as we're going to see. We also evaluate VGGiraffe, which is NVIDIA's GPU version of the VGGiraffe algorithm. And finally, we also have GPU mapping of our algorithm contribution, Motivo GPU on a GPU. And finally, we have the ASIC-based system, which is basically Segram, which is state-of-the-art ASIC accelerator, our accelerator Motivo, and also we have incorporated the Motivo filter block with Segram to see how it enhances performance. So basically, first of all, let's take a look at the CPU-based systems. Here we see that we have a speed up of a very large speed up both for VG and graph aligner. For graph aligner, which is the state of the art, is approximately 190 times across all read lengths. So this is on the average of all three of them. And then also we see that we have Motivo, the software implementation of our algorithm alone can provide speed up over the baselines, and the accelerator comes and gives additional speed up to that. So our software implementation is also very good compared to the baselines. And also, let's also observe that our Motivo filter in the software version also enhances graph align's performance by 47% approximately on average. So it enhances the performance of the state-of-the-art software as well, apart from providing acceleration itself. Then let's see about what about the GPU-based systems. We see that our GPU implementation outperforms VGGiraffe by 2.4 times on average. And then that Motivo also, like the ASIC-based accelerator, outperforms our GPU already accelerated version of the algorithm by 3.4 times, and is 6.2 times faster than VGGiraffe on average. And finally, we also see that, as I said before, that we enable another tool, which is HGA, to run on large graphs and basically enable it to outperform the state-of-the-art GPU implementation, which is the VGGiraffe one. And finally, let's see at what happens with the ASIC-based systems, which is basically Segram. We see that we have an average speed up of approximately 3.2 for longer reads. Here, it's very interesting to see that for shorter reads, the Motivo filter plus Segram outperforms Motivo itself, while for longer reads, Motivo outperforms this combination, which is very interesting because in the way that we have optimized the alignment step, now our seeding part is the bottleneck. So for shorter reads, where we have mostly single node alignments, both systems, Motivo and Motivo filter plus Segram, perform the same amount of computation in terms of alignment, thus meaning that we are bottlenecked by the seeding mechanism in a way. So even though Motivo in overall outperforms Segram, consuming 3 times less area and 2.6 times less power. And we also see, as I said, that Motivo filter increases Segram's throughput by up to 2.4 times on average with minimal area overhead. So and also some small thing about energy consumption. We see that Motivo reduced energy consumption by 4 times compared to Segram, and also that it's the most efficient system of all, while the Motivo filter itself also reduces the Segram's overall energy consumption. So this is the overview of our work. It's the first motif-based co-designed accelerator for sequence-to-graph mapping. It improves performance of software. It has improved performance over the software state-of-the-art system, the GPU state-of-the-art systems, and the ASIC-based accelerator Segram, while reducing energy with low air and power overheads. And that's it. Thank you so much for your attention. I will happily take questions.
Thank you, Rini. Do we have any questions? Maybe I can ask you something then. So you described a bit your mechanism. My question is to you, what do you think is the most important factor that Motivo benefits from? And if you have an estimate of how much less computation and less data it operates on?
So basically, in terms of numbers, it operates like, as we saw in the previous slide, basically because it leads to one final alignment path. So basically, it reduces like we saw that it's the 6 to 200 for Segram, for example, operations. It reduces them by approximately that much.
So because, as I said, we have one candidate alignment, so we perform once the alignment step. So basically, that's, I think, the core reason why we have like such big benefits compared to the other tools.
Okay.
Yeah, yeah, it's a bit back in the slide, but yeah, it was back in one place. But yeah, I think you get.
Thank you, Rini.
Thank you so much.
Anything else? Okay, then I think we can move on because we're a bit behind time. Thank you very much.
Thank you.
More. Yeah. So Eric, you're next.
Okay, so Eric is a master student collaborating with the Barcelona Supercomputing Center, BSc. He got his degree on computer engineering in Universitat Autónoma de Barcelona, UAB. He is now working under the guidance of Professor Santiago Marcosola and Dr. Max Douglas on accelerating the next wave of genomics through heterogeneous architectures, particularly FPGA accelerators. Thank you,
Okay.
Eric.
Thank you, Nika, for the presentation. So I'll just jump straight into it. I'll be riding in giants' shoulders because there's been a lot of background already on the sequence alignment theory. So I'll just skim through it, emphasizing what I see fit, and we can get this over with fast. So basically, there's a lot of genetic data that needs to be processed. There's the new portable sequencers that generate long reads on these edge portable low power constraints. And as we have seen already, dynamic programming sequence alignment is a bottleneck for these long reads, particularly on the genome analysis pipeline. We've already talked about how dynamic programming works. We fill the matrix, then we can reconstruct the path that optimizes the cost, and we get the final alignment. What I want to emphasize about this is that the dynamic programming sequence alignment kernel has a lot of variations. That is, for example, in the algorithm side, there are the heuristics that can be applied, such as a band or a window, that sacrifice the guaranteed optimality in exchange of computing a lot less elements. There's also the Hirschberg algorithm that, in exchange of recomputing parts of the matrix, allows for linear space necessities. There's also that sometimes for filtering, for instance, we only care about the final score and we don't need to compute the traceback, although sometimes we do need it. We also have variations on the alphabet. These are just some of them. There are many possible alphabets that can be encoded in different amounts of bit widths. We have DNA, extended DNA with unknown characters, protein, and even ASCII characters. We also have different scoring schemes. We've talked a bit about some of them already. Edit distance, which assigns unitary cost to all edit operations. There's also gap linear or gap affine that penalize gaps differently. And also for even matches and mismatches, there's different ways to evaluate them. You can assign a fixed match or mismatch cost, or you can use the protein blossom matrix, for instance, or custom substitution matrices that assign different cost and rewards for each substitution of one character against another. Another big thing about genetic workloads is the length, which in the next generation sequencing machines that were circa 2000s wasn't really an issue because they were at most 250 base pairs per read. And also they were of fixed length, so always the same. We can see that doesn't hold for newer technologies, such as PacBio or Oxford Nanopore, where the length can go even to 1,000,000 base pairs for a sequence. And also it's not static. It can vary greatly. And so we must be prepared to handle all these different workloads. The quadratic complexity of dynamic programming has also always been a problem, more so with longer reads, which makes surface this problem more. And the typical answer to a high complexity is hardware acceleration. We can see that hardware acceleration follows a pattern where the more flexible you are, the less optimized you are for a particular problem, and the opposite also holds. And where for our project, we chose FPGA because it was the most reasonable choice we could make. Fabricating an ASIC supposes a lot of costs. It means that you are committed to a single design for a very fast-moving ambit, that is this sequence alignment problem that changes rapidly. So we chose FPGA. When seeing other FPGA contributions, we can see that most of them tend to have limitations on what kinds of sequence alignment workloads they can handle. They can have like always banded algorithm or only a single alphabet, like 2 bits ACGT only, no substitution matrix, or maximum sequence lengths, such as limited to Illumina or maximum 10K or all these kinds of things. So, well, also another point is that there's a lot of FPGA contributions that report only the FPGA itself power consumption, but the FPGA is actually plugged into a very high power workstation that consumes a lot of power. So our goal in this project was to provide a flexible sequence alignment accelerator that can handle most, everything is always impossible, so most sequence alignment workloads. And also we wanted to do that under low power constraints and keeping the solution itself portable. We have two sides to this contribution. The first is more algorithmic or logical, and the second is the accelerator itself. So basically, when looking at the kinds of algorithmic variations that dynamic programming sequence alignment tends to have, you can really divide the matrix into what we call DP blocks, dynamic programming blocks, which are regular. This means they can be more easily accelerated. So that is what we do. We create a hardware-software co-design, or basically a CPU with an accelerator working together, where the CPU does the irregular work of calculating these boundaries of these regular blocks, pre-processing the sequences when needed and as needed, then offloading the DP blocks to an accelerator, which does the regular dynamic programming computation, and which sends back the results of these DP blocks, which are only the borders, as I'll explain now. And then the CPU can do the traceback if it is needed, which is not always, and any other post-processing that could be necessary. So basically, we base the implementation of our idea in the CRIA KR260 board, which is the one we talked about earlier, which has an MPSoC, a multiprocessor system on chip, which contains a 4-core CPU with an FPGA, which are not tightly coupled, but pretty close to each other. They have 4 gigabytes of DDR shared memory, unified, so it eases a bit the difficulty of the implementation. This is the architecture we make use of and configure, where we have an RTL design that can access the last level cache of the CPUs in a coherent way, thanks to the accelerator coherency port, an AXI subset. So the accelerator is basically, well, based on the main ideas of what Max presented earlier. We have a systolic array that has input and output for the borders of what we call the tile. And we have 3 workers, which is what we were able to fit into the programmable logic of this board. And each of one of these workers is handling a different DP block. And their work, what they have to do is fetch from memory the input borders for each tile, and then save the results they get from the engine. Then we have the memory bridge, which handles the requests and responses from and to the last level cache, and a control interface that has two queues, one for jobs or configurations and one for results that can be pulled from the CPU. We can see here the strategy we follow. This is pretty similar to what a bit vector implementation will be, where we divide a 64-bit word into as many elements as we can, depending on the element size that we choose. It's worth noting that this is configurable in runtime, so it can be chosen for every particular alignment. The platforms in which we evaluate our system is the CRIA board itself. We measure only using the ARM cores. We also measure using the ARM cores with the accelerator, and we also compare against a high-performance x86 core to see how that works. The datasets are real datasets from PECBio and Oxford Nanopore, and also a protein dataset for protein alignments. When measuring gigacaps, well, we have 3 different use cases. The first one is 4-bit DNA alignment with traceback using the Hitchberg algorithm. The second use case, B, is a banded alignment without traceback, so only score. And the third one is protein, which is full alignment, but also without traceback, which could be used for a protein database search. We selected these use cases because they are similar to real use cases you could have with sequence alignment. We can see we are 40 times as faster, at least, when using the accelerator versus only using the low power ARM cores. And we are about 2 times as fast as the x86 high power core. This is enhanced in protein alignments because software is very inefficient at those because of the random accesses to memory, which the accelerator handles better because it has a specialized structure for the substitution matrix. So it doesn't have to do this inefficient accesses. Here we have gigacaps per joule or gigacaps per watt. Gigacells updated per joule or gigacells updated per second per watt, which are the same. And we can see we are more efficient than both the cores we compare against. Also, our average, well, this is peak wattage, if I'm not mistaken. So we can see we keep a very low power profile, which will allow to maybe do this on the edge on working with batteries. I would also like to note that even if the x86 is CPU package power, the 4.7 watts is a whole system, the whole board. So we are at that bit more efficient. So basically, those are the contributions. We get up to 120 times speed up against the low power ARM cores on protein, and 15.5 times more efficient than x86 on this use case. So that's it. If you have any questions, I'll be happy to answer them.
Thank you so much. Questions?
So the times you showed there, is it like including the time to bring stuff from the DDR memory?
Yes, this is end to end.
Okay.
So from the moment you have them no, this is end to end. So reading from disk.
Okay.
And
yeah.
everything, yeah.
Good, good. That's great then, yeah.
Yeah, thank you.
More questions? Konstantina, do we have time for one more question? Because I have one, otherwise I can ask him later. Okay, then I will ask you. Sorry, I will ask you later.
Okay, perfect.
But thank you so much for the very interesting
you.
topic.
Oh, yeah, I was gonna take it away. Thank you.
Next one is recommend where it is.
Yes, Shahin, yes.
It's eagle?
What?
Does it mean eagle?
No, it's hawk.
Ah. Okay, introduce him meanwhile. So Berkan Shahin is a master's student in the Department of Computer Engineering in Bilkent University. His research interests include hardware acceleration, heterogeneous computing, and bioinformatics. Shahin obtained his bachelor's degree in computer engineering from Bilkent University as well.
Okay. Thank you for the introduction, Nika. So, all right. Okay. Yeah, once again, thanks for the introduction. And today I'll present our work on in-depth data movement characterization of DNA read mapping algorithms. So read mapping has been like in this workshop described many times at this point. So my background will be kind of light. So like here, read mapping, like in the context of this work, it refers to like just the process of aligning sequencing data to a reference genome. So we use aligner and read mapper like to mean the same thing in this work. So, and like a trend we observe not just with read mappers, but also like other workloads as well is like in a system, increasing the core count does not always translate to performance gains due to some bottlenecks, like for example, related to data movement, etc. And like there's also been like some previous work that analyzes like read mappers in depth in like an architecture perspective, but like this work these works were focused on single core architectures and not multi-core scaling. So, and meanwhile, while this is happening, we have these bottlenecks. Like there are some novel architecture ideas like processing in memory, processing near memory as well, that like they promise to alleviate some of the data movement bottleneck by moving the processing closer to or inside the memory. So what we see is there is a need for like in-depth performance characterization of read mappers that like one takes into account the scaling on multi-core systems, many core systems, and also the data movement bottlenecks in these systems as well. So I'll give an overview of like a memory hierarchy in like a modern multi-core system, like for example, a server. Like it might have many CPUs, but like, okay. So in like in a multi-core CPU, we have like some resources are private to the cores, like for example, like the first levels of cache, but like some resources are shared between cores. So like for example, the memory buses and the last level of cache like is usually shared between cores. So like in a system, sometimes when we just like increase the amount of cores, but they still have to share the same resources, we see increased chance of resource contention, which will result in performance degradation. So we tested 3 classes of read mappers aligners. Like for whole genome alignment, we tested Minimap2, MashMap, and WF-Mash. For long read alignment, we tested Minimap2 again, NGMLR, and LRA. And for short read alignment, we tested BWA-MEM, Bowtie2, and Strobe-Align. So let me introduce our like analysis method. So our method consists of 2 major steps. First, we perform top-down analysis. We observe the general performance of the program and scaling with like increased thread numbers based on metrics from our based on metrics from our real server-grade hardware. And then like once we identify some like specific memory bound functions, we perform in-depth function level analysis using a simulator I'll describe in a bit. Right, so first for top-down analysis, what we do is we classify time spent on like each pipeline slot of a CPU into the following categories. First, we have a front-end bound, which like refers to pipeline stalls, but as the instructions are being fetched, parsed, or decoded into micro operations on the front end of the CPU. And then we have a bad speculation, which is operations that were like flushed out of the pipeline due to branch misprediction. So like we spent some time like decoding these like when we wouldn't end up using them because of misprediction in the CPU. And we also have backhand bound stalls. So these are operations that were not like, in that clock cycle, not accepted to the backhand of the CPU due to resource constraints. So like here, like two subcategories are of interest. So first, memory bound. So here the CPU does not process the operation because it's waiting for the operands to come from the memory. And also, we also have like core bound, where here like the CPU cannot process the operation because all the processing units are currently busy processing other operations, so the operation has to wait. And then we have retiring operations. So these are operations the CPU has finished processing and they now have retired. So like all of these metrics we try to minimize except for retiring. More retiring is, most of the time better, let's just say. And like retiring percentage, we could say like most of the time we have a more efficient utilization of the CPU. And like a high percentage of memory boundness would indicate that the CPU performance is being bottlenecked by the data movement. And moving on to our function level analysis, we pick like from our top-down analysis, we pick functions that that take at least 3% CPU time and like they're more than 30% memory bound. Like we pick those for our in-depth analysis. And here we use this tool called DAMOVE Sim from the DAMOVE benchmark suite. So this tool combines this tool combines the ZSIM x86 simulator and the Ramulator memory simulator to like in a simulator workflow to like capture information about the data movement between the CPU and the memory. And like here, like this has a few advantages. Like here, so we collect metrics for only our regions of interest. And like since this is a simulated workflow, we are more sure that like there hasn't been interference from other processes running on the system compared to the real hardware metrics. And also since this is a simulator, this allows us to more easily experiment with different memory hierarchy models, as I'll show in the next slide. So like we can see their effect on the performance. And like here the metrics that are most useful for us is like the instructions per cycle and like the cache miss rates across all cache levels, and also the last to first miss ratio, because like if we have a high last to first miss ratio, it means that like having a hierarchical cache with many levels might not be suitable because like our first levels are working well, but like the last level has many misses. And like here we test like with our in-depth analysis, we test like two configurations mainly. So like the first one is like the configuration I showed in the background section. It's like modeled after like a typical computer workstation, let's just say, that has like a shared last level cache and then like a bus that connects to the DRAM. And then we have like a PIM configuration that's more like processing near memory that kind of has these like cores that have like only one level of cache and then they're like connected to the DRAM banks. So here like for our purposes, we just like one caveat is we assume that these cores would have the same capabilities as like our CPU cores, which is not most of the time the case. But like here we see kind of like the feasibility of such an approach. So like we would still need to do some more work on like testing this on real processing near memory hardware, and like you might have some more caveats there. Okay, so moving on to our results, first with whole genome alignment, like we observe like across our both of our data sets and like across different thread counts. Like this is just a snapshot of what we have. So we observe that with Minimap 2 on whole genome alignment, the performance does not vary much with thread count. So this caused it to actually fall behind other aligners like on high thread counts. And we also observed that WF MASH is consistently the fastest out of the 3 whole genome aligners we have tested. So moving on to a microarchitecture overview from the top-down analysis, we see that Minimap2 has the highest ratio of retiring instructions. MatchMap displays like front-end boundness and bad speculation that is quite high. Like it's like both of them like adding both metrics together, like we get more than 50%. So that's quite high. And we observe that WF MASH has a very high memory boundness. So like analyzing some of these further, for example, like with Minimap, like the anomaly we observed was like it did not, like when we increased the thread count, the performance didn't change much. So here we have some, like when we analyze that further using VTune, we see that like when we look at the histogram, like it spent 67.5% of its runtime using only a single logical core. And in this configuration, like the machine we used, like the server we used to test this had like 112 logical cores, and we told Minimap2 to use 96 threads. But in this workload, it spends like 2/3 of its runtime using only a single core, which kind of explains, like, even if we increase the thread count, we don't see much increase in performance. And moving on to MashMap, we do a function, like we try to see function by function, top 5 functions to see if there are any specific culprits. But what we observe is instead, we see that like there is this consistent pattern of bad speculation related stalling and flushing across like almost all of the top 5 functions in MashMap. So moving on to long read alignment, what we see is NGMLR is significantly slower compared to the other aligners. There's a reason for this. I'll explain in the next few slides. And we observe, unlike the whole genome alignment, that the relative performance stays the same across thread counts. So this means like, unlike the whole genome alignment, like one program does not have like problems with scaling across multiple cores compared to the others. So their relative performance is the same. And yeah, as I said, it's like points to similar degrees of scalability. And like moving on to NGMLR, what we see is we do not see, like at least with the like top 3-4 functions, we do not see like a microarchitecture related bottleneck. But what we observe is it uses more instructions for the same workload compared to the other tools overall. And like this is due to NGMLR being a more sensitive aligner compared to the other long read aligners. So this is kind of like a sensitivity to performance trade-off. And we observe that especially this function over here. Let me see if I can use it. Okay. This function over here, get real location. This one is highly memory bound. And when we analyze the code of this function, we see that it's a single line. And like, first of all, it takes 9.6 cycles to execute one instruction in this. So the instruction per cycle is pretty low. And we see that this is like some code related to like doing some pointer arithmetic, adding some offset to an existing base pointer. So like this is kind of really memory bound. So like our suggestion is this might cause some of the performance issues with NGMLR. Like maybe it's still sensitive, so like the algorithm is different, but like if maybe this is alleviated, for example, like having like more prefetching friendly code with hints and such, this might like make it come closer to the other aligners. And with Minimap2, we observe that this function KSW-EXD2, like that does banded Smith-Waterman, it's mostly core bound. And we observe that these functions, Minimap index get and collect seed hits, which have to do with like memory operations, like end up being memory bound mostly. And we analyze these functions further using the movesim. And sorry, and like we get similar results for both of them. So I'll like show one of them. Like what we see is like for this specific function, as we increase the thread counts, like especially the miss rate on the last level of cache climbs, and this was also reflected in the last to first miss ratio. Like one of our hypothesis here is this might be due to like contention in the last level of cache. And we also like test with our like quote unquote PIM configuration that's more processing near memory. And what we observe is like as we increase the thread counts, like get into higher and higher core counts, like we we see that like the quote-unquote, sorry, the quote-unquote PIM version is like getting a bit better IPC, like there's a mild improvement in like very high core counts. So like, as I said, like maybe like for like a very massively parallel version of this algorithm, like switching to a simpler cache hierarchy, if like one is designing their own cores, might result in IPC improvements. For example, like the works in this workshop that like have their RISC-V cores, like maybe when they're designing the cores, like the cache hierarchy might be simpler. So, and also with LRA, we also have some like memory bound functions, and like we also observe a similar pattern here. It's not as pronounced as Minimap2. So like we could not get the MoveSim working as of now, so I cannot show those results. So we'll move on to short read alignment. Here we see like strobe alignment is significantly faster compared to both High-2 and BAA MEM. But we also see like there's some diminishing returns, especially after 16 threads. And like based on the configuration we had for our server machine that has like 4 physical processors, we suspected non-uniform memory access, and like we're still testing this hypothesis, but like our preliminary results shows this to be the case, because like when we like at low thread counts, like 8 threads, when we use NUMA CTL to pin it to a single core, we see some performance improvement, and like we see a significant decrease in like the percentage of remote accesses. So pinning to a single core if we're not using a lot of threads might be a better idea in this case. And moving on to the microarchitecture, we will proceed. Like at this point, we have like the in-depth analysis for BAA MEM, so we will proceed with it. So with BAA MEM, we see that like it uses Burrows-Wheeler algorithm for alignment, and we see that like the functions relating to the Burrows-Wheeler transform, like, and also accessing the suffix array using the Burrows-Wheeler transform, like, is highly memory bound. So here we see also like as we go like with higher and higher core counts in our simulated system with the MoveSim, we see like an increase in the last to first miss ratio, like an increase in like level 1, level 2 stay relatively the same, level 3 misses kind of increasing. And also here we see like a much more dramatic increase in IPC with the like quote unquote PIM approach, because like here we see like kind of it doesn't scale as much as we increase from like 16 to 64 to like 120 to 56. It's kind of plateaus, but like the other simpler cache hierarchy shows like it can scale to much higher instruction per cycle overall. And like we see that like overall with this work, microarchitectural inefficiencies and also like sometimes a lack of parallelism are the main culprits of like performance bottlenecks when comparing aligners against each other. And like, for example, like some key results, we see that Minimap2 on whole genome alignment spent like 2/3 of its runtime single threaded, like no matter how much threads we tell it to use, no matter how many cores the machine has. And also we see on MashMap overall like a pattern of being bound by bad speculation and front-end latency. And we also observe like this pattern we observe across categories, a pattern of memory bound functions, like when we analyze using the simulator-based workflow that cause like contention in the level 3 cache. And in this, like one of the main indicators here, like there are more indicators, but like the one we have shown in this presentation is like the increase in last to first miss ratio with increasing thread count, which points to like let's just say a non-ideal use of the cache hierarchy we see in modern CPUs. And like here, like at least in theory, using a simpler cache configuration, that one that doesn't have many levels, provides mild to significant like instruction per cycle improvements, especially in high thread counts. So I would like to thank my colleagues in Alkan Lab and Can Alkan as well. And I can take any questions you have.
Thank you so much, Berkan, for the nice analysis and results. So questions? maybe I can ask first something. You showed some IPC comparisons between baseline and PIM systems. I wonder what are your suggestions on getting higher speed up of those PIM systems? I assume you made an assumption about the PIM system, right? Some configurations. So what aspect of it do you think need to improve to get higher speed up?
Like here, like I think the speed up we see is like, at least in the simulator, is like more related to locality, and like also not just like with like general bound, like memory bounds between like DRAM to processor, like also with like, for example, with strobe, and that's like an ongoing analysis, very recent. So like I don't have like the full results on the presentation yet, but like as I said, what we have seen is like with like if we use all the cores in the system, like on our system, it's for physical processors, it's kind of like it is still quite performance, but like it's not like kind of a perfect scaling. Like, and that's like largely the culprit is like the non-uniform memory accesses, because like one core tries to access memory map to another core. So like in general, what I would say is like what causes like the most, like what would cause the most performance improvements is like having more locality, like trying to like be cognizant of like the non-uniform memory accesses and like the cache hierarchies in modern systems, like, and designing accordingly would like bring on performance improvements.
Yeah, thank you so much. Great. I also have more questions, but I think because of time, I might throttle myself. Okay, great. Thank you so much. The next talk is online because the speakers could not come in person.
Yes. Can you hear us? Can you speak?
I'm hearing you
On
all.
Zoom?
Thank you. Can you hear me? Hello?
Yes, we can hear
Okay.
you.
Okay, thank you.
Okay, now let's stop sharing. Can you stop sharing?
Yes, we're going to stop sharing.
I sent a request to share.
Okay.
Oh, there you go.
Okay, now move this.
Now we have to move this.
It is going.
Okay. Full screen. Okay, but let me try and see the speakers a bit, right? See it.
Maybe you can hotspot them, pin them. Pin Raghu, maybe. Or like, no, actually, I think, yeah,
I think this is
we can
enough.
keep. Okay, yeah, sounds very good. Let me then give the intro. Okay. Raghu Shankar is an entrepreneur incubating projects in semiconductor systems and cloud native architectures. His experience spans product management for servers for HPC and AI at Dell, and processor planning and architecture for data center solutions at Intel. He completed his master's in electrical engineering from University of Texas at Austin and MBA from Kellogg at Northwestern University. Thank you for joining us, Raghu.
Thank you very much. It is our pleasure to be presenting in this forum. Thank you very much. And after viewing the last two Architecture4Health workshops, as well as reading many of the research works from many participants in this forum, indeed an honor for us. I'm looking forward to more of this series going forward. So we've got three objectives today. First is to share the excitement about two major growth areas. One, as we all know, is computational biology, and the other one is systems in packages. And the second objective is that we would like to get feedback on our direction, mostly like outside of this workshop, so we've not enough time. So looking forward to feedback on our direction, and happy to meet with you, many of you, offline as we build our product. And we also open for collaboration to develop many of the innovative areas that overlaps computational biology and systems architectures. So going forward, I think you introduced me, and I have Suresh, my project partner, on the call. He's a CTO at Selectronics, and is a kind of system designer and architect for advanced packaging. And he has got experience on the EDA side with Cadence and AMD, the silicon side, and also many startup companies listed out there. Our contact information is below, so please feel free to contact us and give us feedback. And I think as many of these workshops have been mentioning, this genome sequencing as a high throughput challenge and exponentially expanding sequencing data. Many more living species are being sequenced. These sequences are generating almost 16 terabases today, and these are using single flow and multi-flow cells, as we understand. And these are growing rapidly, as we only expected to go more. And the reads per run are in the hundreds of billion today, and of course they'll be increasing. The read lengths are in hundreds of base pairs today, maybe even 10,000, and I think they'll also be increasing. And we're seeing a lot of great sequencing algorithms today. They are quite complex, but there's a lot of innovation happening in the space. Many of these are multiple sequencing steps that the algorithm needs to go through. Many of these steps are both a mix of sequential and parallel components, as well as they are also some phases are compute intensive, some phases are memory intensive, and some are IO intensive. So it's a challenge to bring it all together in terms of scaling up and scaling out, as well as the managing the energy efficiency. The runtime of these analytics are ranging in tens of hours, but clearly we can do better out here. So we're going to propose this, or we're working on this system in a package that we are calling SciPod. It's a genomic chiplet that can be built onto it, and that will help address many of these issues. Okay, so on the right, you see the system in a package. This is, think of it as like almost 6 times today, the 6 times the size of a monolithic ASIC or a monolithic FPGA. And the roadmap of the systems in a package is extremely strong. These are built on 2.5D and 3D architectures, technologies. And as you can see, these are built for throughput and as well as balancing energy efficiency and other scalability aspects. Starting with the IO bandwidth, as you can see, there's almost like 1 terabytes per second of raw data that translates to, in a genomics equivalent, you can bring in 4 terabases per second. And the HBM, each HBM or host bus memory is 32 gigabytes today, and a roadmap that can keep growing. That translates to almost 96 gigabases at 30x coverage at 2-bit that people will be familiar with. The total HBM capacity is 256 gigabytes today, and that will fit 30 Oxford Nanopore technology datasets of 176 gigabytes. So it filled the whole datasets in the host bus memory. And very soon it can also fit in 50x Pacific Bio dataset of size today is like almost 311 gigabytes. The host bus memory bandwidth is 9.6 terabyte, the total bandwidth, 9.6 terabytes per second. So that translates to almost 40 terabases per second of data that can be fed from these HBMs. The chiplets, now I'll talk about a genomic specific chiplet that can be built onto this. So, but the chiplet to chiplet bandwidth is through these UCIe links, that's 6 terabytes per second. So you can pipeline your data back and forth between these chiplets, and that you can transfer almost like 24 terabases per second. And the total system power at the low end could be around 400 watts. So that will be ideal for a lab deployment. But if you want to scale up and scale out, it can go up to 1,000 watts budget. So it can be put in micro data center deployments where much more of the extensive computational computational biology and bioinformatics can be deployed. So each of these genomic specific chiplets can be built on these platforms. So you can think of this as genomics processing units. And all of these phases, one or more, or in fact, however much you can fit, even all of these phases can be fit on one chiplet or in adjacent chiplets. As you have pointed out, these chiplets can be homogeneous. They can all be the same, or they can be heterogeneous chiplets. So you can have some phases on chiplet 1 and some other phases on chiplet 2. And the other third thing to point out is that there is a lot of, like, for example, in chaining, we see that there are many sequential and parallel computing. So those can happen in the same chiplets or adjacent chiplets, depending how we program it or build it. Extending is the forward phase is parallelizable, while the backtracking is sequential. Again, they can be happening on the same or different adjacent chiplets. Then there are other phases that are also very kind of either compute intensive or memory intensive or IO intensive. So the customization can be done, and I will be sharing in the next slide how that will work. And I think I mentioned about the internal. I mentioned about the internal data transfers that can happen at almost 24 terabases per second between the chiplets. So again, this is built for innovation. Each of the genomic chiplets, the total of 4. So we are working with a RISC-V partner. RISC-V is architecture similar to ARM and x86. And RISC-V, so we can package, and we have done the analysis of how much we can put on this. So we can package, put in almost 16 to 32 RISC-V cores. So it will be 16 scalar cores and 16 vector cores, or it could be 32 scalar cores and 32 vector cores. So you're going to get each of them like independently. L2 cache is 8 megabytes. L3 cache is 32 megabytes. The caches can be hardware managed for cache coherence. There is also a software managed local memory. So in case you want to put some data sets, you can have like 16 megabyte of each of either you can put instructions in, genomics instructions in, and you can also put your genomics data. So it's kind of resident and not churned by the cache management traffic. Net, this comes, there's all these vector units provides 2 teraflops per second at 50 FP64. So there is no compromise out there. Plus, you can also go to lower precisions as needed. This will also provide a high bandwidth vector memory up to 64 megabyte that comes with Linux support. And the most interesting part is the custom instructions. So as innovators, you can define your own custom instructions that I think I've seen many of the talks mentioning before on both the scalar side and the vector side. And this system will provide you the user tool chain for special innovative hardware that can be built onto this chiplet. And lastly, there is also a vector data streaming port. So instead of doing all this fetch and retrieve and all that, you can just stream that vector data in at very, very high throughput. So built for throughput, and if you see on the bottom left, the RISC-V machine, RISC-V cluster along with vector units, you can put like almost like 64 megabyte of RAM per chiplet. And essentially, this has become like a processing near memory system, local SRAM memory as well as the HBM memory. And so we are working, our next big milestone is to work on a FPGA prototyping environment with a workflow that will allow innovators to go and work on their core competency of sequencing research or computational biology research that can then be loaded up onto this system in a package platform or SciPod platform. So I'm going to share with you as a kind of a scale up of a lab deployment. We compared this to today Illumina NovaSec machine that we see has got like 4 FPGAs, the Alveo U250s, and we can put 4 of these in 4 of these genomics chiplets, and you get like 8x the memory bandwidth compared to Alveo today, 16x the IO bandwidth through the PCI lanes, and also you get the internal bandwidth that I mentioned before, like 6 terabytes per second. And since this is all like customized for genomics, you are kind of getting all 2x the utilization. So net, we kind of on paper, we see that there could be an 8x advantage over 4 FPGAs. We can also do a scale out. And so on the left side, you see the system in a package chip that I said is like going to like almost 10x the size of ASIC or an FPGA today. Extremely good roadmap that we have on that. Plus, we get like co-package optics and many other things happening to keep this system in a package roadmap very exciting. Many of the leading vendors are already using it today. You see that from NVIDIA, AMD, Intel, but it has not yet opened up for the general kind of next level of adoption. So we want to democratize this whole thing so that folks like you can build on it. So on the left side is where I think we're going to build, offer you that system in a package platform. And you see that your genomics chiplet can fit into that, and you need to be worried about that. Now, this system in a package can go into a baseboard of an open accelerator module, which is the second thing showed out there. That already exists today. So that's part of the Open Compute project that exists today. And then that module can go under a heatsink, and then we can put 8 of those modules, I can see on the rightmost side, on a universal baseboard. So all that basically means is that you can put in a scale out that I'm showing on the right side via micro data centers. And so you can have like 2 host servers, that typical. You can have solid state drives with petabytes of storage. Your databases could be sitting on those solid state drives. And then you are going to have these, each of these boards, you can put like almost 20 universal baseboards per rack. So that gives you 640, say, genomic processing chiplets on this. So besides genomics, obviously you can do a lot of historic analysis, real-time analysis, bioinformatics, proteomics, multiomics, so many things are happening out here. And the metrics will be that you can speed up your sequencing, your reads per run, your accuracy can go higher, and real-time analytics can be like speeded up, and as well as keep your cost and power in check. So these are the references that we have, thanks to a lot of great work out here. And we would like to basically pause here and thank you for the opportunity. And these are our one more set of links, contact links. And we are happy to take some questions now, and definitely would love to meet with you all, many of you all offline, if we can do that.
Thank you so much for the presentation. Thanks a lot. So are there questions in the audience? Maybe I can start with the question. So you mentioned several terabytes or like bases per second of chip bandwidth. Could you comment on that a little bit more? How is that achieved?
Oh yeah, so we have like almost 8 PCIe Gen 6 lanes coming in. Okay, if you see on the top, if you see the arrows on the top, Nika.
Yes.
So I'm just going to simplify it, but you have like 8 PCIe Gen 6 lanes, each of like 256.
So each of these PCIe blocks are 8 PCIe 6 lanes.
There are eight lanes coming in.
Oh, okay.
Eight lanes, but 8 lanes, each lane is by 16.
Yeah.
So
Each lane is by 16. Yeah. Go ahead, Suresh.
There are 8 total ports, and each port is a by 16 PCI. And it can, in this version, it's Gen 6. So if you take the raw bandwidth of the PCI ports and then get the base equivalent, that's what Raghu is presenting.
Yeah, correct.
Understood.
So it's like basically at like almost you get 2 terabytes per second. Sorry, 1 terabytes per second. And then so you multiply that, you get almost 4 terabases per second.
Right. Also, I was wondering, what is the cost of the specialization? Let's say if you want to specialize for different applications, how is that process, or like how long would that take, etc.?
So the development cost, if you are going to be staying on the design that we provide, so I think as sharing out here that we'll have, let's say, with a partner, we'll have RISC-V cores, right? If you're able to program your code, program it into the RISC-V architecture, for now at least, right? We don't know if you can do ARM or x86 later on, but just for now, RISC-V is open hardware. So if you can do the programming you can use the workflow that we are thinking of and program the RISC-V code. So that's kind of the lowest kind of challenge, lowest kind of entry point. But if you say, hey, Rogod, I got an innovative algorithm, and I want to create some custom instructions, which we saw some of the researchers doing in this forum. So that will take a little bit longer lead time to say, you have to build the instructions and go through the tool flow, and then kind of code those instructions into RTL and put it in the genomics chiplet itself, right? So that will be a longer flow that we are not yet kind of characterized, but it is possible.
Understood. Yeah, perfect. Thank you so much. Maybe are there more questions at the moment? Or yeah, maybe we can have a bit shorter Q&A because we're also running a bit late. But thank you so much for your talk and joining.
Thank you very
We'll
much, and I'm
continue now
looking
with the
forward to
next
staying engaged with many of you. So thank you very much. I really great appreciate this series.
Sure, thanks. Okay, now maybe we can spotlight the.
So maybe you can also
Introduce myself. I am the next speaker. Let me see if this.
I'll just check that this was.
Okay, yes. I will skip the. Okay.
Okay, so let's continue with our next talk, delivered by Nika. So Nika Mansouri Ghiasi has recently defended her PhD thesis and is now a researcher and lecturer in the Safari Research Group at ETH Zurich, advised by Professor Onur Mutlu. Her research interests are in computer architecture and computational biology, focusing on storage systems, large-scale bioinformatic applications and their interactions, and also emerging technologies such as ultra-dense 3D integrated systems. She's interested in designing systems that facilitate the widespread adoption of data-intensive applications needed in healthcare and life sciences. She will be on the academic job market in the upcoming cycle. So Nika, we'll look forward to your talk.
Yeah, okay. So I'm going to start. Thank you, by the way, Konstantina, for the introduction. I'm going to start with my talk, storage-centric system designs for enabling fast, efficient, and local genomic and metagenomic analysis. I'm going to skip this slide because Konstantina already introduced me. So as we have been discussing today, genomic and metagenomic play key roles in many fields related to health, life sciences, and beyond. And to get the genomic information of organisms into digital format for analysis, we rely on high-throughput sequencers, as we have seen very nice backgrounds about them earlier today. And we have seen also that current sequencing technologies cannot sequence very long DNA molecules end-to-end, and instead they extract these smaller fragments called reads, which are then used in genome analysis. For example, in a key step of read mapping that maps these reads then to a reference genome. At these matching locations finds the differences or similarities between the sequences by performing alignment, which is based on approximate string matching, which is a quite costly operation. Also, since sometimes a sample contains information on different species, genomic is augmented with metagenomic, as also we have seen earlier today, and that enables the study of genome sequences of different organisms within a shared environment. And we have seen also examples of very broad environments covered also earlier today. And because we, in several cases, we don't know the species that are present in the sample, we need to look up large database containing information on many species. So because of all these factors, first, like important role of these application domains, and also large reduction in sequencing cost, volume of genomic data has been growing at a very fast pace. So overall, genomic and metagenomic, as we all have heard several times today, require performing expensive computation on large amounts of data and therefore suffer from computation and data movement overheads. And motivated by all these factors, there's been a lot of efforts in both academia and industry to accelerate these operations via efficient heuristics and algorithms, hardware accelerators, or designing some sort of filters. And together, they lead to a lot of advances in alleviating computation overheads, and of course, there is even more to be done there. However, despite the benefits that these techniques provide, we realize specific outstanding problems. One is that relate to accessing stored sequence data and feeding it to these analysis units. So the first problem is overhead of moving large amount of low reuse data from the storage system and the unnecessary computation burden on the rest of the system. So here, as we have seen, while we have alleviated computation overhead, data needs to move all the way from storage system to the rest of the system to be analyzed. And a second problem is data preparation bottleneck, where compressed genome sequence data needs to be first decompressed and formatted before it can be analyzed. So I'm going to tell you in this high-level example about this problem a bit more. So consider a baseline scenario of preparation and analysis over different batches of genomic data. And then consider a second scenario in which analysis has been significantly accelerated. And a third idealized scenario where data preparation is no longer bottlenecking the end-to-end pipeline. So by comparing the first and last scenario, we see that there is large potential benefit of genome analysis acceleration, but a large part of that benefit gets lost because of data preparation bottleneck. We're just feeding this data to the accelerator in the desired format. So motivated by these factors, our thesis statement has been that by designing customized storage-centric systems that efficiently analyze genomic and metagenomic data inside the storage system and enable highly compressed storage and high-performance access of these large-scale sequence data, we can alleviate the overheads of data movement, computation, and data preparation, thereby significantly improving performance, energy efficiency, and cost effectiveness. So I'm going to give you a very brief overview of our work in this direction. First is GenStore, a storage filtering system for genome analysis. And Megis is a cooperative in-storage processing system for metagenomic analysis. Grains, which was last week actually presented at ISCA, is a storage-aware algorithm architecture co-designed for graph-based genomic and metagenomic analysis. And finally, SAGE, which is an algorithm architecture co-designed for efficiently storing and accessing sequence data. I will give you a brief overview of these. So starting from GenStore, so the key idea is to filter reads that do not require expensive alignment operations inside the storage system, so we only send reads that require more substantial computation outside. And examples would be exactly matching reads and non-matching reads, that they do not need to go through the approximate string matching operations of alignment. Maybe I give a very brief background of read mapping. I am sure that it's might not be so necessary, but I will cover it very quickly, just a refresher for people who are not familiar with the area. So consider reference genome, so the search space can be very large. If you want to map a read to this reference genome, mappers typically leverage an index structure that encode k-mers or subsequences of length k extracted from the reference genome along with the corresponding locations of these k-mers. So the mappers typically have these high-level steps. First is seeding to determine potential matching locations or seed hits in the reference genome, and to do that, the mappers extract k-mers out of the read, look up the k-mers in the index structure, and mark the locations as seed hits. But because still these are too many locations that we want to get rid of alignment cost as much as possible, mappers typically also leverage another step, seed filtering or chaining, to prune some of these seeds based on lighter weight heuristics than alignment. And for the remaining locations, we have alignment, which determines exact differences and similarities based on approximate string matching. So GenStore, our design, has two parts for exactly matching reads and non-matching reads. In this talk, I will give you a brief overview of the first one. So it is just an efficient in-storage filter for reads with at least one exact match in the reference genome, and it uses simple operations without requiring alignment. But the challenge is large number of random accesses per read to the reference genome and its index. And that is challenging because of expensive random accesses to NAND flash chips, which is a common technology used in modern SSDs, and also limited DRAM capacity inside the SSD. So you can imagine, like, let's say for every terabyte of storage, you have only 1 gigabyte of DRAM there. So we address these challenges with these ideas. So first, we introduce read-size k-mers to reduce the number of accesses per each read. So as opposed to extracting many k-mers, we consider one read a k-mer. And so that then reduces the number of accesses. And we also introduce sorted read-size k-mers to avoid random accesses to the index. And that leads to having sequential scan of read set and the index, so we don't need to go through the challenges associated with random accesses. Of course, despite the benefits of this approach, because of large number of read size k-mers, the size of the index increases. So we reduce the size of this table by replacing the k-mers with a strong hash value. Here we use cryptographic hash values. So that leads to large reduction of the size of the index. It is still larger than the baseline index, but we realized that because of large internal bandwidth within SSD for sequential reads, that was a right trade-off to make. So it actually is quite efficient to read this index inside the SSD. So I'm going to show you a little bit of results. So consider here mappers, baseline state-of-the-art software, and hardware mappers of the time. And GS here base integrated with GenStore as its, let's say, backend SSD. And we evaluated various SSD configurations. So we show that here I'm evaluating for exactly matching reads compared to software and hardware-based mappers. It provides large benefits, and also we show that it provides large energy reduction. Similarly, GenStore for non-matching reads, I didn't talk about this design, but also it provides large performance and energy benefits. So that was a high-level overview of what's, also, our synthesis results show that it does so by basically consuming very little area and power, which is a small fraction of some of the ARM cores that are on the SSD controller. And also, SSD controllers have many other hardware units as well. So relatively, it's quite small. So that was a very high-level overview of GenStore. I will also give you some very brief overview of Ghiasi. While also performing metagenomic analysis inside the storage system would be beneficial, doing so actually is very challenging because, again, metagenomic analysis tools cannot also directly run inside the storage because of limitations of modern storage systems, such as latency of NAND flash chips, limited DRAM capacity inside the SSD, and limited DRAM bandwidth inside the SSD. So our work addresses these challenges, and we introduced Ghiasi, which is the first in-storage system for end-to-end metagenomic analysis, with the idea of cooperative in-storage processing for metagenomic analysis, which is enabled by algorithm architecture co-design between the storage system and the host system. And to get the best of both of these worlds and coordinate the capabilities of these two systems, Ghiasi's co-design consists of 5 key aspects, which I just tell them in very high level. So the first one is task partitioning and mapping, such that each step executes in the most suitable system based on hardware requirements for that step. And the second one is data computation flow coordination to reduce communication overhead between these and also reduce number of writes to the flash chips so we do not hurt the lifetime of the SSD. Third is the storage-aware algorithms so that we can change the algorithms such that it fits within these in-storage processing paradigm. And lightweight in-storage accelerators such that we minimize the SRAM and DRAM buffer spaces and also bandwidth, because these also do not provide enough bandwidth inside. The DRAM bandwidth is also very limited. And finally is data mapping scheme and flash translation layer, or FTL, which is specialized to the characteristics of metagenomic analysis and how data is being accessed through that so that we can leverage the SSD's full internal bandwidth. Okay, so we evaluate energy, performance, energy, and power based on hardware and software-based components. We evaluate performance optimized software and accuracy optimized software, and a PIM hardware accelerated tool that uses processing in memory to further accelerate the performance optimized software. And we evaluate various cost optimized and performance optimized SSD configurations at that time. So here in a cost optimized SSD, we see that compared to both performance and accuracy optimized baselines, Ghiasi provides large speedups, and similarly, we see large benefits when we have a system with performance optimized SSD. And here we also observe large speedups in systems with both types of SSDs compared to the PIM hardware baseline. And Megis also provides large energy benefits compared to all these baselines, and I would like to highlight that it does so while achieving the same accuracy as the accuracy optimized baseline, which is higher than the accuracy of the other evaluated baselines. And the area and power consumption of the hardware units are also very small, so it's even actually much smaller than GenStorm. So what I want to highlight, which is I think a key point in these storage-centric designs and what we hope that would also lead to larger adoption of these data-intensive genomic and metagenomic, is the impact on system cost efficiency. So let me highlight this particular result. So consider a cost-optimized system with a cost-optimized SSD and 64 gigabytes of DRAM, host DRAM here. And a performance-optimized system with a performance-optimized SSD and 1 terabyte of host DRAM. So as you can imagine, the cost difference between these two systems is huge, and many thanks to the DRAM market, the gap is becoming larger and larger these days. Anyway, so we evaluate the performance and accuracy optimized baselines and the cheaper and more expensive system and compare it to Megis on the cheaper system. And we observe that when on the same system, of course, the benefits are large, but also Megis outperforms these baselines even when running on a much less costly system. And we hope that, as Cindy was also mentioning earlier, before like impacts of importance of portability, we hope that this enables, let's say, portable analysis or like facilitate wider adoption by reducing costs. So that was the overview of Megis. I will also give a very high-level background, like overview of grains. So as we have also seen earlier today, genome graphs represent genomic sequences with graph walks, and that leads to powerful and efficient representation of genomic data because of great expressive power and compact representation of these graphs, and which are actually critical for enabling massive population scale analysis. And again, of course, it would be beneficial to handle things inside the storage, but doing so is challenging because of limitations of SSDs, as we mentioned before. But then again, we have new challenges here because of dependent and random accesses to the sequence graph and different properties from conventional non-genome graphs. And then we introduced Grains, which is the first system for analysis with larger scale genome graphs in storage, and is enabled by a storage-aware algorithm architecture co-design based on properties of larger scale genome graphs so that we can make the entire execution pipeline more storage friendly and also further improve performance, energy efficiency, and cost effectiveness via storage-centric computing. And there are 4 new aspects in its co-design. First is new batching technique and execution flow to make the whole, like basically, we can reduce the number of random accesses to the storage system. And then in storage and in flash processing so that we avoid transferring low reuse parts or unused parts of flash pages all through the system. And then again, specialized FTL and data placement, and a lightweight scheduling technique that is enabled by repurposing the existing SSD structure to leverage the full parallelism of flash dies when we are doing in flash processing. Again, based on hardware and software components, we evaluate performance, energy, and power. We evaluate a state-of-the-art node-centric tool and a state-of-the-art edge-centric tool, and an idealized accelerator for graph queries with no main memory overheads, but still suffering from I/O overhead of moving large amount of low-reuse data. And we evaluate various SSDs with PCIe Gen 4 and 5. I show that in both cases, we observe large benefits compared to both evaluated software baselines and also large energy efficiency. And similarly, significant speed up and energy efficiency improvements compared to the hardware baseline. And the synthesis results show that the uncontroller logic, like the units that are on the SSD controller, are less than 1% of the 4 cores on an SSD controller, and the on-die logic units and the flash die logic units take only 0.2% of the flash die's area and fit very well within its power budget. So that was an overview of our works in the first direction. I will also very briefly talk about SAGE before concluding. So as we have discussed, data preparation can significantly hinder end-to-end performance of the pipeline. So we need to effectively mitigate this while meeting 4 key requirements: high performance, high energy efficiency, a high compression ratio comparable to state-of-the-art genomic-specific compressors, and have very low overhead such that we can integrate this design into genome analysis systems in different places. Meaning if you have PIM or like in storage or portable systems that are doing genome analysis, we want our design to be so lightweight that preparing the data does not consume kind of like more resources than analyzing it. And that becomes challenging because of resource-constrained environments that some of the genomic systems are implemented in, as I gave some examples. So we propose SAGE, which is an algorithm architecture co-design for highly compressed storage and high-performance access of large-scale sequence data. And it is based on the insight that information encoded in genomic data follows some specific properties that we can leverage in our algorithm architecture co-design, such that we can not only represent the data very compactly, but then we can decompress and prepare it using very lightweight operations and very efficient streaming accesses. So consider here a genome analysis system that requests data with SAGE's interface commands to access SAGE compressed genomic data that is operating with SAGE's data layout. Then SAGE's hardware receives this compressed data, decompresses and reformats it, and sends prepared data in desired format to the genome analysis system. And because it's very lightweight, it can very easily integrate with genome analysis systems in different ways, like either as a standalone accelerator or on the same chip as genome analysis accelerator, or in resource-constrained environments, for example, inside the SSD. So again, performance, energy, and power based on hardware and software-based components. We evaluate read sets with different species and different sequencing technologies. And I'm going to show you end-to-end performance, which includes both analysis, both data preparation and analysis. In these 2 scenarios I'm going to show. So for genome analysis, we evaluate a state-of-the-art hardware accelerator for read mapping, near data processing or NDP baseline GenStore, as I've talked about before. So it alleviates the burden of moving and analyzing large amount of low-reuse data, but it is implemented in resource-constrained environment of the SSD. So for data preparation, we evaluate a commonly used general-purpose software, a genomic-specific software, a genomic-specific hardware, and an idealized decompressor with 0 decompression time, but inefficient for integration in resource-constrained environments. Inefficient meaning in case that it requires also a large amount of resources so that it could not be met in the resource-constrained environment. And we also evaluate SAGE, which is implemented as a standalone accelerator for data preparation, and SAGE SSD, which is SAGE inside the SSD for integration with the NDP analysis system. So we show that compared to general purpose and genomic-specific baselines, SAGE provides large benefits on end-to-end analysis performance. But also we show that SAGE achieves the same speed up as the 0 decompression baseline, and that shows basically that SAGE's data preparation in the pipeline is no longer in the critical path. So it's not like bottlenecking the analysis stage. And we also show that by efficiently integrating SAGE SSD with NDP, SAGE SSD plus NDP leads to larger speedups. So what it does is that it enables us to design systems without, let's say, designing accelerators without relying on genome analysis, genome sequence data to be stored perpetually in uncompressed ways. So we also show here large energy reduction compared to the evaluated baselines, while also achieving much higher compression ratio than general purpose baselines and comparable compression ratio to genomic-specific baselines. And again, our synthesis results show that SAGE's hardware unit consume very small area and power. By the way, it also provides large benefits in software, not as much as hardware, but still it's faster than other baselines, and we will be open sourcing that as well soon. Okay, so that was overview of our work in this direction. So in conclusion, by designing customized storage-centric systems that efficiently analyze data directly inside the storage system and enable highly compressed storage and high-performance access of large-scale data, sequence data, we can alleviate the overheads of data movement, computation, and data preparation, and thereby improve performance, energy efficiency, and cost effectiveness. And we hope that this together would also help making accurate genomic and metagenomic analysis more accessible for its wider adoption. Also, you can see more about my research in my website. Like I also work in different directions other than the ones that I presented, and more details about previous Arch4Health initiative websites and videos and all. Yeah, I think that brings me to the end. Yeah, thank you.
Thank you so much, Nika. Do we have any questions?
So I know you went very fast, so I didn't quite understand the SSD story. Did you use a real SSD or did you use just the MQSIM?
No, we used MQSIM because we were relying on some changes on the FTL and also on the hardware inside the SSD. So here is simulation based, but we are trying to work on the real adoption of these techniques.
So did you use something like OpenSSD or?
No, we used here simulation based. So we necessarily do not also rely on OpenSSD, meaning we don't need the host system to handle the FTL as long as we can load our own FTL inside the SSD, not, let's say, managed by host directly, it would be also fine.
So have you considered using ZNS SSDs?
Zone namespaces? Yes, so but however, in the real adoption, we are thinking of different directions. One is that to implement some of these and some DPUs next to the SSD, but outside the controller. That's one direction. The other one is that some companies that work on specializing the SSD controller, so we can directly actually let's say adopt or like implement some of those, but that takes of course like a longer time. The ZNS directly would not enable handling some of these things, some of these designs, like some of the specialization that would be needed.
Yeah, so with the ZNS, you can choose your zone sizes and
Yes,
you can control exactly what's going on. So that might help.
Yeah.
I'm not sure.
It would definitely be helpful, meaning it would be essential but not sufficient in that sense.
Okay, thank you.
But thank you so much.
Thank you. Any more questions?
Can I ask one, Nika?
Of course.
Yes, of course.
Nika, very good work.
Thank you.
Very good work. So thanks for sharing. I think, have you started looking at host bus flash as part of your research?
Host bus flash?
Yes.
So think
of it as a next big evolution, next big jump over solid state drives. And host bus flash is like similar to a host bus memory, where you have the NAND flash stacked up and you have a base die. And the host bus flash, it's by the way, it's already commercial. So a lot of big leading vendors who are doing also offering it. And it gives you much higher capacity. It gives you almost like 0.5 terabyte of capacity. And it also gives you 1.6 terabyte per second of throughput from the host bus flash. So that is my question. Have you started looking at that as a possibility of speeding up your throughput?
Right. That's a very good question. So we have not like exactly for that technology, but we have done some, let's say, limit sort of analysis to implement some of these optimizations outside the SSD, but leveraging, let's say, sweeping number of SSDs or like external bandwidth in the end, right? And we observe benefits still because of one reason is that our designs make the whole access pattern also more storage friendly, meaning it avoids channel contention anyway. So it helps, because of that, it helps reading the data even more effectively, even if it is implemented outside. But the one problem that still remains that leads to some of these designs lead to better performance is the low data reuse, meaning because some of this data have very low reuse, and even some parts of the pages, because of the granularity of access, some part of the pages would be unused. So the closer we go actually to where the data is, meaning in the flash chip, it leads to higher benefits so that you don't pay the time of these things moving and also energy. Because even if we have, let's say, infinite bandwidth, we still need to pay the energy of bringing that. But other than that, I think within the scenario you mentioned, like despite the weaknesses I mentioned, it is still interesting, definitely.
Yeah, no, I'll send you some, if I may, I'll send you some link for host bus flash, like one link. And if you are interested, if you feel that's like, I think it's a good evolution, because all of these host bus flashes can be loaded up right next to the host bus memory that I was showing before. So you have like 8 kind of stack of 8 host bus flashes connected to 8 host bus memories. And so they are like feeding at very high data rates into the host bus memory that can come to the accelerator in the middle. So so it can be exciting area for innovation for this computational biology.
Right, yeah. Good, thank you so much, great.
Yes, thank you.
Sure.
Any more questions? Maybe I can ask one. So I wanted to ask, I look forward to seeing what's next basically after this work. And my question is, how do you envision that we could actually do an end-to-end thing that includes multiple steps of the genomic pipelines within the SSD?
Okay. Yeah, that is a very good question. So we have, looked at read mapping steps, or like in metagenomics, some of the steps. I think, of course, like it would be interesting to look at different parts of the pipeline in genomics and also other domains in computational biology. Like, let's say variant calling would be another interesting thing to look into. And other things like to expand into would be signals analysis, because it also looks into not only this genomic data, but also various other modes. But another thing which I'm very excited about is processing everywhere, which is to kind of like design a system that enables processing inside the storage, memory, and processors, because we are dealing with large amount of data, right? So I think that is also something I'm very excited about. And I think if we look at all steps and pipelines, probably such type of systems would be also a good fit.
That's very interesting. And it's also interesting to see how the existing components you have developed can be already applied, probably as is.
Yeah,
There must be some overlap.
Yeah. Hopefully that will be the GPU, the genomic processing unit. Yeah.
Okay. Thank you very much, Nika.
Sure. Okay, now we come to the last talk today, and that is. Okay, it's a bit late. Yeah, we have one more talk. So it's Konstantina. So
yeah, I'll do it fast, guys. And let me make the change here.
Okay, meanwhile, I introduce Konstantina. So she is a postdoctoral researcher at the Safari Research Group at ETH Zurich. She received her PhD degree in electrical and computer engineering in 2023 at National Technical University of Athens and TUA. Her research interests are in the field of computer systems and architectures, heterogeneous computing, and hardware acceleration, targeting mainly genome analysis applications.
Thank you. Sorry, I forgot to use this. Okay, everyone. Thank you for sticking around this long. I'm going to give, I think, a high-level overview of this work. I think it really nice aligns with what Nika has been discussing previously, and it also gives an additional perspective, basically incorporating also some processing in memory into the overall system.
So we're going to see Mars, which is the first processing in memory acceleration effort of raw signal genome analysis inside the storage subsystem. So we're going to first see a brief background to remind ourselves of raw signal genome analysis, move on to the motivation and goal of this work, describe on a high level Mars system, and then show some promising evaluation results. So we've seen a lot so far today about genome sequence analysis and its importance. The study of the genome is very essential to understand the genetic variation, species, and evolution, and has led to some groundbreaking advances in evolutionary biology, outbreak tracing, and personalized medicine. So we've seen so far multiple pipeline and steps of the genome analysis. So usually it begins with two steps, the sequencing and the mapping. During sequencing, we have some sample from an organism that we use as input to a sequencing machine that generates some small fragments of the DNA. Then we use those fragments during the mapping to manage to reconstruct the original genome of the organism and basically find the potential mapping positions of the small fragments on the reference genome. So typically, after the sequencing, we have the base calling step. During the base calling, we leverage deep neural networks that translate the signals to the bases, so ICGTs. So even though this is a very accurate procedure and it manages to eradicate any noise from the raw signals, it is at the same time very costly, power-hungry operation. So lately, there's been some emerging approach that maps signals directly to the reference genome without doing the base calling. This allows for some efficient analysis that can also enable scalability and portability, and even some different type of analysis because raw signals actually contain some rich information. So this work here focuses on raw signal genome analysis to explore the high throughput of modern sequencing technologies and also even achieve real-time analysis. So let's give a high-level overview of the raw signal genome analysis read mapping. It happens in two steps. We have an offline step, which is the indexing, and then the online step, which is the mapping. During the indexing, we first need to transform the reference genome that is stored in databases by using the base domain ICGTs into the raw signals. So typically, we use some sort of sketching mechanism to segment the reference genome into some smaller fragments, and then use some sort of hashing scheme to store these smaller pieces of recurring signals in a hash table. Then we have the mapping procedure that is happening online and has the purpose to actually find potential mapping locations of the core sequence on the reference genome. So let's see here step by step what's happening during the mapping. There are three main steps: the event detection, seeding, and chaining. So in the event detection, we further segment the query sequence into smaller segments of the signal, which are called events. Then we are applying some hashing, the same hashing mechanism that we used when we created the index to these events. And we use these individual events or a combination of them to query the hash table for mapping locations. The matches are then used during the chaining so that we can form some consistent collinear regions that will then depict closely enough the final alignment. And this is heavily relies on some sorting and dynamic programming based operations, so it's particularly expensive. So now that we saw some very high level overview of this algorithm, let's go and see the motivation behind this work. So let's see. So the sequencing throughput is growing rapidly and is expected to continue to grow. And raw signal genome analysis really struggles to keep up with these real-time requirements. So software pipelines are not expected to continue to keep up with it. Therefore, there is a need for acceleration. Typically, we're going to actually try and identify some key performance bottlenecks within this pipeline and accelerate them. For that purpose, we have performed here an experiment using 5 different types of genomes, from smaller ones, so this is D1, to bigger ones, D5. We're going to study the time distribution per stage. So we're looking into how much time the event detection takes, the seeding, as well as the chaining. But we also incorporate here in our study the time it takes to bring data from the SSD, so the I/O data movement overhead. So as we can observe, We consider the chaining and the seeding consistently take up most of the execution time. However, the event detection and the I/O overhead also are not negligible. So in a typical accelerated approach, what we would do is develop accelerators to target and minimize the time that the bottlenecks take, which is the seeding and the chaining. Now, if we do that, we should also see what happens with the remaining steps of the pipeline. Here we have modeled what happens with the I/O when we minimize the computational time of the seeding and chaining. And as we can see, the I/O data movement from the SSD becomes actually now the dominant overhead. So this brings us to the goal of this work, which is to design a high-performance and energy-efficient system for raw signal genome analysis that addresses both the I/O data movement bottleneck, but also the computational ones. So let's see what is MARS. MARS is the first storage processing system for raw signal genome analysis. The key idea is to leverage the storage processing system so that we can address the I/O data movement overhead, but also exploit the inherent acceleration capabilities of the SSD to accelerate the computational expensive steps. So MARS is actually a co-design between some targeted software modifications and some dedicated hardware components that are instantiated within the storage subsystem. The targeted software modifications have the goal to actually manage to adjust raw signal genome analysis to leverage the SSD parallelization capabilities, while the dedicated hardware components are actually some processing in memory, let's say, accelerators that are used by either developing the leveraging the processing using the internal SSD DRAM or processing near the DRAM. So let's see now the modifications that we propose on a software level. These have the main purpose of reducing the computational workload, but also reducing the intermediate storage requirements to manage to fit the entire pipeline within the SSD. So first, we are proposing some filtering techniques that have the purpose to discard the candidates that do not actually lead to valid mapping. And these are main frequency filters, as well as some vote-based filtering, and also some arithmetic conversion that converts, and instead of using floating points, we're using some fixed point with the goal to reduce the memory footprint as well as the computational time that it takes. So the challenge to do this, to apply actually these software techniques, comes when we take into account that raw signals actually have more noise. So we need to be especially careful on where to place them in the pipeline, and of course do some fine-tuning specifically for the filters. Now, before moving on with the accelerated system, we have to make sure that the software modifications that we propose do not actually harm the accuracy. So there we're going to validate this by comparing to a typical pipeline that uses as a base calling Dorado and does read mapping the minimap. We use the same data sets, and then we're going to compare two software variants of our algorithm, which is MARS by using only the filtering techniques, and then filtering plus the arithmetic conversion. We're also using the RoHS 2 as the state-of-the-art baseline for raw signal genome analysis. And we can see that already our MARS raw signal genome analysis modified workflow manages to improve the recall on F1 score over RoHS, as it actually focuses on more useful and informative regions and eliminating some ambiguous ones. Secondly, we can see that by using fixed point arithmetic, we actually don't have any significant losses, which is mainly achieved by making sure we apply some early normalization and quantization before we actually do the rest of the computation. So now let's take a look into the overall system. We're proposing an storage processing system for raw signal genome analysis that operates in two modes. We have the conventional mode. So basically, the SSD performs all the standard SSD storage operations. And then we have the accelerator mode, where the SSD executes the raw signal genome analysis without needing any host intervention rather than except for just initiating the accelerator mode. So let's go one by one for the components. First, we're going to take into a look to the components that are created within the SSD controller. We have the MARS control unit, which is basically a finite state machine that orchestrates all the pipeline execution, controller data flow. Then we have the sorter unit and the merger unit, The sorter unit performs some parallel sorting of sequences using some dedicated logic, while the merger unit manages to combine some sorted subsequences into longer sorted ones. This is basically done by using processing near DRAM, leveraging the DRAM inside the SSD. Now let's go to the internal DRAM of the SSD, where we're going to find two other types of units. This is the arithmetic unit. This one performs some arithmetic and logical operations that are required for raw single genome analysis. This is processing near DRAM again, and it's based on a state-of-the-art Fulcrum architecture that indeed leverages the DRAM subarray proximity to perform some low-latency parallel arithmetic operations. We instantiate 1 per 2 subarrays in this case. And then we have the querying unit. This is meant to perform efficiently in memory hash table lookups for the seeding part. And it's implemented using processing using DRAM. And it's again based on some state-of-the-art architectures, specifically Pluto. So how do we actually transform the entire raw single genome analysis pipeline into something that can be executed on this architecture? We break down raw single genome analysis steps into some very fine-grained tasks that basically correspond to arithmetic, querying, and sorting operations only, which is actually the components that we have created. Therefore, Mars is now a pipeline of only these tasks, and each task is mapped to a dedicated compute unit. So every time we have a multiplication across the entire pipeline, we're going to map it to our arithmetic unit. Lastly, the control unit encodes this pipeline into a finite state machine. We have a predefined, basically fixed execution sequence, and as data flow through, they dynamically trigger the execution of the tasks when the data are ready. So some steps could be actually be bypassed, but overall the sequence is always the same. So some quick distribution demonstration of how the data flows. We start with our raw input signals, which are stored in flash memory, and we transfer them to the SSD internal DRAM to do the event detection in the arithmetic unit. Then we go on to the seeding, which also happens in the arithmetic unit, where we have the hash value generation and the filtering. We do the querying using the processing using DRAM, again within the SSD internal DRAM. And once we have our matches, we have to go on with our chaining. So we create some batches of the sequences so that we can send them to be sorted to the flash controller, to the sorter and merger unit. Once these are sorted, they are returned again to the SSD internal DRAM to continue with the dynamic programming part of the chaining. You can see many more details about these individual components in the paper, and also some more information about how we integrate them in the system. So let's now also quickly see how we evaluated their system. Now we compared against 4 systems for performance and energy analysis. So let's first check the domain that uses base calling. In this case, we have a typical pipeline that consists of base calling with Dorado and Minimap2 for read mapping. And then a hardware accelerated NVM-based pipeline for base calling and read mapping, which is called GenPip. Then for the raw single genome analysis domain, we compare with ROHAS2, which is the state of the art in raw single genome analysis, and it's CPU-based. And a software-only version of Mars to be able to evaluate the impact of our software techniques. And lastly, we have our full system. How now we create these components. For all CPU-based pipelines, we're going to execute them on a state-of-the-art server that has 2 AMD CPUs with 64 physical cores, 1 terabyte DDR4 DRAM. And when we have some parts that can be offloaded to GPU, such as the Dorado, we do it in NVIDIA RTX 6000 CPU. All SSD configurations comprise of 8 channels, each one having 8 flash chips and PCIe 4 interface, as well as an internal DRAM of 4 gigabytes. When we have to create accelerators for the logical components, we use the synopsis, and we emulate everything regarding the SSD operations and the DRAM operations with MQSIM and Cacti 7. So here we can see our results regarding performance evaluation. So as we can see here, Mars outperforms all other baselines across all datasets.
It's really important to notice that it outperforms GenPeep, which is the other hardware accelerated base that we have, and shows that hardware acceleration was not and is not the most fundamental reason why we managed to get performance, but rather the fact that placing this hardware acceleration within the SSD manages to solve fundamentally the IO data movement bottleneck. And lastly, it's also important to say that even only our shorter modifications actually achieve some almost 2 times speedup over OHAS 2. Similarly, we get very good results regarding the energy reduction. And it's again important to notice that it's not just the fact that we have reduced the execution time and thus reduced our energy consumption, but have completely avoided some redundant IO data movement overheads. So therefore, we have again decreased the energy in this manner. So this brings to my conclusion. So we have implemented Mars, which is the first storage processing system for single genome analysis that is enabled through some targeted software modifications as well as dedicated hardware components. We have managed to implement this by preserving the accuracy and improving both performance and energy consumption in comparison to both software and hardware accelerated state-of-the-art tools for raw single genome analysis as well as a typical base calling based mapping baselines. So thank you for your attention.
Thank you so much, Konstantina. So questions?
So you, I guess you emulated the DRAM processing using some of these tools. Did you think about having a cache also along with it? And what kind of cache do you need and how fast and how much it will help?
So we didn't incorporate something like this in our design, but in the paper, there are some ablation studies where we have used many different configurations. So for example, we considered our system only in software, using only processing in memory outside the SSD. So in that case, maybe we have something similar, but no, we have not added caches to this hierarchy.
So it depends upon how much locality you have in the workload.
Yes, so basically in this type of workloads, locality would not actually help a lot because you don't know which area of the genome you're going to target. So it's not enough to do that. For sure.
Yeah, if it is just more or less a stream processing, then cache will not. Yes. Okay, thank you.
Thank you.
Are there more questions? So one very quick one. Konstantina, if we have, let's say, base calling very fast, do you still suggest raw signal analysis?
Yes, so basically, even though raw signal genome analysis is important in the sense of the computational side that can speed things up, however, you can still find some value in it to enable some novel type of analysis, which we have yet to see a lot of things. So I think we should move forward to moving both, even in some sort of synergy to get the most out of it.
Right, like basically some sort of data that otherwise would be lost if we analyze it.
Exactly.
Makes sense, yeah.
Thank you.
Great. Thank you so much, Konstantina. And yeah, the bottle was also excited. Thank you so much, everyone, for this third edition of Arch4Health. We also look forward to the next iterations. We will post about them soon. It was great to have you all, and we hope to stay in touch.
Thank you, everyone.
Great.
Hopefully we can take a picture later then.
Yes.
Right? Just a moment. Let me check. Professor Kant, did you get your email? Will you be able to attend the dinner?
I think I should be.
Good, good. I think it's close enough. Is it close to your hotel also? It should be like 10 minutes from here.
Oh.
Did you open it?
We open.
Ah, cool, cool.
It's not always easy.
Yes,
There are two current live screen. Wait, no. Okay, Konstantina.
Are you coming to the end?
Like, you should ask him for the hard stuff if he doesn't.
Oh yeah. If you don't want to do